So let's say you found a skeleton. How would you tell it's a dinosaur? You personally, how would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
Hey there YouTube, the Dapper Dinosaur here. Today I'm answering 15 questions for evolutionists. But first,
let us pray. Our Darwin, who art in the British Museum, hallowed be thy name. Thy evolution come, thy selection be done, on land as it is in water. Give us this day our daily sexual selection as we sexually select, and lead us not into inbreeding, but deliver us from extinction. Amen.
okay, now that we've opened an evolutionist prayer, let's talk about what we're here to do. Answers Canada is, as the name suggests, the Canadian arm of Answers in Genesis. The ministry led at the time of this recording by an elderly chimpanzee with thinning hair named Ken Ham. This video was uploaded 6 years ago, but it seems to be a re-upload of an even older video, and Calvin Smith, who will be on the right in this video, seems to be even more than a decade younger than he seems now. Anyway, they say they have 15 questions. Let's see if I have answers.
Welcome to Genesis Unleashed, the show where we unleash the truth of Genesis.
Welcome to Dapper Dinosaur, where I have yet to see anything to indicate that anything in the book of Genesis is historically reliable or even plausible.
And pose 15 questions evolutionists have a hard time explaining.
I suspect that I won't really have that hard of a time, but hey, I've been wrong before.
Welcome to Genesis Unleashed. On this program, we're going to look at, once again, the Question Evolution Flyer, and we're on question number 3.
Oh, so probably not 15 questions then. Okay. Sorry? Oh, and also, welcome to Dapper Dinosaur, where I have yet to see anything to indicate that anything in the book of Genesis is historically reliable or even plausible.
Right. Question number 3. So I'm just going to read it out of the flyer here. How could mutations, accidental copying mistakes, in brackets DNA letters, exchanged, deleted, or added, genes duplicated, chromosome inversions, etc., create the huge volumes of information in the DNA of living things? Right.
Each mutation alters the genome, and many of them, such as duplications and insertions, make it bigger. So that will increase information, and since any genetic sequence can be reached by mutation, there's no particular barrier to getting extant genomes from previous ancestral genomes. The real question is, how could mutation not be able to generate the genomes of today? What would prevent it?
That is question number 3.
It sure is.
Right, and then there's some additional details. How could such errors create 3 billion letters of DNA information to change a microbe into a microbiologist?
Well, first, a microbe has never turned into a microbiologist, unless the microbe in question was the zygote that grew into the microbiologist. A population of microbes evolved into a population of humans, as well as all other animals. And the population of all animals includes a few microbiologists, although almost none as a proportion of all living animals. But the biggest way is whole genome duplication, which has occurred a few times in the history of most animals. This creates redundant genes which allow one copy to evolve a new function while the other copy continues on with the old function, or to develop complementary functions, or even in some cases to both break in certain ways such that they now only function together as a set, which creationists generally will then pretend is a sign of amazing complexity indicating design, even though it's a stupid thing that no designer with have a brain would ever do. Is this whole discussion about this question just going to be different ways to ask, how could a process that produces changes and additions to the genome have changed and added to the genome? I thought we were supposed to be answering questions that are hard to answer. Maybe I misunderstood the point of this whole thing.
There is information on how to make proteins and also for controlling their use, much like a cookbook contains the ingredients as well as instructions for how and when to use them.
That's true.
Without one or the other, one without the other is useless.
Are you sure about that?
You see, the thing is that while that's true for modern life, that's not necessarily true for the earliest life. It's quite probable that the earliest life didn't actually have regulatory mechanisms for its protein synthesis. It probably just made proteins more or less at random. And yeah, sure, that's fabulously inefficient. But that's why if any mutation allows for better regulation of gene expression, it will be selected for. The way things work in biology now is neither the only way they could work, nor is it likely how biology worked in the deep past a few billion years ago. Some mutations started before regulation was probably much of a thing.
See, for example, creation.com/meta information for more details on that particular dilemma.
Right.
Homework? Lame. But okay, let's go through this article, which is read by Phil Rivara, a longtime supporter of the channel. So if you like this section, make sure to thank him in the comments if you appreciate the audio work he did. And also, remember that when I point out how dumb this article is, I'm speaking about the author, not Phil Rivara, who is not responsible for the composition of any of these words.
Evolutionists have never been able to give a satisfactory answer to the problem of where new information comes from.
Well, first, drink for evolutionists.
Let us pray. Our Darwin, who art in the British Museum, hallowed be thy name. Thy evolution come, thy selection be done, on land as it is in water. Give us this day our daily sexual selection as we sexually select, and lead us not into inbreeding, but deliver us from extinction. Amen.
Second, it would really help if the term information could be defined. You'll see that nowhere in this whole article does that happen. As a result, I will simply continue with the definition that the information in the DNA is literally the DNA sequence, that you can take the number of nucleotide base pairs, multiply it by 2, and that's about how much information in bits is in a genome. So for a human, that's 12.8 billion bits.
That evolution requires for turning a microbe into a mycomycete or a maze-mastering mammal.
I appreciate the alliteration.
Their best guess is gene duplication, which gives them an extra length of DNA.
Which is indeed new information. Speaking of information, YouTube clearly doesn't have the information that people like these videos. Please help them out by hitting like on this video, subscribing if you haven't already subscribed, and sharing the video. If there's a hype button on this video, why not hit that too? YouTube has been stingy of late, and I'd like to see that turn around, because I want to keep this channel running until I absolutely can't anymore.
But it contains no new information.
Yes, it does. Because before that, the genome doesn't have that stretch of DNA, and now it does. If I duplicate a chapter in a book, that book has new information. That it's a replica of information elsewhere in the book doesn't change that. That you might not want to read it doesn't change that. And of course, DNA isn't a book anyway, so don't take this analogy too seriously.
Followed by random mutations that are supposed to turn the duplicated information into something new and useful.
As has been observed in notothenioid icefish, bacteria in the Lenski lab, and in the wild in Anthrobacter colonies near a nylon plant.
They have no direct experimental evidence for this claim.
Except for what I just mentioned, and that was just a sample. Of course, what is direct evidence? It's just a weasel word that lets the creationists dispute any evidence by calling it indirect. Just like historical versus observational science, this is not a real distinction that scientists care about. Science is about hypothesis testing, and we can test for the presence of genes derived from duplication events, and we get statistically significant evidence sufficient to show that that is by far the most likely source of some genes. Whining that the evidence isn't direct means nothing.
And there's much against it.
Citation needed, amigo.
So they have to rely on indirect evidence, such as the so-called gene families.
Looking directly at genetic sequences is indirect evidence now. See what I mean? It's just a weasel word.
Some genes are similar in both structure and function to other genes.
No, it's sequence, and they don't even have to be similar in function. That's the cool part. Now, nucleotides are arranged into groups of 3 called codons, and because there are 4 nucleotide types, the number of codons is 4 to the 3rd power, or 64. But there are only 21 functions assigned to codons, which means that there is a huge overlap. So we can tell the difference between neutral variation, such as, say, varying between the 6 codons that all code for the amino acid leucine, and functional variation, such as, say, varying which amino acid is being coded for at a particular locus. This means that even with identical proteins, we can use neutral variation to detect common ancestry, because the only way to share a nested pattern within neutral variation is either a trickster god who wants us to think common ancestry is true, or common ancestry being true. Some creationists seem to be cool with having Loki as their god. I'm not, so I reject that option on philosophical grounds, the same grounds by which I reject last Thursdayism.
And evolutionists point to these and say they originated by chance, copying, and mutation from some common ancestral gene.
Yes, because we know what genetic descent looks like on the molecular level, and these gene families bear all the hallmarks of that. Creationists have yet to produce a better alternative that can actually explain the data, or even just an alternative at all that produces testable predictions and explains any of the data. At least not one that hasn't already been falsified, usually before it's even proposed.
But this is just evolutionary speculation.
No, it's a set of testable hypotheses that keep not being falsified. Also, imagine being the guy who thinks that all organisms were created by magic, then most of them were killed in a magical flood that violates more or less every law of physics, and then whining about speculation.
It is not experimental evidence.
Yeah, because sequencing genomes and doing statistics isn't an experiment, right? Of course, that's also why there's the Lenski long-term E. coli experiment, which watched not just a new gene, but a whole new metabolic pathway evolve in real time. But hey, creationists lie about that. So they just pretend that that's not what happened, even though it is.
The globin gene family is a favorite example.
Maybe it is, but I have only really heard about it from creationists. Not that this isn't a real thing.
Hemoglobin carries oxygen in our blood and can be made up of different combinations of different kind of globin proteins.
That's true.
For example, hemoglobin in human fetal blood contains a different combination of globins to that in postnatal blood. Evolutionists claim this resulted from an original globin molecule that duplicated in an early blood-using animal.
No, it would be an early globin gene, not a globin molecule itself. The globin protein can't mutate and pass down changes. That happens in the DNA. The gene coded for this early ancestral globin protein, but the globin itself left no descendants, because that's not how proteins work. By the way, if you're enjoying this video, hit like and consider that if you had joined my Patreon, you could have seen this months ago. Unless you're actually watching it early, in which case, thanks for the support. My Patreon gets you access to videos like this months before they come out, with often more than a dozen finished videos ready for you to watch right now. You also get access to the supporters-only Discord server, and it helps keep this channel going. Without my patrons and channel members, this channel wouldn't be able to exist.
And mutated to form a family of different kinds of globins, which then allowed the diversification of complexity and oxygen-using processes that we see in the animal world today. But this example is far better explained by intelligent design.
Well, maybe, except that we have no mechanism for intelligent design. We've never seen anyone but a human intelligently design any genetic information, and no one proposes that the human globin complex was designed by humans, and the globin genes share neutral variation in a nested pattern that is characteristic of common descent, but not common design. So, no. Intelligent design is a worse explanation in every way. It's just that it's the explanation that the author, Alex Williams, is emotionally invested in.
The human baby in his or her mother's womb has to compete for oxygen in its mother's blood supply with a demand for oxygen from two other sources, the placenta that feeds it and from the mother's womb that surrounds it. So the fetal hemoglobin has to have, amongst other things, a higher affinity for oxygen than the mother's hemoglobin. In contrast, when the baby is born and can draw oxygen from the air in its own lungs, it no longer has any competition, so it requires a different kind of oxygen uptake system.
And? What about this as a problem for evolution?
A wise creator would ensure that the hemoglobin could change its form and function to cater for these very different conditions.
Wow, that's a mighty nice claim Alex has there. Too bad it has literally no argumentation other than that's what he thinks. Maybe that's what a designer would do, or maybe it's not. But the fact is that this need for different globin isn't universal and probably goes back not to the beginning of humans, but to placental mammals, who are the only organisms with this challenge. The problem is that the evolution of placental development started with babies being born far earlier than in modern placental lineages. We can see this from the level of development at birth or hatching of the other two branches of mammals, the monotremes and marsupials. They are born when extremely small, and so at no point are they a huge drain on oxygen. But as the gestational term of placental mammals starts to get longer and longer, this produces a selection pressure for higher oxygen affinity in hemoglobin during embryological development. Creationists often assume that because a group of organisms needs a structure or gene or whatever now, that all their ancestors similarly always needed it. But that's basically never true. And the resolution to conundrums about how it could have evolved are often resolved simply by recognizing that they were not always needed, and that the advantage they provide was realized and required gradually. And since the argument is that it couldn't have evolved, all that needs to be shown is even one plausible pathway for the evolution of the feature. It doesn't even need to be the actual historical pathway, because the argument is about possibility, not actuality.
And integrate this change into the other vast complexities of the almost miraculous reproductive process.
Regulatory systems evolve in tandem with genes because they too affect fitness. It's not really more complicated than that. Creationists are just making up problems that don't really exist.
The idea that such complex interactive changes could all occur by chance is rather hard to accept.
Argument from incredulity. I don't care how hard it is for a creationist to accept something. It's hard for some people to accept that Joe Biden won the 2020 presidential election or that Donald Trump won the 2024 kingship election. That doesn't change the fact that they did.
Information about information. But the problem of information origin in biology is far bigger than most people realize.
Well, since as far as most people can tell, including all the experts, there is no such problem. I suppose it would have to be bigger than people realized if it were to exist at all.
Information by itself is useless unless the cell knows how to use it.
The cell doesn't know anything, so that can't be literally true. Or is this a creationist doing that thing that they always complain about when normal people do it? You know, using anthropomorphic language about a non-human entity for ease of communication, while keeping in mind that the cell doesn't actually know anything? But no, surely not. That would be hypocritical. And we know that a good Christian would never do that, right?
Evolution not only requires new information, but it also requires extra new information about how to use that new information.
So regulatory regions, is that it? Just regulatory regions? The thing that are already subject to mutation, drift, and selection, just like the protein coding regions? Why would that be some big problem over and above the evolution of genes? There is no real reason, but since creationists love just making things up, we're just inventing an extra problem here.
Information about information is called meta-information.
Fun fact, meta-information is a less common synonym for metadata, which isn't actually what regulatory regions are, so already we're getting this wrong. Metadata gives information about things like the location, size, type, modification date, origin date, and author of data. The genome does not actually encode this. At best, we can infer it from multiple genomes using statistical analysis, but the DNA doesn't store any metadata directly.
We can see how it works in making a cake. If you want to make a cake, you need a recipe that contains A, a list of ingredients, and B, instructions on how to mix and cook the ingredients to produce the desired outcome.
Yeah, that's all information. The meta-information would be things like the copyright date, the author, the publication, the page number of the recipe, and stuff like that. Creationists really struggle to get even the smallest things right, don't they?
The list of ingredients is the primary information, and the instructions on what to do with the ingredients is the meta-information.
Pro tip, primary usually contrasts with other terms derived from Latin ordinal numbers, such as secondary, tertiary, quaternary, quinary, centary, septenary, octenary, nonary, and denary. Plus, since primary is Latin, contrasting it with meta, which is Greek, is just bad form. Oddly enough, this whole section would be far less stupid and wrong if the regulatory regions of the genome were called secondary information rather than meta-information.
The human genome contains an enormous amount of information, far more than we ever, until recently, imagined.
I mean, it's like 1.6 gigabytes if my math is correct.
But we now know that most of it is not primary information, protein coding genes, but meta-information.
Well, except for the fact that Alex is wrong about what meta-information is. It is true that most of the genome has nothing to do with protein coding, and more of it is regulatory than is protein coding, although it's still mostly junk that is non-functional.
The information that cells need to have in order to return those protein coding genes into a functional human being and maintain and reproduce that functional being. This meta-information is stored and used in a variety of ways. DNA consists of a double helix, two long chain molecules twisted around one another.
That's true.
Each strand consists of a chain of 4 different kinds of nucleotide molecules. The shorthand symbols are T, A, G, and C.
Since he isn't telling, I will. That's thymine, adenine, guanine, and cytosine. Never give abbreviations for things without first giving them their full name in a text. It's bad form.
Bad form!
With a few exceptions.
About 3% of this in humans consists of protein-coding genes.
I usually hear it as a 2%, but I don't think it actually matters to this discussion, so I don't really care to check.
And the other 97% appears to be regulatory meta-information.
Wrong.
In fact, this might be the most wrong statement in this whole article. The vast majority of the genome is neither regulatory nor coding. It is in fact functionless junk. Regulating coding genes requires sequence specificity, which means that mutations to those regions will be affected by natural selection, which means that they will have a lower variability across the population. Whereas if a region has high variability, that means that selection is basically not working there, and so that's why it can accumulate these changes. Most of the genome is at a relatively high variability in humans, which means that most of it is not under selection pressure, which further means that most of it is not sequence constrained, which means most of it is neither protein coding nor regulatory, since both of those are sequence constrained functions. This isn't hypothetical, this is directly testable and mathematically certain to the degree that anything in science can be. The idea that all the non-coding regions are regulatory in some way is just a lie. I don't know if Alex is lying, since I don't know how much he actually knows, but the statement is a lie. We also know this because we can do knockout tests in animals with very similar genomes. Mice, for example, have a very similar genome to primates, including humans, because rodents and lagomorphs together make up the sister group to primates and colugos. Mice can have huge chunks of their genome simply removed, and they go on to live happy, healthy mouse lives and even reproduce, which results in happy, healthy pups. If those regions were so vital, that wouldn't be the case.
DNA is an information storage molecule, like a closed book. This stored information is put to use by being copied into RNA molecules, and the RNA molecules put the DNA information into action in the cell. For every molecule of protein-coding RNA, primary information, there are about 50 molecules of regulatory RNA, meta-information.
No, there aren't. That number was derived by just using the rough ratio of coding DNA to non-coding DNA and assuming all of it gets transcribed, and that all of it that is transcribed is functional. We know from experiment and observation that neither of those assumptions are valid.
Down the sides of the DNA double helix, several different kinds of chemical chains are attached in patterns that code meta-information.
Thanks, Captain Vagueness. That was really helpful.
For turning unspecialized embryonic stem cells into the specialized cells that are needed in fingers, feet, toenails, and tailbones, etc.
Okay.
DNA is a very long, thin molecule.
I feel like he already said that.
If we unwound one set of human chromosomes, the DNA would be about 2 meters long. To pack it up into the very tiny nucleus inside the very tiny human cell, it is coiled up in 4 different levels of chromatin structure into 46 chromosomes. This coiling chromatin structure also contains yet further levels of meta-information.
That's actually kind of true, in that this folding will actually allow or disallow access to certain parts of the genome via the binding of histone proteins, which does function kind of like metadata for the DNA. It's not exactly the same thing, since metadata is about ease of retrieval, not access restriction, but it's close enough that I won't really nitpick.
The first level, the histone code, codes information about the cell's history, i.e., it is a cell memory.
Kind of, and sometimes. That's how a lot of epigenetic transformations work. Certain conditions cause histones to bind to certain sites or unbind, which will then activate or deactivate regions of the genome in response to environmental stimuli. But calling that a memory is a pretty big stretch. But you know who does remember some cool stuff? My channel members. They get to remember videos months before they go public. So why not join the channel? It starts at only $1.99 a month, and in addition to early access, you get a cool green name in chat, a channel badge, access to members-only emojis, some members-only videos, and access to my supporters-only Discord. Plus, it helps keep this channel going, since YouTube is very fickle about views, subscribers, and ad money. Help a dinosaur out if you can. And of course, if you can't, remember to hit like on this video and share it with your friends, enemies, and just randos.
The 3 further levels of coiling code further information, some of which is described below, and there's no doubt more that we have yet to unravel. The amount of meta-information in the human genome is thus truly enormous compared with the amount of primary gene coding information.
Oh wow, that means that we must have a way that Alex will accept to measure it, and he must be able to give us numbers, right? Oh, he doesn't have numbers? Is that because he still hasn't actually defined information, and so he has no way to actually measure it and give us numbers and units? You know, the most basic thing you can do in science? Yep, that's exactly why. Because once a creationist defines information, it becomes trivially easy to point out that all of their claims about it are absurd and in no way problems for the theory of evolution. Cognitive dissonance sure is a hell of a drug, I guess.
Self-replicating molecules?
Yes?
In this monumental work, The Ancestor's Tale, Richard Dawkins traced the supposed ancestry of humanity back through all the evolutionary ages to the very first supposed common ancestor of all life.
It's been a while, but I remember him doing the last universal common ancestor, Luca. But I could well be misremembering, so whatever. If you've read it and remember if he went back to the first universal common ancestor, Fuca, I suppose? Let me know in the comments. I'm not going to dig out my copy of the book to check.
He supposed this original ancestor to have been an RNA type of life form, although he admitted ignorance of the precise details.
Oh, so he was being a good scientist and not claiming to know things for which we have no data or insufficient data. Creationists could, in this case, stand to be more like Dawkins. Of course, in many ways, they're already far too much like him when it comes to bigotry, so I should be careful what I wish for.
His choice of an original RNA life form is well-founded because RNA is the only known molecule that can do all of the 3 basic functions of life. A. Store coded information. B. Combine with itself and other RNAs to create molecular machines. And C. Self-replicate, but only in a very limited manner, under very special circumstances.
I guess metabolism has been yeeted out of the list of the most basic life functions. But yeah, Alex is basically right here. These are why RNA is usually thought of as the basis for the first self-replicating chemical systems.
However, recent studies showing how living cells actually replicate have made this RNA world concept ludicrously unrealistic.
Not scientifically possible!
And it's not scientifically possible because modern cells are almost nothing like the first RNA-based self-replicators. In fact, the first RNA life probably wasn't even made of cells. RNA self-replicating systems that have been created in labs sure aren't made of cells. It's like saying Proto-Indo-European couldn't have existed because surveys of modern English, Albanian, and Hindi have determined that they don't actually work like Proto-Indo-European is supposed to have worked. And yeah, they don't, because that's kind of the point of this evolution thing that happens both in living organisms and in languages.
A central problem in cell division, that is, what living cells actually do, as opposed to Dawkins' imagined self-replication.
Okay, so what? Something Dawkins wasn't talking about does. Cool. So this will have absolutely no bearing on the plausibility of RNA world. Neat.
Is that large proportions of the whole genome is required for the normal operation of the cell, probably at least 50%, in unspecialized body cells and up to 70 to 80% in complex liver and brain cells.
Well, since at most about 20% of the genome is functional, that was just a lie.
When it comes time for a cell to divide, not only does the DNA have to continue to sustain normal cell operations, it also has to sustain the extra activity associated with cell division.
It sure does, and I don't know why that's relevant. In fact, I know it's not, because Dawkins' RNA self-replicators aren't cells. They don't even have metabolism in the way we know of it now.
This creates a huge logistic problem. How to avoid clashes between the transcription machinery, which needs to continually copy information for ongoing use in the cell, and the replication machinery, which needs to unzip the whole DNA double helix and replicate a zipped copy back onto each of the separated strands.
Do you think that the cells ever actually activate their copies of WinRAR that they're using to zip and unzip all this DNA? I doubt it. I sure never activated my copy.
The cell's solution to this logistic nightmare is truly astonishing. Replication does not begin at one point, but at thousands of different points. But of these thousands of potential start points, only a subset are used in any one cell cycle. Different subsets are used at different times and places.
Which isn't relevant to small replicating chains of RNA. It is neat though, so there's that.
Can you see how this might solve the logistics problem?
Absolutely, it solves the logistics problems for modern cells, a problem that the first self-replicators did not have because they were radically smaller, simpler, and didn't have a metabolism in the way we understand it from biology today.
A full understanding is yet to emerge, because the system is so complex.
As predicted by both population genetics and thermodynamics. By the way, both of which favor increased complexity over time in biology.
However, some progress has been made. The large set of potential replication start site is not essential, but optional.
Wouldn't that remove one of the main objections to it evolving? I honestly don't know what this part of the article is trying to demonstrate. I think it's just look at the trees, but in this case, it's look at the mitosis.
In early embryogenesis, for example, before any transcription begins, the whole genome replicates numerous times without any reference to the special set of potential start sites.
Probably something to do with the fact that the earliest cells in embryogenesis are pluripotent stem cells. Pluripotent stem cells can differentiate ultimately into any cell type, and are also being investigated for their medical potential in healing wounds, and someday perhaps even regrowing lost appendages.
The pattern of replication in the late embryo and adult is tissue-specific.
That makes sense since each tissue has different processes it needs to maintain for as long as possible. But does Creation Ministries International think that precellular life had tissues? That seems far-fetched. Not even all animals have tissues. For example, sea sponges, or to give them their scientific name, poriferans, don't have tissues, although they do have cell types.
This suggests that cells in a particular tissue cooperate by coordinating replication so that while part of the DNA in one cell is being replicated, the corresponding part in a neighboring cell is being transcribed.
Seems plausible, and hey, that also seems like the kind of research that a creationist could do. Get on it, creationists. Do some good in the world instead of spending all your energy trying to drag science back centuries in service to racism, sexism, queerphobia, and fascism. You can do it. Who am I kidding? You probably can't and don't want to. I mean, you, at the time of my writing this, are presiding over the vultures feasting at the corpse of the United States of America, not realizing that it is, in fact, your own corpse, and that unless things change, it's only a matter of time before you are the ones devoured by your own policy choices.
Transcripts, thus, can be shared so that normal functions can be maintained throughout the tissue while different parts of the DNA are being replicated.
Look, I'm talking to Alex now. Alex, if you ever see this, I'm writing this early in the morning, a morning when I woke up not feeling the best, a morning where just getting coffee in me so I can write coherently has been its own struggle, a morning after realizing that I had to throw out a paintbrush, a morning after finding out that despite a local store having a store brand for a product I need, they don't actually carry it. A morning in which I'm worried about my friends and loved ones who are the targets of a secret police force wearing masks and kidnapping whomever they want off the streets of the city I live in. For the love of all that is holy, get to a fucking point! I do not have the patience for this shit. No one is disputing that cell replication is neat, but what in the fucking nine hells of Beator does that have to do with Dawkins' RNA replicators, Or is it just a tangent when we're talking about meta-information being a problem for evolution, or it supposedly being some challenge for evolution that there is information in DNA? Help me put it together.
DNA that is transcribed early in the cell division cycle is also replicated in the early stage, but the transcription and replication machines are carefully kept apart. The early transcribed DNA is that which is needed most often in cell function. The correlation between transcription and replication in this early phase allows the cell to minimize the downtime in transcription of the most urgent supplies, while replication takes place. There is a pecking order of control. Preparation for replication may take place at thousands of different locations. But once replication does begin at a particular site, it suppresses replication at nearby sites, so that only one copy of the DNA is made. If transcription happens to occur nearby, replication is suppressed until transcription is completed. This clearly demonstrates that keeping the cell alive and functioning properly takes precedence over cell division.
Yeah, that sounds like a strong selection pressure. There's not much point in dividing a cell into new cells if those cells are going to die in the process. This sounds like a problem that was probably solved before the last universal common ancestor even existed. And also after the rise of cellular life, after Dawkins' RNA replicators that I'll remind you, we're still supposed to be talking about.
There's a built-in error correction system called the cell cycle checkpoints. If replication proceeds without any problems, correction is not needed. However, if too many replication events occur at once, the potential for conflict between transcription and regulation increases, and/or it may indicate that some replicators have stalled because of errors. Once the threshold number is exceeded, the checkpoint system is activated. The whole process is slowed down and errors are corrected. If too much damage occurs, the daughter cell will be mutant, or the cell's self-destruct mechanism, the apoptosome, will be activated to dismantle the cell and recycle its components. An obvious benefit of the pattern of replication initiation being never the same from one cell division to the next is that it minimizes the effect of any errors that are not corrected. Such a magnificent solution to such a monster logistics problem could only have come from a master designer.
Nuh-uh. Seriously, that's my response. They made no argument, so I need no argument to disagree. But what about this requires a designer? I don't know. Why couldn't it evolve? Alex doesn't tell us. I really don't know what argument this article is even making. I mean, I get that they're trying to say that there's so much cool information in a genome that it had to be a miracle, but nothing about there being information is a barrier to evolution. Also, starting off by getting what meta-information wrong isn't helping.
The impossible conundrum. Now comes the impossible conundrum.
For me, the impossible conundrum is what the hell any of this has to do with anything.
Keeping in mind the cake analogy.
The cake analogy based on knowing what meta-information is? Okay.
Let's recall that the vast majority of information in humans is not ingredient-level information, or code for proteins, but meta-information.
No, although more of it is regulatory than protein coding, so with that understanding, let's see what they have for us.
Instructions for using the ingredients to make, maintain, and reproduce functional human beings.
Right, so regulatory and developmental functions, not meta-information.
Evolutionists say that all this information arose by random mutations, but this is not possible.
Ooh, I think we're finally going to get to an argument after so much waffling on about nothing.
Random events are, by definition, independent of one another, but meta-information is, by definition, totally dependent upon the information to which it relates.
And gene regulatory sequences are in fact independent of what they regulate. If you take a promoter and stick it onto a new gene, it will promote that gene just fine. That's what happened with the Lenski E. coli experiment. It happens in evolution all the time, and there have been experiments in which scientists have replaced promoters with literal random DNA and found that a significant fraction of those sequences functioned as promoters, some better than the wild type, and a huge proportion of them functioned as promoters after only 1 or 2 mutations. So not only can these sequences evolve, even random sequences can sometimes function as regulatory sequences, and because they don't actually function like a cookbook, they can regulate whatever right off the bat. It just depends what gene is downstream that they could regulate.
It would be quite nonsensical to take the cooking instructions for making a cake and apply them to the assembly of, say, a child's plastic toy.
It sure would, but since DNA isn't the cookbook or the assembly instructions for my cool Dino Megazord that can break apart into the constituent Dinozords, of which only 2 of 5 are dinosaurs, I don't know what the point is.
If nothing else, the baking stage would reduce the toy to a mangled mess.
That depends on the toy.
Cake cooking instructions only have meaning when applied to cake making ingredients.
Something we can demonstrate does not carry over to DNA and protein regulation, so this is pointless.
So too, the logistics solution to the cell division problem is only relevant to the problem of cell division.
Yeah, that's how solutions usually work.
If we applied the logistics solution to the problem of mate attraction via pheromones, scent, in moths, it would not work.
I don't think that even means anything. What would it look like to even do that? If you have an idea, tell me in the comments, but I think we're basically asking, what if this article smelled like purple?
All the vast amount of meta-information in human beings only has meaning when applied to the gene content of the human genome.
All of the zero meta-information, from what I can tell.
Even if we granted that the first biological information came into existence by a random process, the meta-information needed to use that information could not come into existence by the same random, independent process because meta-information is inexorably dependent upon the information that it relates to.
Except that it does, in real time, in labs and in the wild, at least for the wrong definition of meta-information that this article is using. Even completely random sequences can regulate genes. We know this from testing it directly. This isn't even the so-called historical science that creationists love to complain about. This is directly observable.
There's thus no possible random mutation solution to this conundrum.
You serious?
Can natural selection save the day?
Yes, to the extent that the day needs saving. But let's hear stupid reasons why Alex thinks that the answer is no.
No. There are at least 100, and probably many more, bits of meta-information in the human genome for every bit of primary, protein coding gene, information.
I've already addressed this. That's not even close to true. At a maximum, it's more like 18 and probably much lower.
An organism that has to manufacture, maintain, and drag around with it a mountain of useless information while waiting for a chance correlation of relevance to occur so that something useful can happen is an organism that natural selection is going to select against, not favor.
That's true, but no one is suggesting that any such organism ever existed or needed to exist. The need for regulatory systems for genes increases as the complexity of the organism increases. And also, even when it's not strictly necessary, if it improves energy efficiency, it can be selected for even before the presence of such regulation is vital to survival. Evolution does not work by evolving a fully functional multicellular animal, but it just doesn't have regulatory sequences. This is basically the same objection as when creationists ask which evolved first, the blood, the heart, or the blood vessels. And the answer is kind of the blood? The fuller answer is that all of them evolved in tandem as the size of the organism and the complexity of the oxygenation requirements of a lineage increased. The earliest animals didn't need blood, hearts, or blood vessels, and many animals still don't bother with any of them. Similarly, The earliest life didn't have or need regulatory regions in the genome.
Moreover, an organism that can survive long enough to accumulate a mountain of useless information is an organism that does not need useless information.
And yet organisms routinely have useless information because the accumulation of it is more of a byproduct of genetic drift than it is a result of selection.
It must already have all the information it needs to survive.
Well, yeah, that's obvious. But evolution doesn't require organisms that couldn't survive to have existed.
What kind of organism already has the information it needs to survive? There's only one answer.
All of them that survive. And for some of them, that amount of information isn't very large.
An organism that was designed in the beginning with all that it needs to survive.
Non sequitur. Your facts are uncoordinated.
Mutations are known for their destructive effects.
They were often harmful, yes, but not always. There have been many documented neutral and helpful mutations,
including over 1,000 human diseases such as hemophilia. Rarely are they even helpful.
Rarely is all evolution needs. Even if they're rare, when they occur, they can be selected for. Think about it with dice. I'm a nerd, so I have a lot of d20s. Now, rolling a 20 is a rare occurrence. And if I wanted to collect 100 rolls of 20, it would take a long time. But what if every time I do roll a 20, which is rare, I get to set it aside and only roll the dice that did roll a 20? Well, suddenly, I've gone from needing the rest of my life to get all the 20s and then still failing, to needing only an afternoon to do it.
But how can scrambling existing DNA information create a new biochemical pathway or nanomachines with many components to make goo-to-you evolution possible?
Because the genome of any extant organism is made of particular character states for all loci in the genome, and because the state for the system exists in sequence space, and mutations cover all of sequence space. In other words, there is no genome that cannot be derived by mutations. And while a truly random walk would be unlikely to arrive at any kind of functional state for such a system, a partially guided and partially random walk that favors functionality is guaranteed to find such states for the system.
For example, how did the 32-component rotary motor like ATP synthase, which produces the energy currency of the cell ATP for all life, or robots like Kinesin, a postman, a delivery man essentially, that delivers parcels inside cells,
We'll
originate?
talk about him.
There are many papers on the evolution of both. I'm not going to go over every single step here, because nothing about these structures was even proposed as a barrier to evolution. So if these guys are curious about it, they could just go read up on it, as could you. But I'm not going to go on a long biochemical journey to counter absolutely no argument other than argument from incredulity. The fact that these bozos haven't read up on the literature isn't a good reason to reject science.
And there's another web link in the article, we'll put it up as well, creation.com/train.
Well, that URL no longer works, but this video is from a decade ago, so I won't fault them for that. It's hard to keep URLs valid for that long unless they're like homepage URLs. Anyway, I linked it in the description.
Where Dr. Carl Whelan talks about these changes in living things, they're taking things in the wrong direction.
Right.
They're not getting better, they're getting worse. And that's really the answer to, we'll go into more detail here,
Yeah.
but that's the answer to the question.
Wouldn't it suck for these guys if we could experimentally demonstrate sustained fitness gain in populations over time in some kind of famous biological experiment? Too bad for the scientists that we can't, I guess. Hey there, YouTube, the Dapper Dinosaur here. Last time we were watching Creation Ministries International explaining a pamphlet full of questions for non-science deniers, and they sent us to an article by Alex Williams called Meta-Information on Creation.com. Now they've sent us to read the article Variation, Information, and the Created Kind by Dr. Carl Whelan. Carl is, of course, not a scientist, but a medical doctor. This, of course, means he has no credentials to talk about evolutionary biology whatsoever. Apparently, he's going to tell us how mutations can't improve an organism, even though we've directly observed that happening. Now, the original URL given in the video was no longer functional, and they don't name the article. However, the article I found does talk about this, and so I'm like 70% confident it's the one the hosts want me to look at. Once again, Phil Rivera has lent his voice to this project, and I can't thank him enough. Anyway, let's see what Carl has for us.
All observed biological changes involve only conservation or decay of the underlying genetic information.
Right off the bat with a lie, and one involving a word that will never be defined. No matter how we define information, as long as it's actually measurable, new information arises. If we define it as just the DNA sequence, if we define it as the number of genes, if we define it as what contributes to fitness, all of them result in humans having observed new information arising through evolution both in the wild and in the lab.
Thus, we do not observe any sort of evolution in the sense in which the word is generally understood.
I'm not going to speak to a general understanding. This is an article about a scientific question, and so should use the scientific definitions for terms. The process of evolution is simply a change in allele frequency in a population through successive generations. If that happens, then evolution has happened. It doesn't matter if you don't like it. It doesn't matter if it doesn't fit whatever definition you have in your head. It doesn't matter if you don't understand how it works. It doesn't matter if you don't think that the process can explain modern biodiversity. And it doesn't even matter if it really does explain modern biodiversity. It's still evolution. Now, the theory of evolution is the collection of ideas about how this change proceeds, how to predict it, how it results in evident biodiversity, and the model of universal common ancestry.
For reasons of logic, practicality, and strategy, it is suggested that we 1. Avoid the use of the term microevolution.
Fine by me, I don't think it's a particularly useful term.
2. Rethink our use of the whole concept of variation within kind.
To be fair, it does need a rethink, since kinds are not real in any observable manner. While I contend that that's true of species too, kinds need to be objectively real in young Earth creationism, whereas species do not in evolutionary biology.
3. Avoid taxonomic definitions of the created kind in favor of one which is overtly axiomatic.
So avoid having a testable hypothesis. Instead, just assume that you're right. Well, I guess Carl is just completely abandoning science. Good that he's being open about that, at least.
Most popular literature on evolution more or less implies that since we see small changes going on today in successive generations of living things, we only have to extend this in time, and we will see the types of changes which have caused single-cell-to-man evolution.
Yeah, just like you only need normal, regular, everyday erosion and some time to dig a canyon, and just like every journey of 1,000 miles can be broken into single small steps, small changes from one generation to the next add up. In order for this not to work out, the creationists would need to propose some barrier to evolution. Some Rubicon that evolution cannot cross no matter what.
Creationists are thus seen as drawing some sort of imaginary Maginot line and saying, in effect, this much variation we allow, but no more.
Yeah, they're seen as doing that, because that's manifestly what most of them are doing. Now, I can't say that they all are. When you have no evidence, you tend to make up wild crap. And that happens in young Earth creationism, just like in its twin sister ideology, Flat Earth. So far, all that this article seems to be proposing is that this fact be concealed with vague terminology. But perhaps there will be more to it than that.
Call it microevolution or variation within kind. When a creationist says that, after all, mosquitoes are not seen turning into elephants or moths, this is regarded as a simplistic retreat.
It's seen as more than that. It's seen as not knowing the very most basic facts about evolution. It's like telling a pastor that Christianity isn't for you because you're pretty sure that God doesn't have a penis. It's objecting to something that generally isn't really a thing in Christianity in the first place.
Such a criticism is not without some justification, because the neo-Darwinist can rightly say that he would not expect to see that sort of change in his lifetime either.
No, not just in a single lifetime. Ever. In any number of lifetimes. Not in an infinite amount of time. Mosquitoes cannot become members of other non-mosquito groups. That's a fundamental misunderstanding of what evolution is and how it works. Evolution follows what is often called the law of monophily. Basically, you can't evolve out of your ancestry. This should be fairly obvious. If you have kids, your ancestors are also their ancestors, and their descendants will also have your ancestors as their ancestors. Nothing can possibly change that fact. It's not logically possible. So mosquitoes will always produce mosquitoes. They may eventually not be easily recognizable as such, but they still will be mosquitoes. They could give up drinking blood, become fully aquatic, and become 6 meters long, but they'd still be mosquitoes.
The post-neo-Darwinist...
Post-neo-Darwinist? My goodness, how many different ways will conspiracy theorists come up with to just say normal person?
May say that our sample of geologic time is too small to be sure of seeing a hopeful monster.
Nope, it's more that hopeful monsters aren't really a thing. Hopeful monsters were an idea of the saltationists, who thought that while variation within species proceeded from small changes to the genetics of organisms, that major changes from one species to the next, or even between groups like orders and classes, happened as a result of rare macromutations, which resulted in an entirely new species in a single generation, with vast morphological differences from the ancestral species. The classic example being a crocodile that laid an egg that hatches into a bird. Such things simply are not possible. They don't happen, and we don't expect them to happen. Indeed, finding evidence of such a thing would be a major challenge to the theory of evolution as it is now understood. Not only does there not seem to be a mechanism to allow for such mutations, even if there were, that would result in a single member of a new species with no prospective mates, so even if hopeful monsters were real, they couldn't really go on to found new groups.
Or any sort of significant saltational change.
Or really, any, because evolution does not proceed by saltation. The closest that it comes is whole genome duplication, but this does not generally radically change the morphology of the organism.
Another reason why the creationist position often appears as one of weakness is that we are perceived as admitting variation only because of being forced to do so by observation.
Which again is just manifestly true. Since until it became undeniable, creationists tended to believe in species fixity and species-level special creation. Linnaeus himself, famously the creator of our system of taxonomy and an advocate for divine special creation, thought that God had created each species as is and that they neither changed, arose, nor went extinct.
Then simply escaping the implications of variation by saying it does not go far enough.
I love how much of this is just people often point out the things that we do. Yeah, that's how being in a society works, bucko. People will notice the things that you do, and they may even comment on it. For example, this video has a comment section, where people very well may comment on what I do in this video. In fact, if you're watching right now, go ahead and leave a comment talking about both what I'm doing and what Carl is doing.
And we appear to redraw our Maginot line depending on how much variation is demonstrated.
Generally, yes. Although there is one major exception, which is to say humans, since that's the only species that is created by God and named as such a creation in the Bible. The other species are all glossed over by generalizing them to their habitat or mode of locomotion. So creations can be wishy-washy on most organisms, but not with humans. Humans have to be the special boys.
It will be shown shortly, though, that this is a caricature of the creationist position, and that the limits to variation arise from basic informational consideration at the genetic level.
I really should add another keyboard to my Amazon wishlist. I keep breaking my X key with all this doubting I'm doing.
The created kinds.
Something that is supposed to objectively exist, but that can't be tested for and so has no place in the scientific discourse until that changes. Let's see what Carl has for us.
Observed variation does appear to have its limits.
No, Carl wants it to have limits. That's not the same thing as actually appearing to have such limits.
It is tempting to use this fact to show that there are created kinds and that variation is only within the limits of such kinds. However, the argument is circular and thus vulnerable, since creationists, by definition, regard all variation as within the limits of the created kind. See, for example, the statement of belief of the Creation Research Society of the USA. How can we then use observations to prove that variation is within the limits of the kind?
You can't. You'd first need to establish what a kind is in a way that is not dependent on observed variation. The problem, of course, is that no one can define a kind in a falsifiable manner, and so the concept itself is worthless in science.
To put another way, of course we have never observed variation across the kind, since whatever two varieties descend from a common source, they are regarded as the same kind.
And if that's a definition that creationists would like to go with, then it means that there is only one kind, life kind, and evolution does not propose that anything ever went from one kind to another.
It is no wonder that evolutionists are keen to press us for an exact definition of the created kind, since only then does our claim of variation is only within the kind become non-tautologous and scientifically falsifiable.
Thanks for doing my job for me, Carl.
Circular reasoning does not invalidate the concept of created kinds, however.
What invalidates it is that there's no possible way to check for such things, and that all available evidence points to universal common descent.
In the same way, natural selection is also only capable of a circular definition.
No, it's not. Watch, in a population of replicators who are non-identical, those replicators which replicate most are the fittest, and their traits will go on to become present in higher proportion in the future generations of replicators. That this happens is natural selection if those traits influence the rate of replication. Wow! I defined it without circularity.
Those who survive are the fittest.
Wrong.
And the fittest are those who survive.
It's about reproduction, not survival. If an organism manages to have 10,000 babies and then keels over dead at the age of 2, while another organism lives to be 10,000 years old and only has 3 kids, the first one is fitter.
But it is nevertheless a logical, easily observable concept.
All that's happening here is that Carl Wieland is showing that he has no clue what he's talking about.
All we are saying is that arguments which are inherently circular cannot be invoked as independent proof of the kinds.
How magnanimous he is.
When I claim that such independent proof may not be possible by the very nature of things, this statement is in no way a cop-out.
And he's not wrong. Circular definitions are not going to help creationists out when it comes to figuring out what kinds are in a falsifiable manner.
For instance, let us say we happened upon the remnants of an island which had exploded, leaving behind the debris of rock, trees, sands, etc.
Look, for now, I'll take Carl's word for it. But it sure seems like a cop-out for the fact that if there is any truth at all to special creation, then kinds would be objectively real groups of organisms and should be detectable as such. As it is, no one can figure out how to do that except by just doing evolutionary biology badly, and then pretending that discontinuity in cherry-picked samples means that maybe you have found a kind. Or just by sort of eyeballing it. I'm not kidding there. Creationists call it the cognitum, and it's literally just them using a Latin word so they don't have to say what it is in English. What it is just looking at organisms, usually animals, since apparently creationists don't much care about plants, and then deciding that, yeah, goats and cows look different. They're probably not in the same kind. On the other hand, all frogs seem to be about the same to a person who doesn't know anything about frogs, and so yeah, I guess they're all in the same kind. It's not a coincidence that kinds get smaller and less diverse the more similar the organisms in question are to humans. It's because they're not being identified in some scientific manner, but rather by humans just kind of going by vibes.
It may be impossible, in principle, to reconstruct the original positions of the pieces in relation to each other before the explosion. This does not, however, mean that it is not possible to deduce with a great degree of confidence that the current state of the debris is consistent with that sort of an explosion which was recorded for us by eyewitness testimony, rather than arising by some other mechanism.
Sure. I didn't really think we'd be doing island explosions today, but I'm game. Let's go.
In like manner, we can show that the observations of the living world are highly consistent with the biblically described concept of original created kinds.
Okay.
And inconsistent with the idea of evolution.
Then why hasn't it been done? Look back to the island explosion. In that, Carl contemplates the ability to rule out other possibilities besides the explosion. What that means is that he's envisioning hypothesis testing, even if he doesn't realize it. The way that this would work is by proposing alternative explanations, and then finding that there is evidence that precludes those explanations, and presumably this is done with every plausible explanation, and probably a few implausible options as well. In the end, the only hypothesis that hasn't been disconfirmed is the explosion. That's science. That's how it works. How was this done with kinds? What other options were ruled out? What statistical tests were used? What were the falsification criteria? Of course, there are no real answers to these questions, as near as I can tell, because kinds aren't real, and so any actual tests will fail to uncover any such things.
This is best done by focusing on the underlying genetic informational basis of all biological change.
Once again, using the word information, or in this case, informational, without telling us what it means. I don't generally use Wikipedia as a source, but I think that their article on pseudoscience is quite revealing. It has a section called Indicators of Possible Pseudoscience, with a handy little checklist you can use to see if something is probably pseudoscience. It's broken up into subsections, each of which has a number of bullet points. One of the sections is Use of Misleading Language, and has these bullet points. Creating scientific-sounding terms to persuade non-experts to believe statements that may be false or meaningless. For example, a long-standing hoax refers to water by the rarely used formal name dihydrogen monoxide and describes it as the main constituent in most poisonous solutions to show how easily the general public can be misled. And using established terms in idiosyncratic ways, thereby demonstrating unfamiliarity with mainstream work in the discipline. That second point sounds a lot like what's going on every time a creationist uses the term information.
This is more realistic and more revealing than focusing on the degree or extent of morphological change.
He does make a decent point there. Genetics is more revealing than morphology when it comes to things like taxonomy and common descent.
The issue is qualitative, not quantitative.
Now that sounds like a cop-out for the fact that there is no way to measure information that does not show that evolution can and does create new genetic information.
It is not that the train has had sufficient time to go far enough, it is heading in the wrong direction. The limits to variation, observed or unobserved, will come about inevitably because gene pools run out of functionally efficient genetic information or teleonomic information.
Two more undefined terms, and that's fun. Too bad it's entirely meaningless until those definitions are provided. Let's go back and remember the signs of pseudoscience. Creating scientific-sounding terms to persuade non-experts to believe statements that may be false or meaningless. For example, a long-standing hoax refers to water by the rarely used formal name dihydrogen monoxide and describes it as the main constituent in most poisonous solutions to show how easily the general public can be misled. And using established terms in idiosyncratic ways, thereby demonstrating unfamiliarity with mainstream work in the discipline. I don't know what creationists could do to make it any more obvious that they're pseudoscientists. If you have any ideas, let me know in the comments.
A full understanding of this eliminates the image of the desperately backpedaling creationist, redrawing his line of last resistance depending on what new observations are made on the appearance of new varieties. It also diffuses the whole issue of micro and macro evolution. I believe it is better for creationists to avoid these confusing and misleading terms altogether.
I'm just waiting for him to make a point.
The word evolution generally conveys the meaning of the sort of change which will ultimately be able to convert a protozoan into a man or an reptile into a bird.
Protozoan isn't a real taxon, and birds are still reptiles, but yeah, sure, evolution includes the evolution of humans from microbes and of birds from reptiles. And for most of that evolutionary history, the birds and humans have the same ancestry.
I hope to show that in terms of that sort of meaning, we do not see any evolution at all.
I mean, except that we do. Here's a bit of it. Also, here's a phylogeny of life on Earth based on genetics. I don't know what to tell you, but all the evidence points to universal common ancestry of all extant life on Earth. So far, nothing in this article has even attempted to counter the metaphorical and literal mountains of evidence for evolution.
By saying we accept microevolution, but not macroevolution, we risk reinforcing the perception that the issue is about the amount of change, which it is not. It is about the type of change. This is not merely petty semantics, but of real physiological and tactical significance.
Tactical? There's no war. There are a bunch of people lying to churchgoers, and there are scientists who are pushing the knowledge of humanity forward.
Of course, one can say that microevolution occurs when this word is defined in a certain fashion, but the impact of the word, the meaning it conveys, is such as to make it unwise to persevere with this unnecessary concessional element. Microevolution, that is a change no matter how small, which is unequivocally the right sort of change to ultimately cause real, informationally uphill change, has never been observed.
Yeah, let's ignore the microevolution that resulted in the CYP+ trait in E. coli, or adult lactose tolerance in humans, or dogs evolving whole new body parts by microevolution. Those are fake, I guess. Although, given how much pizza I see people eating and how good the dogs I live with are at looking at me with those puppy eyes, it seems like they're real. But what do I know? I'm just a theropod. I bite the heads off of sauropods.
In any case, leading biologists are themselves now coming to the conclusion that macroevolution is not just microevolution, using their terminology, extended over time.
Really? I would love a citation for that, but let's see what some actual evolutionary biologists have to say.
Hello, my gentle and of course very modern apes. My name is Erica. I'm Gutsa Gibbon here on YouTube, and I'm a 5th-year PhD candidate in biological anthropology. I taught Introduction to Biological Anthropology in lab form or in honors form for 4 years, and I'm here to let you know that macroevolution and microevolution are the same thing. They're just happening on different scales. Microevolution usually happens within species, and macroevolution usually happens between species or is considered to be speciation. Of course, it goes without saying that we see both of those things, change within species and change between species, or speciation, in the wild and in the lab today, but that's not really the point. The point is that the same mechanisms that drive microevolution, the 4 big ones that we teach are natural selection, mutation, gene flow, and genetic drift, those same mechanisms also drive macroevolution. The only difference between the two is the scale, within species or speciation, between species. In class, I always illustrate this with a color gradient. I show yellow to red as macroevolution, and I show dark red to light red as microevolution. Anyways, I hope that was helpful. Good job, Dapper.
Hi folks, my name's Zach Hancock. I'm an evolutionary biologist and currently an assistant professor at Augusta University. I specialize in population genetics and evolutionary theory. Now, a persistent question that emerges in discussions of evolution, especially with creationists, is whether or not macroevolution has distinct mechanisms or processes from microevolution. To answer this, we need to understand the distinction between evolutionary patterns and evolutionary processes. The distinction between microevolution and macroevolution is really just one of hierarchy. Microevolution is the study of change at the species level or below, whereas macroevolution is the study of change above the species level, such as the origin of new genera or families. All evolutionary change, that is, change in the heritable characteristics of populations over time, occur, obviously, at the level of the population. A new mutation emerges and must spread through the population and eventually replace the original form. All change in morphology that is heritable occurs in this way. Thus, within a population, new variation can be supplied either by mutation or by gene flow, which is when migrants from other populations, or sometimes even species, interbreed with the population of interest. That new variation then spreads to all members of the population across successive generations by either natural selection, if it provides an advantage to fitness, or genetic drift if its impacts are neutral. These are all processes. The aforementioned mechanisms propel populations to change over time and are acting on the heritable components of individuals within those populations. While microevolution is concerned with studying processes, macroevolution is about patterns. For example, a macroevolutionary study might ask, are families of species with large ranges more species rich than those with small ranges? The idea being that species with large ranges have a greater chance of getting isolated if the environment changes. If true, then over time, you'd expect families of organisms with large ranges to have more species diversity. This is a macroevolutionary pattern. But what is the process? Notice the processes here are all microevolutionary. Limited gene flow, mutations that appear in each isolated population, and their eventual fixation thanks to drift or selection. When this population-level process is repeated across many different species, it leads to a macroevolutionary pattern. Families with lots of species tend to have large ranges. This is also true for new features of organisms instead of just patterns of their diversity, like the evolution of feathers in birds or bipedalism in hominids. All of these features are genetic, implying their emergence required changes in the genes of the ancestral population. Only in retrospect do we designate these novelties as being representative of higher taxonomic categories. When they emerged, they were just features of the genetic variation in populations. Thus, there is no evolutionary process at the macroevolutionary level that is distinct from the microevolutionary one. Now, some folks might say, wait, what about punctuated equilibrium? Isn't that a process of evolution at the macroevolutionary level? Nope. Today, PE is mostly just a description of the rate of evolution across time. Specifically, it claims that all morphological change occurs when species split instead of gradually over the course of time, and so is quite rapid. This is a description of a pattern, not a process. The processes invoked by PE to explain this pattern are all microevolutionary and can be boiled down to either rapid selection or rapid drift, depending on the flavor of PE you subscribe to. In summary, the patterns of macroevolution are emergent properties of the processes of microevolution. And even supposed mechanisms of macroevolution, like PE, are really just descriptions of those patterns. Ultimately, this is because evolution requires genetic change to occur, and inheritance happens between individuals at the population level.
Hello everyone, my name is Dan, I'm an evolutionary biologist, and I just want to take a minute to clarify the terms microevolution and macroevolution. You often see people drawing a distinction between those two terms in terms of the processes that are involved, but they really just describe the scale of evolutionary change, not the underlying mechanisms. Microevolution refers to changes within a species or lineage. Basically, you're talking about the population genetics of whatever group it is you're looking at. Macroevolution refers to the longer-term processes of speciation and the origination of new and distinct lineages. The processes that are responsible for micro and macroevolution are the same. Variation generated through mutation and recombination, acted upon by natural selection and genetic drift. In the short term, those processes cause microevolution, and over longer timescales, they drive macroevolution.
Well, y'all know my sources.
In November 1980, a conference of some of the world's leading evolutionary biologists, billed as historic, was held at the Chicago Field Museum of Natural History on the topic of macroevolution.
Imagine citing something that isn't even an actual peer-reviewed publication from the 1980s and using the present tense as if that represents the current state of the field.
Reporting on the conference in the journal Science, Roger Levin wrote, The central question of the Chicago Conference was whether the mechanism underlying microevolution can be extrapolated to explain the phenomena of macroevolution. At the risk of doing violence to the position of some of the people at the meeting, the answer can be given as clear no.
Let's read the next two sentences after that. What is not so clear, however, is whether microevolution is totally decoupled from macroevolution. The two can more probably be seen as a continuum with a notable overlap. The issues with which participants wrestled fell into three major areas: the tempo of evolution, the mode of evolutionary change, and the constraints on the physical form of new organisms. Yet this was a conference about punctuated equilibrium that took place nearly half a century ago. Since then, the idea that macroevolution requires new processes that microevolution doesn't have available to it have been discarded. It was an interesting thought and taken seriously, and now we know that it's wrong. That's why I asked current scientists working in the field right now, rather than cherry-picking a science article that's not even a paper from 46 years ago.
Francisco Ayala, Associate Professor of Genetics, University of California, was quoted as saying, "But I am now convinced, from what the paleontologists say, that small changes do not accumulate."
Once again, let's read the next sentence for some context. The emerging picture of evolutionary change, therefore, is one of periods during which individual species remain virtually unchanged, punctuated by abrupt events at which a descendant species arises from the original stock. Wow, once again, this is just about the pace and mechanisms of evolution, and in no way is saying that macroevolution doesn't happen, or disputing universal common ancestry. Even if the conclusions in favor of punctuated equilibrium were true, it would be no help at all to special creation.
The fact that this article reaches essentially the same conclusion in the following pages can thus hardly cause it to be regarded as radical.
Yeah, I don't regard it as radical. I regard it as almost 50 years out of date, and of no help to creationism, even if it were not.
Nevertheless, the vast majority of even well-educated people still persist in this ignorance of this. That is, they believe big change equals small change times millions of years.
Because it does, and nothing about a conference from decades ago discussing a now largely discredited idea in evolutionary biology does anything to change that.
The concept of information.
Oh look, it's the section that should have been right at the start.
The letter of this printed page, that is, the matter making up the ink and paper, all obey the laws of physics and chemistry.
I'm sure this was published in physical form before being posted on Creation.com, so I'm not going to complain about the fact that there's no ink or paper involved in the version I'm reading.
But these laws are not responsible for the information they carry. Information may depend on matter for its storage, transmission, and retrieval, but is not a property of it.
I don't even know what that means, probably because even in the section about the very concept of information, the word remains undefined.
The ideas expressed in this article, for instance, originated in mind and were imposed on the matter.
Yeah, no one has, ever, to my knowledge, disputed that information can be encoded into matter by intelligent agents. And that's true for any definition of information that I can think of.
Living things also carry tremendous volumes of information on their biological molecules.
Not in the same way, though. Living things have no symbolic or linguistic information in them, unless that living thing is a human or something similar with the information in question in its brain. And for now, I'm sticking with Shannon information as my definition for information. It's one of the most widely used definitions, and it's the definition that underpins information theory. Shannon information is measured the same way data is on your hard drive, bits and bytes, and their derivatives like kilobytes and megabytes. The object under consideration in information theory is a signal. It can be of any kind. It can be made up of characters on a page, pulses of light, or electricity, sound waves, whatever. As long as it can be rendered as a sequence with identifiable differences in a single dimension, we can analyze it with information theory. We can even do this for things that are more than 1D. For instance, an image file can be described as a signal that defines its height and width and then just uses a pattern to give an integer to each pixel and lists out the color values for each pixel in order. The amount of Shannon information in such a signal is equal to the number of bits it would take to store that signal such that it can be reproduced. Some signals can be condensed into very small amounts of information. For example, the signal of pulses of radiation from a pulsar consists of evenly spaced pulses of a known duration that happen with a known periodicity and at a known intensity. So that signal doesn't need to store more information than the duration, period, and intensity of the pulses. On the other hand, a book can't be condensed that far. But since the order of letters isn't entirely random, some compressions of the signal is possible, and so a book has less information than it would take to, say, store it as plain text in ASCII. In terms of Shannon information, DNA is more like a text than a pulsar signal, although it's not linguistic in any way.
Again, this information is not a property of their chemistry, not a part of matter and the physical laws per se.
But it is. No one has to interpret it. It doesn't need a mind to parse it. Genetics and protein synthesis is purely a result of chemistry and physics, and chemistry is just part of physics ultimately. So yeah, it is a property of the matter, just like the information in a pulsar signal is a property of the matter and energy.
It results from the order, from the way in which the letters of the cell's genetic alphabet are arranged.
Which is the chemistry. That's what chemistry is, how atoms are arranged in molecules. Is it just me, or is he contradicting himself here?
Doesn't anyone notice this? I feel like I'm taking crazy pills!
This order has to be imposed on these molecules from outside their own properties.
No, it doesn't. They form and change according to the laws of chemistry. Is there magic going on? What does he actually mean by this? Does he think that DNA has a soul or something? That is the real information? As if the information is anything other than the nucleotide sequence, which is itself the signal that contains all the Shannon information that's in DNA.
Living things pass this information from generation to generation. The base sequences of the DNA molecules effectively spell out a genetic recipe book, which determines the ultimate properties of the organism.
That's true to some degree, but the actual form of an organism is determined by both the environment and genetics. That's why identical twins don't remain completely identical as time goes on.
In the final analysis, inherited biological variations are expressions of the variations in this information. Genes can be regarded as sentences of hereditary information written in the DNA language.
They can be, but they shouldn't be. DNA doesn't have words, sentences, paragraphs, chapters, or anything else like that. DNA is the language. It doesn't have grammar. It doesn't have lexemes, morphemes, or phonemes. It doesn't have symbols. It's a class of molecule. Comparisons to texts are only useful in terms of high-level overviews, but the instant you dig into anything deeper than just the most basic DNA is a bunch of nucleotides arranged into chromosomes, the linguistic comparisons are worse than useless. They mislead more than they elucidate, and it's why I really don't like using them. Generally, I think analogies are not very useful when discussing genetics at all.
Imagine now the first population of living things on the evolutionists' primitive Earth.
It's actually hard to imagine, since the transition from life to non-life isn't like the flipping of a switch. There would be life-like populations that might not meet whatever criteria we set for life, and it's also not clear just how simple or complex the earliest thing we could reasonably call life would be. I'm not even positive that it would be cellular in the way that we understand it today. That being said, I'll do my best to imagine it.
This so-called simple cell
I did not agree that it would be a cell, so we're already off to a bad start.
Would, of course, have a lot of genetic information.
Maybe, I don't know. I don't know how much of a genome is required to be alive, or whether the first living population would have had that amount or some higher amount. These are very much questions to which we do not have firm answers. This is one of the reasons I don't talk much about the origin of life. Virtually all we know about it is uncertain, because it is still a young field, and it is also very hard to research one.
But vastly less than the information in only one of its present-day descendant gene pools, i.e. man.
That at least seems plausible.
The evolutionist proposes that this telegram has given rise to encyclopedias of meaningful, useful genetic sentences. See later for discussions of meaning and usefulness in a biological sense.
He said evolutionists.
Let us pray. Our Darwin, who art in the British Museum, hallowed be thy name. Thy evolution come, thy selection be done, on land as it is in water. Give us this day our daily sexual selection, as we sexually select, and lead us not into inbreeding, but deliver us from extinction. Amen.
Thus, he must account for the origin with time of these new and meaningful sentences. His only ultimate source for these is mutation.
That's true.
Going back to the analogy of the printed page,
I feel like I'm getting seasick every time a creationist wants to dig into a linguistic analogy for DNA. At this point, I just know that they're going to rely on intuitions about the fraction of sequences of letters that can mean something in a natural language being exceptionally low to make it seem like random nucleotide sequences or random changes to existing ones will be nonsensical too. But that's just not true. But hey, let's see how this goes.
The information in a living creature's genes is copied during reproduction, analogous to the way in which an automatic typewriter reproduces information over and over.
I don't think it's particularly analogous, no. Other than the fact that a new copy is made, I don't think that the mechanisms are at all similar, nor is the type of information similar.
A mutation is an accident, a mistake, a typing error, although most such changes are acknowledged to be harmful and meaningless.
Harmful and meaningless are not the same thing. In fact, they're mutually incompatible. A mutation can be harmful or meaningless, but not both. It turns out nearly all of them are meaningless and neither harmful nor helpful. Of the rest, more are harmful than not, and a few are known to be beneficial.
Evolutionists propose that, occasionally, one is useful in a particular environmental context.
No, it's not just proposed, it's demonstrated. We've seen it happen. It's like saying zoologists propose that giraffes have very long neck bones, or like saying physicists propose that opposite electrical charges attract each other. These aren't hypotheticals you're free to ignore when you engage with the science. These are simply facts of reality that are not in dispute, and are part of the routine operation of the world we live in. To deny this isn't just to deny science, it's to deny reality at the most fundamental level. It's a hair's breadth away from solipsism.
And hence its possessor has a better chance of survival and reproduction.
For what feels like the quadrillionth time, reproduction is the key here.
By looking now at the informational basis for other mechanisms of biological variation, it will be seen why these are not the source of new sentences, and therefore why the evolutionist generally relies on mutation of one sort or another in his scheme of things. Mendelian variation. This is the mechanism responsible for most of the new varieties which we see from breeding experiments and from reasonable inferences in nature. Sexual reproduction allows packets of information to be combined in many different ways, but will not produce any new packets or sentences.
That's not necessarily true. While it is true that when reproduction occurs to create a diploid organism from haploid gametes, the chromosomes are inherited wholesale from each gamete, during gametogenesis, things are not so simple. When gametes are forming, the chromosomes can swap parts, allowing different alleles to be shuffled, breaking up the linkage groups that are the chromosomes. Normally, these crossovers occur in intergenic regions, such that no genes are changed. But this is not always the case, and it can result in new alleles or even new genes at crossover sites. Whether this counts as new information or not is a matter of semantics, but no matter what one concludes about that, it remains the case that new variations can and do occur this way. It is distinct from what we normally term mutations, such as insertions, deletions, duplications, and point mutations, though.
For example, when the many varieties of dog were bred from a mongrel stock, this was achieved by selecting desired traits in successive generations, such that the genes or sentences for these traits became isolated into certain lines.
That's true, but it's also important to note that many of the alleles, which is what he really means in most cases, were not ones that existed in the original population from which dogs were bred. For instance, the alleles that give modern pugs their absurd brachycephalic heads can't be found in wolves or modern populations of feral dogs. It was a mutation that occurred in this line. But this is not the only example of new alleles or genes in dogs that arose from mutation. Dogs also have many more amylase genes than can be found in wolves. These have to have come about through gene duplication. They also develop more muscles than wolves, which similarly had to come about through mutation. Even something as simple as all black fur is a mutation that occurred in dogs, and those wolves with this trait have it because of dog ancestry. The breeding of domestic animals does not simply result in a group of animals with a subset of the genetic traits of their wild ancestors. It results in new alleles and new genes, which are often then selected for, although not always. I doubt anyone ever selectively bred dogs for their increased amylase production.
Although some of these sentences may have been hidden from view in the original stock, they were already present in that population.
Nope, that's just a lie.
We are disregarding mutation for the moment, since such varieties may arise independently of any new mutations in the gene pool. Some dogs undoubtedly have mutant characteristics.
First, I don't know what "we are disregarding mutation for the moment since such new varieties may arise independently of any new mutations in the gene pool" means. I'm trying to parse it, and the best I have is "we are disregarding mutations because mutations happen independently in different gene pools." And while it's true that mutations arise independently, I don't see why that's a good reason to disregard one of the most important factors in the breeding of domesticated animals, and one that explains many of the differences between breeds. As for the statements that some dogs undoubtedly have mutant characteristics, all dogs have mutant characteristics, and all their characteristics are the result of mutation. Mutation and recombination are how genetic variation arises, along with horizontal gene transfer, which is not at play much in dogs. So we'll ignore it for now. Since creationists have yet to demonstrate another mechanism for genetic diversity, the only conclusion we can reach is that essentially all such diversity is the result of mutation. Much of it deep in the past for sure, but still, it's from mutation. It's hard not to interpret this parenthetical pair of sentences as Carl Wieland telling us that he is about to lie to us about genetics.
This sort of variation can only occur if there is a storehouse of such sentences available to choose from.
But that's only the case because Carl decided to ignore mutation. Once we don't do that, since there's literally no reason to, we realize that new genetic variations are in fact generated at every reproductive event, and so breeding need not select only from variation that was in the original stock of organisms from which the new breed is being derived. So yeah, it does indeed seem like Carl was just setting us up to be lied to. I don't particularly appreciate that.
Natural or artificial, selection can explain the survival of the fittest, but not the arrival of the fittest, which is the real questions.
And for some reason, we're just ignoring the answer.
These Mendelian variations tell us nothing about how the genetic information in the present stock arose.
That's actually not true, at least not fully. You can assess the genetics of a current breed as well as those of other breeds to build up a phylogeny of their genes and alleles, and you can thus tell in which lineages mutations arose, the approximate time when the breeds split from each other into separate gene pools, and even reconstruct the likely state of the variability in the original gene pool from which they all arose. This is just done through statistics and hypothesis testing. Granted, you can't do that from the genome of one individual, but you don't have to. It's just lie after lie with Carl right now, and in some cases, it seems he knows he's lying to us. Or maybe he's just a moron. Creationists, if you want to argue that Carl is actually just incredibly dumb, please let me know why you think that.
Hence, it is not the sort of change required to demonstrate upward evolution.
Oh no, the weird, dishonest strawman version of selective breeding that Carl concocted in his head specifically so it is missing one of the major mechanisms of evolution doesn't actually do what evolution does. That's neither surprising, significant, nor impressive.
There has been no addition of new useful sentences. And it is in spite of the fact that it is possible to observe many new varieties in this way, even new species. If you define a species as a freely interbreeding natural unit, it is easy to see how new species could arise without any uphill change. That is, without the addition of any new information coding for any new functional complexity.
Is there a point coming?
For example, mutations could introduce a defect, which served as a genetic barrier, or simple physical differences, such as the sizes of Great Danes and Chihuahuas, could make interbreeding impossible in nature.
Okay, cool. So what?
It is a little surprising to see the occasional creationist literature clinging to the concept that no new species have ever been observed.
Well, creationists are denialists right up there with flat earthers, and to a lesser extent, Jesus mythicists like Richard Carrier. So nothing they say surprises me as long as that thing that they're saying would make their ideas more plausible if it were true.
Even if this were true, and there's some suggestion that it actually has been observed, there are instances of clines in field observations which make it virtually certain that two now isolated, reproductively, species have arisen from the same ancestral gene pools. Yet the very same creationists who seem reluctant to make that sort of admission would be quite happy to agree with the rest of us that the various species within what may be regarded as the dog kind, including perhaps wolves, foxes, jackals, coyotes, and the domestic dog, have arisen from a single ancestral kind. So why may this no longer be permitted to be happening under present-day observations?
I don't know, seems like an internal problem for creationists.
It is not only biblically and scientifically unnecessary, but it sets up a straw man in the sense that any definitive observation of a new species arising is used as a further lever with which to criticize creationists.
We wouldn't want that now, would we?
What we see in the process of artificial selection or breeding giving rise to new varieties is a thinning out of the information in the parent stock, a reduction in the genetic potential for further variation.
In practice, yes, but not because it could not be otherwise, but because selective breeding tends to happen at a pace that is far higher than the rate at which mutations can accumulate in a lineage. Artificial selection is one of the harshest selection regimes a population can be under. And in nature, selection is rarely so strong. But further, this is something we could test. We have fossils of populations that, if this hypothesis is true, would represent ancient, more genetically diverse populations that arose significantly earlier than present, but still after the supposed flood of Noah. In many cases, they're so young that we can get genetic samples from them, like in the case of the mammoths or Neanderthals. If this hypothesis is true, then we should see a consistent pattern of increasing genetic diversity in lineages as we trace them back through time. But we don't. That's because genetic diversity can increase as well as decrease.
If you try and breed a Chihuahua from a Great Dane population, or vice versa, you will find that your population lacks the necessary sentences.
Yeah, because the mutations that occurred to allow for either breed to exist did not occur in both lineages. Since, as Carl knows, mutations arise in different lineages independently. But could you breed Great Danes into animals as small as Chihuahuas? There's no reason why not. Mutations that result in smaller Great Danes are sure to arise, and if you select for them, you'll get a smaller Great Dane. There's no reason to think that given sufficient time you couldn't keep this up until you got tiny toy Danes. A similar process is how we got toy or miniature poodles. They were bred to be almost as small as a Chihuahua from standard-sized poodles.
This is because, as each variety was selected out, the genes it carried were not representative of the entire gene pool.
That's probably not true. I can't find anything indicating that Chihuahuas and Great Danes have different genes, although it is possible. The big difference is their alleles for those genes, which are quite different in those genes that determine things like growth rates. I have to say, having a creationist who doesn't know the difference between allele and a gene, trying to tell me how science can't account for how genetics works, is pretty rich. It's on the level of someone who can't define a pronoun, telling me that they don't use pronouns because pronouns are woke.
What appeared to be a dramatic example of change within the appearance of apparently new traits thus turns out, when its genetic basis is understood, to be an overall downward movement in informational terms.
That hasn't been established at all. Now, in the real world, it is the case that artificial selection tends to result in lower genetic diversity, but not because such things are required in order to make a dog really big or really small. It's because in doing so as fast as possible, humans also reduce the diversity in other parts of the genome. Let's say you're breeding some dogs for large size, and you're only allowing the largest 10% of your animals to reproduce. Those animals will also likely have only a subset of the genetic diversity of the whole population from the generation before them. For example, let's say you have 100 dogs in your breeding stock. Only 10 of them are going to reproduce. Chances are that in the other 90, there will be alleles that have nothing to do with size, but that are not present in the 10 dogs who will reproduce. And so when you eliminate them from the gene pool by not breeding them, the resulting next generation will not have those variations, even though they were not directly selected against. On the other hand, if you decide to do much weaker selection, which humans tend not to do, then this would not occur. For example, if you selected the largest 90% of dogs to breed, the chances are that very few, if any, alleles will be lost that way in any given generation, regardless of their connection to size. This will also mean that it will take much longer to get the population to reach the size you want them to. And in a case where 90% of the population are reproducing at every generation, it's quite likely that mutations can outpace the elimination of alleles or even new genes from artificial selection. And the genetic diversity of the population may well not decline or even increase.
The number of sentences carried by each subgroup is thus reduced, making it less likely to survive future environmental changes.
Did we just swap from artificial to natural selection? Dogs generally don't adapt to new climates. They adapt to new human desires, because humans are the ones who decide which dogs reproduce, except in feral populations. But also remember, the only reason that genetic diversity must decline in Carl's model is because he already arbitrarily excluded mutations from the model. Why did he do that? Well, it seems to be so that he could get a model of reproduction that would make long-term evolution impossible. But that's just a straw man of evolution. It doesn't do anything to tell us whether or how evolution could occur in reality, where mutations are real.
Extrapolating that sort of process forward in time does not lead to upwards evolution, but ultimately to extinction, with the appearance of ever more informationally depleted populations.
Yes, when you model evolution without important mechanisms of evolution, you get results that indicate that evolution doesn't work. It's kind of like making a model of how an airplane works, intentionally leaving out anything that generates thrust, like jets or propellers, and then noting that since the plane has no way to add energy to its glide, no planes could ever take off or fly any significant distance. Therefore, aviation is impossible. It's an obviously wrong thing to do, and it's only explicable, as near as I can tell, if someone is just incredibly stupid or is lying. Let me know in the comments which one you think it is.
2. Paloidity.
This is a minor correction for Feuerwache, who is otherwise doing an excellent job with reading this. It's polyploidy, not paloidity. But hey, his English is way better than my German, so overall, I want to say thank you for all the voice work. It's been great, and this video couldn't get done without it.
Again, no sentences appear which did not previously exist. This is the multiplication, photocopying, of information already present.
So, since Carl is once again not bothering to tell you what the words he's using mean, polyploidy is when an organism carries more than 2 copies of each chromosome. It's common in plants and rare but not unheard of in animals. The reason that this is an important process in evolution is that it gives a larger number of genes in an organism that can now go on to mutate without changing the presence of whatever the original gene made. For instance, if you have a gene for some structural protein, and there is a whole genome duplication resulting in polyploidy, one of the copies of that structural protein can mutate, allowing for it to code for a new product, while the original product is still produced. This is a way that lineages can significantly increase the number of genes that they have without losing vital genes that were already present. On its own, it does not produce new genes, but on its own, bricks and mortar don't produce new buildings either. Yet they are an important and vital part of many new buildings being erected.
3. Hybridization. Again, no new sentences. This is the mingling of 2 sets of information already present.
That's true.
Although it is new information if all you look at is one of the populations, so it still increases the diversity of the hybridizing populations overall. This means that it is, in fact, significant in terms of evolution.
4. Mutation.
Oh hey, the thing that Carl decided to ignore in hopes that we wouldn't notice that his model has nothing to do with reality.
Since mutations are basically accidents, it is not surprising that they are observed to be largely harmful, lethal, or meaningless to the function or survival of an organism. Random changes in a highly ordered code introduce noise and chaos, not meaning, function, and complexity, which tend to be lost.
Actually, in this case, it tends to be maintained, and that's without taking selection into account. The vast majority of mutations cause literally no change at all, because when you add up mutations that occur in non-functional regions and those that result in synonymous codons, you get the majority of mutations. Such mutations have little to no effect on anything. Of the remaining mutations, it's true that most are going to be harmful in most or all environments.
However, it is conceivable that in a complex world, occasionally a destructive change will have a limited usefulness.
Then how is it destructive? We see new genes arise, new proteins form, New anatomical features arise, new metabolic pathways evolve. How is any of this destructive? I guess you could say that everything is destructive. My jaw had to destroy ancestral gill arches to evolve, and my limbs had to destroy ancestral fins in order to exist, and so on. But no one with any sense would frame it that way, unless of course they wanted to pretend that the simple fact that evolution causes changes was on its own bad, but wanted to do so without coming out and saying it directly, since that would be an obviously stupid take.
For example, if we knock out a sentence such that there is a decrease in leg length in sheep, and there is such a mutation.
There is indeed. Or rather there was. The breed is now extinct, but it did exist and they were the Ancon sheep. Now the mutation has cropped up a few more times, but it was not bred for and so there are no current flocks of such sheep. As for what caused the trait, it was obviously a mutation, but as for knocking out a sentence, I don't know what that means. As far as I can tell, no one has actually determined exactly what mutation causes this trait. Carl seems to be implying that the mutation was the deletion of a gene, but I can't find any reason to think that that's true. I do know that whatever causes it is recessive because when an Ancon sheep is crossed with another sheep, all the offspring have normal legs. But when those offspring mate with other offspring of Ancon sheep, some of the resultant sheep have short legs. So all we can say about this mutation is that it's recessive and causes short limbs with enlarged joints that tend to become crooked with age.
This is useful to stop them from jumping over the farmer's fence.
And that's it. No further discussion about Ancon sheep. I don't know why not. I don't know what point that was supposed to illustrate. Except maybe that the fitness-affected mutations are dependent on the environment, which is true. In fact, that's a core aspect of evolutionary biology.
A beetle on a lonely, windswept island may have a mutation which causes it to lose or corrupt the information coding for wing manufacture.
What does that even mean? Beetles don't have wing blueprints. Does this mean that their wing covers, which are themselves broken wings, fuse? Does it mean that the wings under the covers are small? Does it mean that the wings never grow at all, meaning no wings or wing covers? These are all different things.
Hence, its wingless successor will not be so easily blown out to sea, and will thus have a selective advantage.
Oh, okay, that's it. Just again pointing out that a mutation might be helpful in an environment, while it obviously might not be in another one. Cool. I bet it happens again with the next point.
Eyeless fish in caves.
Wait, is this secretly being written by Randy Galuza? I mean, I've never seen Carl Weiland and Randy Galuza in the same room at the same time. So maybe.
Some cases of antibiotic resistance. The handful of cases of mutations which are quite beneficial do not involve the sort of increase in functional complexity which evolutionary theory demands.
I don't know what functional complexity is or how to measure whether it has gone up or down. This is a continuous problem with not just this article, but really Creationist talking points as a whole. They'll demand that some quantity of something that they haven't actually defined go up, and then since no one knows what it would even look like for some unexplained quantity to go up, or how to measure its amount in the first place, no one can do it. The Creationists will then count their own inability to actually define terms as a win. What really happened, though, is that it shows that Creationists can't even make an argument against evolution that makes sense on its own terms. At most, I can kind of parse this as saying that we don't see new genes evolving, but we do. New antifreeze proteins evolved in some fish from digestive enzymes. Bacteria have evolved brand new enzymes to digest various kinds of plastic. Primates evolved new genes for a third kind of cone cell in the eye, allowing them to be more or less the only mammals with trichromatic vision. The fact that he cherry-picked some beneficial mutations that did not involve new proteins doesn't mean that examples that do involve such don't exist.
Nor would one expect this to be possible from a random change.
Or maybe they would expect it. I guess it depends on what in the name of the dark gods functional complexity is.
At this point, some will argue that the terms useful, meaningful, functional, etc. are misused.
I would love to say that, but I can't, because I don't know how they're being used at all.
They claim that if some change gives survival value, then, by definition, it has biological meaning and usefulness.
I would not say that, no. What I would say is that it's beneficial, because that has a standard definition in evolutionary biology and I like to use the terms that are actually used to describe things in the fields I'm discussing. Something that's beneficial to me is when people like, share, and subscribe to the channel. It's one of the only ways that YouTube knows to send my videos to more people, even the people who already told YouTube that they want to see my videos. So why not hit that like button, subscribe if you haven't already, turn on all notifications, and leave me a comment.
But this assumes that living systems do nothing but survive, when in fact they and their subsystems carry out projects and have specific functions.
Well, let's assume that Carl meant beneficial, in which case, no, it doesn't assume that all cells or organisms do is survive. First, evolution isn't about survival, it's about reproduction. Survival only matters in evolution to the degree to which it permits more reproduction. Also, survival itself as well as reproduction are not simple tasks, and for most organisms, both involve all sorts of other processes. Mutations are beneficial if they allow an organism to contribute to a larger degree to the genetic makeup of future generations, and deleterious if it causes the organism to contribute to a lesser degree to the same. Why is this what is relevant in evolution? Well, evolution is all about change in populations of organisms over time, and only reproduction affects this. If you can't make copies of yourself during your lifetime, then after that time you won't be relevant to what genetics the organisms that exist will have.
That is, they carry teleonomic information.
Teleonomy is the apparent design and purposefulness in organisms, and if we accept that information has any actual definition in this paper, then I would say that yes, genes do usually, although not always, carry information which appears purposeful, and so it's fair to say that some of them carry teleonomic information.
This is one of the essential differences between living objects and non-living ones, apart from machines.
That they have heritable teleonomic information? Actually, I can kind of get on board with that, but that's a problem though. Creationists want them to have teleological information. That is actually design and purposeful information. Teleonomic information is just information that appears that way because its existence has helped perpetuate itself, regardless of anyone's or anything's wishes or design.
These projects do not always give rise to survival or reproductive advantages. In fact, they may have very little to do with survival, but are carried out very efficiently.
Sometimes things that help with survival or reproduction are done when they aren't doing that thing. But that doesn't negate that they're there because they help with survival or reproduction. An obvious example might be a human couple who have voluntarily opted not to have children, but who still engage in sexual intercourse. Clearly, their sexual intercourse isn't helping with reproduction, but because it being fun and having bonding effects on humans who engage in it together evolved to encourage reproduction, they do it for the fun and bonding without the reproduction. This isn't really that hard to understand, unless of course you're trying to not understand it. The same is true of the people who drink diet soda. Diet soda has effectively no nutrients in it, and it's not even a great source of hydration. However, it has an artificial sweetener that makes it taste like a high-calorie substance, and animals in general love calorie-dense food, so to humans it tastes great, so they drink it.
The Darwinian assumption is always made, of course, that sometime in the organism's evolutionary history, the project had survival or reproductive value.
Put in the comments if you think he'll have a counterexample where we can say with any confidence that it did not.
For example, the archerfish with its highly skilled hobby of shooting down bugs, which it does not require for survival at the present time.
That's literally how archerfish eat. That's pretty damn tied to survival and reproduction. What does Carl think archerfish eat? Because it's insects and other small animals that they shoot down with jets of water from highly derived mouthparts. They're not hunting for trophies, they're hunting to put food on the metaphorical table.
However, since these are non-testable assumptions, it is legitimate to talk about genetic information in a teleonomic sense, in isolation from any possible survival value.
That's it? His only example is a fish who uses its so-called hobby as its actual primary way of getting food? If this article were a high school paper, I'd give it a D at best.
Gene pools of today carry vast quantities of information coding for the performance of projects and functions which do not exist in the theoretical primeval cell.
Well, yeah, creatures today have to contend with a far more complex environment and much more competition than any first cell would have had to. Also, genetic drift can and does needlessly complexify biochemical processes. And it's been a few billion years, so drift has had a lot of time to do that.
Hence, in order to support protozoan to man evolution, one must be able to point to instances where mutation has added a new sentence or gene coding for a new project or function.
Okay.
Who's that Pokemon? It's
Notothenioid icefish. These are fish that live in the Antarctic and had a partial gene duplication in their trypsinogen gene that neo-functionalized into an antifreeze protein, allowing them to survive in colder water than their close relatives who lack such a protein, allowing them to colonize the Southern Ocean. In case you're wondering, trypsinogen is a protein that is made primarily in the pancreas, and it is converted into trypsin when the protein digestion is needed. Trypsin breaks down proteins and is stored as trypsinogen because it would otherwise start to break down the pancreas itself, since that's largely made of protein. Well, that was easy.
This is so regardless of one's assumption on the survival value of any project or function.
I get a level with you. I have no clue what that sentence is doing in this paragraph. Was Carl okay when he wrote this? Like, was he suffering from a neurological problem and me doing this whole series is now just me mocking a man with a medical problem that caused him to do this? I'm starting to feel uncomfortable. But on the other hand, he said things that make me think he knows better and is just being dishonest, as is typical for the professional creationist. So maybe I shouldn't feel bad about this at all and should continue eviscerating this nonsense.
We do not know a single mutation giving such an increase in functional complexity.
Don't we, though? I mean, we did just have this. Okay.
Who's that Pokemon? It's
Notothenioid icefish. These are fish that live in the Antarctic and had a partial gene duplication in their trypsinogen gene that neo-functionalized into an antifreeze protein, allowing them to survive in colder water than their close relatives who lack such a protein, allowing them to colonize the Southern Ocean. In case you're wondering, trypsinogen is a protein that is made primarily in the pancreas, and it is converted into trypsin when the protein digestion is needed. Trypsin breaks down proteins and is stored as trypsinogen because it would otherwise start to break down the pancreas itself, since that's largely made of protein. Well, that was easy.
Probabilistic considerations would seem to preclude this in any case, or at least make it an exceedingly rare event.
It is exceedingly rare, and if you have enough goes of it, then they'll happen quite a lot. And if you have a mechanism for preserving the effects of rare events, then you'll be able to accumulate the products of rare events. Life has such a mechanism. It's called natural selection. So of course most organisms look like they result from a series of very rare events. They do. Another thing that's exceedingly rare is for you viewing this to have joined my Patreon. But if you like this kind of thing here on YouTube and you want to see it stick around as long as possible, why not consider joining and making it a bit more common for my viewers to be supporters? The price starts at only $1 a month and there's a 15% discount if you join with an annual pledge. You get early access to videos like this and access to my supporters-only Discord server. Link in the description.
Far too rare to salvage evolution, even over the assumed multi-billion-year time span.
And of course, Carl has no numbers to back that up. Just trust me, bro. Well, I don't trust creationists, because I've consumed too much of their content and spoken to too many of them. None of them can be trusted. In some cases, it's because they blatantly lie. But for most of them, it's simply because they're woefully undereducated on all the relevant fields. And so everything they say has a higher than even chance of being wrong and needing to be checked on.
To illustrate further, the molecule hemoglobin in man carries out its project of transporting and delivering oxygen in red cells in a functionally efficient manner.
Is he about to tell us that that isn't relevant to survival or reproduction? Because, it super is.
A gene, or sentence, exists which codes for the production of hemoglobin.
It's actually more than one gene, because hemoglobin is actually composed of more than one protein, which are later joined together. See what I mean? I don't think Carl is lying here. I think he's just incompetent. So why would I ever trust him on anything he says? He's shown himself to be incapable of having this conversation in an intelligent way, And also, he's almost certainly lying about some of this, so no one should ever trust him.
There is a known mutation, actually 3 separate ones giving the same result, in which only one letter in the sentence has been accidentally replaced by another. If you inherit this change from both parents, you will be seriously ill with a disease called sickle cell anemia.
That's true.
Or at least it's true that you'll be sick, and he does seem to be right that it is caused by a single point mutation, specifically in the 6th codon in the beta-globin gene.
And will not survive for very long.
That'll depend on medical care availability, but because the condition is most prominent in areas without easy access to quality medical care, that is sadly true.
Yet evolutionists frequently use this as an example of a beneficial mutation.
Yeah, because when a person carries one sickle cell allele and one normal beta-globin allele, they are resistant to malaria and therefore able to produce more than those who have either 2 sickle cell beta-globin alleles and thereby tend to die of sickle cell disease, or those who have 2 normal beta-globin alleles and who then tend to die of malaria. Because having the heterozygous condition leads to significantly increased resistance to malaria. This means that in an area with a lot of malaria, those who are resistant to it will reproduce more. And that the condition is seen in such high proportions in places like sub-Saharan Africa, where malaria is most prominent, is a prediction of the idea that the sickle cell allele is beneficial.
This is because if you inherit it from only one parent, your red cells will be affected, but not seriously enough to affect your survival, just enough to prevent the malaria parasite from using them as an effective host.
Yup.
Hence, you will be more immune to malaria, and better able to survive in malaria-infested areas. The mutation-caused gene for this disease is maintained at high levels in malaria-endemic regions by this incidental phenomenon of heterozygote superiority. Its damaging effect in a proportion of offspring is balanced by the protection it gives against malaria. It is decidedly not an upward change.
There's no such thing. There is only the genome of organisms and their environment. Evolution isn't a ladder. It's not a great chain of being. There is no hierarchy of awesomeness. Like the rest of the natural world, evolution doesn't particularly care about you or what you want. It simply is.
We have not seen a new, efficient oxygen transport mechanism, or its beginnings, evolve.
Well, yeah, all lineages that need oxygen transport mechanisms evolved good ones like 2 billion years ago. Why would a new one need to evolve? What lineage needs better oxygen transport that isn't likely to get that by just further refining their own mechanism or amping up its activity? I'm not aware of anything or anyone in evolutionary biology that would predict that we should see new oxygen transport mechanisms evolving today.
We have not seen the hemoglobin transport mechanism improved.
Whoops, that's not true. In fact, beneficial mutations that occurred naturally in human globin genes are currently being investigated for gene therapy to use to treat sickle cell disease. Maybe it was true when Carl wrote this, but in that case, I think it counts as a failed creationist prediction.
One more loose, but possibly useful analogy.
I bet this analogy is worse than useless rather than being useful.
Let us say an undercover agent is engaged in sending a daily, reassuring telegram from enemy territory. The text says, "The enemy is not attacking today." One day, an accident occurs in transmission and the word "not" is lost. This is very likely going to be a harmful change, perhaps even triggering a nuclear war by mistake. But perhaps, in a freak situation, it could turn out to be useful, for example by testing the failsafe mechanisms involved. But this does not mean that it is the sort of change required to begin to convert the telegram into an encyclopedia.
Okay, so far it's at best useless.
The very small number of beneficial mutations actually observed are simply the wrong kind of change for evolution.
Yeah, that's not really a sentence that means anything. We see new genes that gain new functions while either losing or keeping their old function. We have even seen whole new metabolic pathways evolve in real time. The fact that Carl wants to cherry-pick mutations that he personally doesn't like that are nonetheless beneficial does nothing to change that. And let's remember, that he was just dead wrong about there being no beneficial globin mutations.
We do not see the addition of new sentences which carry meaning and information.
Whoops, except for antifreeze proteins in fish, digestive enzymes in everything from bacteria to mammals and more, new genes are actually not all that rare. There are plenty of examples.
Again, surprisingly, one often reads creationist works which insist that there is no such thing as a beneficial mutation.
Yeah, like Flat Earth, you have to tell lies to support special creation. I'm glad Carl is calling out some of those lies, but he's just spreading some of his own in their place, which isn't really an improvement.
If benefit is defined purely in survival terms,
which is not, it's defined purely in reproductive terms,
then we would not expect this to be true in all instances. And in fact, it is not.
Correct.
That is, there are indeed beneficial mutations in that sense only.
And that's the only sense that exists in evolutionary biology, and the only one evolutionary biology needs. Last episode, I gave examples of Wikipedia's list of characteristics of pseudoscience, and one of them was using technical jargon incorrectly. And in this case, that's what Carl is doing with the word beneficial. He's pretending that the actual definition of beneficial in evolutionary biology isn't what it actually is, then proposing his own alternative, which is also wrong. This is pseudoscience par excellence.
Information depends on order, and since all our observation and our understanding of entropy tells us that in a natural, spontaneous, unguided and unprogrammed process, order will decrease, the same will be true of information.
First, the second law of thermodynamics has nothing to do with order. It states that in an isolated system, entropy must increase or stay the same. Well, that has some terms that need definition. Entropy is a measure of the energy in a system that is not available to do work. Work is the displacement of mass over time, so it's energy that can't be used to move anything. A system is just whatever is under consideration, and it is arbitrarily defined by the person doing the thermodynamic analysis. Isolated, in the case of thermodynamics, means that neither matter nor energy can enter or leave the system. So entropy has nothing to do with order, and if we take either organisms or Earth as a system, they are not isolated, so the second law of thermodynamics doesn't apply to them anyway. In fact, it's worse for this. Life actually accelerates the rate at which entropy increases on Earth by exploiting energy gradients that exist, usually chemical or based on sunlight. Every time you eat something with calories, that food contains chemical energy in the bonds of the chemicals like carbohydrates and sugars. You break those bonds in order to release that energy to fuel your own metabolic processes, and the amount of energy you actually use is lower than the total amount in those chemical bonds. The rest is lost to heat that you can't use. That means you've turned some of that energy into heat that can't be used to do work. You've increased entropy in your own body. But it's okay, because you can take in new energy the next time you eat, so you're not an isolated system. But it's even worse than that. You see, the interactions that increase entropy are interactions that can happen spontaneously. In fact, in some ways, spontaneous interaction and interaction that increases entropy are basically the same thing. And you know what? Complexity tends to increase the rate at which entropy increases, because complexity, if measured by number of chemical interactions, means more reactions, each of which increases entropy if they're spontaneous, which means that actually complex chemical systems are thermodynamically favored for as long as there is an energy gradient, like maybe the one caused by sunlight falling onto a planet. And life is just one such complex chemical entropy engine, which means that thermodynamics actually favors not just life, but complex life.
The physicist and communications engineer should not be surprised at the realization that biological processes involve no increase in useful or functional teleonomic information and complexity.
I'm not going to speak for either engineers or physicists, but evolution observably does cause new, useful, and seemingly purposeful genes to arise. It does it kind of a lot. So if physicists and engineers are under the impression that that doesn't happen, well, they're wrong, and they might want to spend a little time talking to people in the biology departments of their universities.
In fact, the net result of any biological process involving transmission of information, i.e., all hereditary variation, is conservation or loss of that genetic information.
Or the generation of new information, which is about as likely as conservation or loss. In fact, in many cases, loss of previous information is the gain of new information, such as in transpositions or point mutations, which replaces previous nucleotide sequences with new ones. And since Carl still hasn't bothered to define information, I'm still going with Shannon information. Now, I'm sure he'd say that that's not what he's talking about, but this isn't my first rodeo, and any definition I went with that would allow me to actually measure information would be one he'd object to. Which means this article isn't really being written in good faith, so I don't particularly care that he doesn't like the definition I'm using. Until he gives one, I'll just keep using it, and I would encourage you listening, even you, Gary, to do the same if you have these conversations with a creationist. Insist that they give a definition of information that would allow it to be measured in a genome, or just use your own definition that does allow that.
This points back directly to the creation of the information, supernaturally, in the beginning. It is completely in harmony with the biblical concept of a world made very good as a balanced, functioning whole with decay only subsequent to the fall.
I don't care if it's in harmony with that story or not. Nothing about biological evolution demands a divine starting point for genetics. And Carl has to tell lies to get to that point, and not all of them even helped him to do it.
This is the reason why there are inevitable limits to variation, why the creationist does not have to worry about how many new species the future may bring, because there's a limit to the amount of functionally efficient genetic information present, and natural processes such as mutation cannot add to this original storehouse.
Amazing. Every word of what you just said was wrong.
You know what else is amazing? My channel members, because they not only help keep the channel going, but they also get cool emojis for live chats, early access to scripted videos like this, and also access to my super secret Discord server, which you can only know about if you join. There's a join button below this video, and the price starts at just $1.99 a month.
Notice that since organisms were created to migrate out from a central point at least once and fill empty ecological niches,
objection assumes facts not in evidence,
as well as having to cope with a decaying and changing environment,
wouldn't that make the fall inevitable and thereby part of God's design, and so really God's fault, not humans' fault? Since according to this, they were created with really no other option. I don't know, I'm not a theologian. All I know is that this kind of smells like God being the author of evil. You know, like it says he is in Isaiah 45:7. But hey, it's not my problem. I'm not someone who thinks that taking the Bible literally is a good way to learn about the world.
They would require considerable variation potential.
Wow, it sucks then that that's impossible if only 2 representatives of any given lineage that persists to this day were on the ark, giving a maximum of 4 alleles for any gene. I guess the Noah's flood story is impossible for genetic reasons as well as hydrologic, thermodynamic, and geological reasons. Almost like it was a story about events that didn't really happen, written by people who had virtually no knowledge of things like thermodynamics, geology, hydrology, or genetics.
Without this built-in genetic flexibility, most populations would not be present today. Hence, the concept of biological change is in a sense predicted by the biblical model, not something forced upon it only because such change has occurred.
Except that it was forced on it historically because of such change. Like, we can go read older creationist literature, going all the way back to Linnaeus. Creationists consistently rejected as much change as possible, accepting only the minimum change that they cannot deny, and also always claim that they would expect this much change anyway.
The created kind.
This ought to be good.
The scriptures imply that this originally created information was not in the form of one superspecies from which all of today's populations have split off by this thinning out process, but was created as a number of distinct gene pools. Each group of sexually reproducing organisms had at least 2 members. Thus, 1. Each original group began with a built-in amount of genetic information, which is the raw material for virtually all subsequent useful variation.
By the way, I'm just going to let him describe his own model. I can't really critique it on its own grounds as the thing he believes. But Where the critique can come is where his model makes predictions that fail to live up to reality, or when another model makes predictions that, if true, would preclude his model, and they turn out to be true.
2. Each original group was presumably genetically and reproductively isolated from other such groups, yet was able to interbreed with its own group. Hence, the original kinds would truly have earned the modern biological definition of species. We saw in our dog example that such species can split into 2 or more distinct subgroups, which can then diverge, without adding anything new.
Something he had to tell lies about to support, I'd like to add.
And can end up with the characteristics of species themselves. That is, reproductively isolated from each other, but freely interbreeding among themselves.
Making interfertility useless as a criterion for falsifying the hypothesis that organisms are of the same alleged kind.
The more variability in the original gene pool, the more easily can such new groups arise. However, each splitting reduces the potential for further change, and hence, even this is limited.
And it creates a falsifiable prediction. The genetic variability found in paleogenomic sequences should be systematically higher the deeper into the past we go. This is demonstrably not true, so there's a failed prediction.
All the descendants of such an original kind, which was once a species, may then end up being classified together in a much higher taxonomic category, e.g., family.
I love the use of Linnaean families as the default level for the kind, because what makes up a family is entirely arbitrary. So what are the chances that in even a few cases, the God-created kinds would line up with the taxonomic groups that humans arbitrarily chose to label as families? Well, basically 0. If kinds are real, they should probably vary between genus and order depending on what we're looking at, and there really shouldn't be a consistent rank at which we find a large majority of kinds.
Take a hypothetically created kind A, truly a biological species, with perhaps a tremendous genetic potential. See Figure 1. For the sake of simplicity, the diagram avoids the issue of what is meant by 2 of each kind aboard the Ark.
Yeah, let's for the sake of simplicity ignore the most important event in the history of any gene pool that experienced it. That won't make the following discussion irrelevant.
However, the basic point is not affected.
I find that very hard to believe.
Note that A may even continue as an unchanged group, as may any of the subgroups. Splitting off of daughter populations does not necessarily mean extinction of the parent population.
And that's something I wish more creationists would realize. It's the very reason why asking if humans evolved from monkeys, then why are there still monkeys is a question that betrays a lack of understanding of evolution. And it's the same basic misunderstanding if instead you ask, if birds evolved from dinosaurs, then how can we see birds being eaten by dinosaurs in the fossil record? That second one is a question Answers in Genesis has asked repeatedly in the last few years, despite this article being recommended to me in a video from their Canadian branch. It's like they don't even read their own literature, never mind the scientific literature.
In the case of man, the original group was not diverged sufficiently to produce new species.
Really? So I wonder what Carl does with those other species in the genus Homo, like H. erectus, H. habilis, H. antecessor, H. capronensis, H. ergaster, H. floresiensis, H. georgicus, H. heidelbergensis, H. juluensis, H. longi, H. luzonensis, H. naledi, H. neanderthalensis, H. rhodesiensis, and H. rudolfensis.
Hence, D1, D2, D3, E1, E2, E3, P1, P2, Q1, QT, Q3, and Q4 are all different species, reproductively isolated. But all the functionally efficient genetic information they contain was present in A. They presumably carry some mutational defects as well.
This would be a much more compelling story if paleogenomics didn't already show that it's false. In fact, I'm honestly not sure that I'll have much more to say beyond cool story, bro, too bad we already know it's fake.
Let us assume that the original kind A has become extinct, and also the populations X, B, C, D, E, P, and Q, but not D1, D2, etc. If X carried some of the original information in A, which is not represented in B or C, then that information is lost forever. Hence, in spite of the fact that there are many new species, which were not originally present, we would have witnessed confirmation of most of the information, loss of some, and nothing new added apart from mutations, harmful defects, or just meaningless noise in the genetic information.
Or, sometimes beneficial mutations that can and do result in entirely new genes making new proteins. But we're pretending that's not a thing. It's amazing the degree to which just looking around at nature can show you that this whole story is nonsense.
All of which is the wrong sort of information change if one is trying to demonstrate protozoan to man evolution.
Good thing we actually do get such information, then, isn't it?
Classifications above species are more or less arbitrary groupings of convenience.
Yeah, including the family where the bozo set the kinds at. Holy, how is he so bad at this?
Based generally on similarities and differences of structure.
I kind of want to scream at this point, but I won't. I respect your oral integrity too much, dear viewer.
It is conceivable that today D1, D2, and D3 could be classified as species, belonging to one genus, and E1, E2, and E3 as species in another genus, for example.
That's actually pretty likely.
It could also be that the groups B and C were sufficiently different such that their descendants would today be in different families. We begin to see some of the problems facing a creationist, who tries to delineate today's representatives of the created kinds,
while pretending that other problems that they face, and that are insurmountable, in my estimation, simply don't exist.
Creatures may be classified in the same family, for example, on the basis of similarities due to common design, while in fact they belong to two totally different created kinds.
Wow, and you know what? We actually have tested those models, putting the best case scenario as determined by Markov chain Monte Carlo's statistical techniques against the prediction of evolutionary biology to determine whether all primate families could be not related at all or were actually the result of common descent. It was the harshest crushing of a hypothesis that I have ever seen in any paper, and the hypothesis it absolutely crushed was separate ancestry. We cannot actually quantify the chances of separate ancestry being true given the genetics of primates. It's literally 0, according to the paper, in several tests, and range from 3 times 10 to the negative 4th to 2 times 10 to the negative 219th. In other words, no. We know with more confidence than we know almost anything else that common ancestry is true for all primates, humans included, and that separate ancestry is not.
This should sound a note of caution against using morphology alone, as well as pointing out the potential folly of saying, in this case, the Baramanon is the family, in this case, it is the genus, etc.
Yeah, because it's literally impossible to not only determine where a kind is, but also we know that there are no such things with more certainty than we know where the moon is.
Baraman is an accepted creationist term for created kind.
Oh hey, making up bespoke technical jargon not actually used in the relevant field is also a sign of pseudoscience. But also it's horrible Hebrew. You can't just stick a word that means he created in front of a noun and think that you've made a compound noun you can just stick the Greek suffix ology onto. That's not how Hebrew or Greek works. You'd think that since they love Genesis so much that Answers in Genesis would get some Hebrew speakers on staff to figure this shit out, but apparently not. Of course, they're also the ones who are notorious for showing Hebrew texts upside down, because that way it looks left justified. Because of course all languages are just really relexes of English, and work just like English does, down to the grammar and orthography. Which is the thing you would have to think to justify the word baraminology and upside-down Hebrew.
There is no easy solution as yet to the problem of establishing each of these genetic relationships.
Yet there should be, since each gene pool would be entirely independent of any other gene pools, meaning that you couldn't find any hierarchical pattern of mutations or retroviral insertions or chromosome structure above the kind level. and yet we do, meaning that life just isn't arranged into a bunch of separately created kinds. It's not a possibility that the data has left open to us.
In fact, we will probably never be able to know them all with certainty.
It would be super cool if just one could actually be demonstrated, and not just assumed, which is what they do with humans. Alas, such a thing has yet to come to pass.
Interbreeding, in vitro fertilization experiments, etc., may suggest membership of the same baraman, but lack of such genetic compatibility does not prove that two groups are not in the same kind.
Making baramins, or maybe baramanim, non-falsifiable under this testing regime, and so not a scientific concept.
See earlier discussion, genetic barrier could arise via mutation deterioration.
Nah, I'm not going to see it because mutation degeneration isn't a real thing in the way Carl means it, and because I don't dispute that speciation, including reproductive isolation, can occur.
However, newer insights enabling us to make direct comparisons between species via DNA sequencing open up an entirely new research horizon.
Yeah, one that utterly destroyed creationist predictions.
Although the question of where the funding for such extensive research will come from in an evolutionary-dominated society remains enigmatic.
Creationists managed to put together a multi-million-dollar building with a wooden facade that looks like Noah's Ark from one side. They could get together some funds for research if they wanted to. But by and large, they don't want to, because they don't care about what the evidence says. They reject evidence that they don't like and cherry-pick or make up evidence that they do like. So what's the point of wasting money on research? That's why they do things like write books for lay audiences and build Jesus-themed parks rather than actually getting any work done in science that's of any significance at all.
What then do we say to an evolutionist who understandably presses us for a definition of a created kind or identification of same today?
Just be honest and say that it's an unfalsifiable bit of pseudoscientific jargon that was made up to explain away evolution that is too obvious to exist, and to get rid of the problem that the ark is way too small to carry enough animals. Of course, no professional creationist would ever be that honest, they just don't have it in them.
I suggest the following for consideration. Groups of living organisms belong in the same created kind if they have descended from the same ancestral gene pool.
Okay, but that just means that all of the evidence says that there is only one kind in existence, and that all extant life belongs to it.
To talk of fixity of kinds in relation to any present-day variants thus also becomes redundant. No new kinds can appear by definition.
Yeah, making this idea just evolution, but you quit after a while of tracing things back.
Besides being a simple and obvious definition, it is axiomatic. Thus, it is as unashamedly circular as a rolled-up armadillo, and just as impregnable, deflecting attention, quite properly, to the real issue of genetic change.
For which there is ample evidence, making the term useless to creationists. I mean, I'm not going to stop creationists from shooting themselves in the foot.
The question is not, what is a baraman? Is it a species, a genus, or a family? Rather, the question is, which of today's populations are related to each other by this form of common descent, and are thus the same created kind?
All of them, according to every scrap of evidence, and in some cases that claim is so overwhelmingly well supported that it makes creationists' attempts to make up numbers so big or chances so small that they seem ludicrous appear as if they were attempts to seem impressive by a toddler who's watched too much pro wrestling and now wants to seem tough.
Notice that this is vastly removed from the evolutionists' notion of common descent.
Is it? Because it seems to be virtually identical, except that creationists aren't willing to actually follow the data to the end, and they want to give up partway through because it makes them feel uncomfortable to be related to bonobos, bananas, and bacteria.
As the creationist looks back in time along a line of descent, he sees an expansion of the gene pool. As the evolutionist does likewise, he sees a contraction.
Good thing we can actually extract the genomes of some ancient creatures and test this to see which model actually gives better predictions. Spoiler, we did that. It's evolution. As it ever is whenever evolution and special creation have different predictions.
as with all taxonomic questions, common sense will probably continue to play the greatest part.
Not in science, it won't. It plays no part in science. Common sense tells you that the Earth is flat and stationary, that heavy things fall faster than light things, and that there is no speed to light. All of that is very wrong.
The scriptures, the fossil record, and common sense unite to prevent creationists doing too much lumping together as we go back in time.
Well, I guess since only 2 of those have anything to do with the question, we can see why creationists are so god-awful at science. They accept 2 sources for their ideas that are just terrible at providing correct answers to scientific questions: common sense and the scripture.
For instance, it is conceivable, though not necessarily so, that crocodiles and alligators both descended from the same ancestral gene pool which contained all their functionally efficient genes, but not really conceivable that crocodiles, alligators, and ostriches had a common ancestral pool which carries the gene for all 3.
True enough, I guess. And with that, we're finally back to our Answers in Genesis Canada hosts. It's been a while. It'll almost be nice to be back to them.
Where Dr. Carl Whelan talks about these changes in living things, they're taking things in the wrong direction.
Right.
They're not getting better, they're getting worse. And that's really...
Not true?
That's really the answer to... We'll go into more detail here, but that's the answer to the question.
Not unless the question is, how do creationists lie about observed genetic and evolutionary phenomena?
I mean, really, the thing people need to understand right away is what a mutation is.
It's any variation in the genome from an organism that was not inherited from an ancestor in that state.
You know, if you watch popular science fiction, mutations are good things. They give you special powers.
X-Men.
And all that kind of stuff.
I don't think anyone actually thinks that the X-Men are real. Mutations are real, and clearly the X-Men aren't. So reality doesn't seem compatible with the idea that mutations will give you laser eyes, shape-shifting, weather control powers, absurd healing factor, or the ability to make playing cards explode.
But when you understand that DNA, obviously a language or code system, we've discussed this before.
And I've discussed it before too. It's not a language, it's not a code. It's a chemical system that has a few loose similarities to a code, but fundamentally acts in a way completely foreign to linguistics. There's a reason why geneticists don't normally co-author papers with linguists. It's because neither has any insights to offer the other, because the objects of their study are not similar to the degree that those disciplines have insights that would be useful to the other.
Made up of these chemical letters, when there's spelling errors, that's what a mutation is. As DNA makes copies of itself, you get all these spelling mistakes that can occur. And really what the evolutionist is now saying is if we start off with something simple, the Little Red Riding Hood, if you copy Little Red Riding Hood enough times, you make enough copies with enough spelling errors, millions of times, it's going to turn into the works of Shakespeare sooner or later.
That is in fact possible, but also DNA isn't anything like a copy of Little Red Riding Hood, so we don't need anything like that to happen. All that happens is that the genome determines in large part the phenotype of an organism, and that phenotype is either more or less fit than other phenotypes, and so the underlying genotype either spreads or diminishes in that population. Evolution doesn't have a goal like one might have in terms of hoping that a children's story turns into Shakespeare. Evolution isn't trying to produce humans or anything else. It's just what happened to occur.
Okay.
Really, that's
Which
what you're...
seems absurd when you say it that way.
Right.
But yet it's a legitimate analogy.
No, it's as absurd as it sounds, because it was designed to be absurd in the first place. That's the point of it, to make evolution seem as implausible as possible. That it does sound that implausible isn't exactly groundbreaking.
Well, yeah. I mean, you get a little bit of information, turn into a whole bunch of information, and it's all got to be meaningful with punctuation and purpose and all these types of things.
Right.
Well, it doesn't have teleological purpose. It has teleonomic purpose, as their guy, Carl, already pointed out.
So, we did have some people write in and try to give answers to the question.
As with the other questions.
Yes. And so answer number 1 came back and it said, if only 8 mutations per year were passed on for 3 billion years, that gives 3 gigabytes of information.
Well, that's going to depend on what those mutations are. Are we assuming single point mutations? What if some of them are deletions? You have to be much more specific with this. Person who I don't even know is real and who wrote into a creationist show probably like a decade or more ago.
Which is true.
The 3 billion DNA letters in the human genome.
Right.
I guess we were assuming that it's actually an addition of a single nucleotide each time. Weird, but yeah, I guess that's true. This is a weird response, since most populations don't accumulate mutations nearly that fast. And also, single nucleotide additions are not very common mutations compared to the sum of all other mutations. And we also know that the vertebrate lineage has had at least one full genome duplication, which is probably where the bulk of the genome comes from. So all in all, this is not how I'd respond to any of this.
However, there's a challenge with this. You need to define that. For example, it's not just if only 8 mutations were passed on. It would have to be 8 information-gaining mutations that coded for some new function or feature or special ability.
No, actually, it would just have to be single nucleotide insertions and you'd have that amount of information. The amount of information is the amount of information regardless of what it does. Noise is information. In fact, it's generally more information than the so-called meaningful parts of the signal. But really, the problem that the creationists are putting forward isn't how did the actual physical bulk of the genome come to be, but more how did new genes or new functions for old genes come about? And the answer to that is a combination of things like subfunctionalization, neofunctionalization, gene duplication, promoter duplication, and such things. All of which are observed, none of which are hypothetical, and all of which can combine with selection or in some cases drift to create new genes, new metabolic processes, and even new organs.
That wasn't in the organism to begin with.
This part is where I want to point out that creationists are usually not asking this in good faith. You see, when presented with nylonase, a brand new enzyme evolved by some bacteria to break down and digest, yep, you guessed it, nylon, I've seen the response that since nylonase is a modified enzyme that previously existed, it doesn't count. But that's how evolution works. It's modifications of what came before. If that doesn't count as evolution, then nothing does. But that's the thing. Creationism isn't about facts, evidence, or science. It's held to despite those things, not because of them. It's a pseudoscience, pure and simple.
Right.
So if you're a bat and you don't have echolocation, and all of a sudden you have echolocation, that would be a neat new mutation because it would give you a new ability, right?
Sure, but echolocation is not the result of a single ability. But also, being able to echolocate isn't a binary state. Some blind humans can do rudimentary echolocation by tapping a cane forcefully on the ground and listening to the reverb to determine the approximate size and shape of the space they're in. It doesn't actually require special mutations to echolocate at this basic level. But if you're a nocturnal animal who's hunting for insects and navigating a complex 3D world, you're already using all your senses, and your chittering, which you already use to communicate with your conspecifics, is also giving you feedback in the form of echoes. Echolocation in the sophisticated form that bats and whales both use is a refinement of hearing and normal animal calls. Just like with the nylonase, it's a refinement of what came before. It doesn't pop into existence ex nihilo. The same is true for basically any complex body part or behavior you can name. Jaws are actually modified gill arches, teeth are modified skin structures, arms and legs are modified fish fins. Even the eye is basically modified skin.
If you don't have teeth and all of a sudden you get teeth through a mutation?
Which again isn't how this works. Teeth probably started out as denticles, the kind of skin covering that sharks have, which are essentially tiny teeth that cover the skin. Then a mutation that made them more prominent in the mouth occurred early in the vertebrate line, which was obviously useful for food acquisition. Then they became more and more specialized for food acquisition and processing, while the ones on the rest of the skin were replaced by other kinds of skin covering, which is why animals like humans have teeth in their mouths, but not all over their body, which is already a rather horrifying concept.
That would be a good, beneficial mutation.
The more and more study that's done on the human genome and other animals as well.
Yeah.
The notion of junk DNA, which was popular years ago,
has only been confirmed. Junk DNA, that is, non-functional DNA, makes up the majority of the human genome and basically every other animal genome we've looked at.
Is falling by the wayside. Most of those 3 billion letters code for something valuable, for something important.
Nope, we know for a fact that most of them don't. And the way we know is twofold, and both ways are pretty simple. The first is knockout tests. Now, it's obviously unethical to do these tests on humans, but it turns out that mice and humans have very similar genomes because rodents and primates are actually fairly closely related. You can take a mouse genome and remove like 80% of it, and have a fully functional, happy, healthy mouse that can even grow to have happy, healthy mouse pups when it meets another mouse of the opposite sex. The other way we know is that you can see the different rates at which different parts of the human genome accumulate mutations by doing comparative studies and then look at regions of high or low variation in the genome between individual humans. And what we find when we do that is that the areas that are not known to regulate, promote, or code for anything have much higher rates of mutation accumulation, indicating that they are not sequence constrained. That is, nothing about the sequence of the nucleotides in that bit of DNA has any effect on the survival of the host organism. because changes there aren't being affected by natural selection. No creationists have, to my knowledge, proposed a function for these regions of the genome that does not require sequence specificity. And if a function requires sequence specificity, and that function is actually useful to the organism's reproductive output, then it will be under selection.
So
yeah, I mean, we're going to
response.
talk about the mutations that we see and what they actually do, and the fact that you can have beneficial mutations. But we need to define this as, it would have to be 8 mutations that added genetic information that was useful and meaningful, et cetera, et cetera.
Right.
And you know what? That's assuming that those things actually occur, and we haven't seen it.
Yes, we do, and have already listed many examples in this series, so I won't do so again. But this is a lie, and I suspect it's one that Calvin just told knowingly. He has been doing this long enough, even at this point, to be exposed to other counterexamples.
That's the problem. I mean, you're saying this is. It's kind of like saying, well, if pigs had wings, they could probably fly.
I don't know. Lots of things that can't fly have wings anyways, like some species of beetle, ostriches, kakapos, etc.
I mean, I don't want to be really facetious here, but it is like saying that, because if you haven't seen it, if it's not happening, if science isn't recording that, it's just a wishful...
Yes, it's an if-then.
Yeah.
If this happens, then this could be
that
true.
That could be true, and so on. So we haven't seen that.
Well, we have seen beneficial mutations that create whole new proteins and metabolic pathways, but what we haven't seen is any evidence of a global flood or God creating anything at all, let alone living things. Seems like we have a bit of a pot calling the kettle black. Also, my kettle is chrome and electrical. Maybe we need a new saying. It's like the creationists calling the flat earther a liar. I don't like it. I'll workshop it. If you have suggestions, put them in the comments.
And second,
so that's our first response to that question.
That's right. We just haven't.
If you have a whole bunch of time and you have these mutations passed on generation to generation, and he said 8. Did he not say?
Yep.
If only 8 mutations were passed on in 3 billion years. We need to do another show on this, but
right.
It's actually much worse than 8.
My dino sense is tingling. I think he's about to go talk about genetic entropy.
It's 60 point mutations is the latest figure.
Right.
Latest figure for what? But before we see if that question is going to be answered, I want to remind you to hit like, share this video, and leave a comment before you move on to something else. You probably know that YouTube can be very fickle, and as of the time of recording, this has been hitting a lot of channels with lower views and watch times. So everything you can do to help the video reach more people helps a lot. It's free to you, but it means the world to me.
But that's only point mutations within the human genome. 60 per generation.
Right, that are occurring right now.
Yes.
So more than the 8 that were being talked about. I guess that's probably significant to something, but I don't even follow where this is going. And I can follow along when my geneticist friends talk about their work. So I don't think it's a me thing.
And of course the problem is most of them...
They're not they're information destroying.
No, they're information changing, and the vast majority are in non-functional and so non-sequence constrained regions. Let's just assume that all loci in the genome have an equal probability of mutation, which is actually very generous because coding regions actually have fewer mutations, but let's just assume. That means that 48 of the 60 are going to be in regions where they don't matter at all. On average, about a third of point mutations will result in no change to the actual function of a codon. They won't matter either, bringing us down to 8 mutations that will actually likely change anything. On top of this, most proteins are pretty robust to having a large portion of their amino acid sequence altered, which is why there are more than 1,000 variations on hemoglobin across the human population. So of those 8, chances are that most of them will do absolutely nothing, and in a few people they will cause a problem, and in some much smaller proportion they will be a benefit, conferring something like what we've already seen: lactose tolerance, stronger bones, stronger muscles, resistance to malaria, resistance to heart disease, resistance to HIV infection, etc. All of these are documented mutations that have occurred in humans.
They do what mutations do. They wreck things.
Right.
Except when they don't.
And so if that had been happening for 3 billion years, that would have been a problem.
Yes, if evolution proceeded according to the straw man version that creationists like to make up, it wouldn't have worked. Good thing that the version bears only a passing resemblance to the actual model of evolution and common descent.
So second, one of the problems is, as the population grows larger, it's harder to fix new mutations in the population. This is because of the cost of substitution. This is what they call Haldane's dilemma.
Oh, Haldane's dilemma. I guess not technically genetic entropy. Of course, one of the things I want to point out is that Haldane was not saying that the dilemma was real. It couldn't be, because if it were, then according to Haldane, most species couldn't survive 300 generations. He was highlighting that some of the models of genetics used in his day had to be wrong on some level. This is usually what's happening when a scientist proposes a paradox or dilemma. One of my favorite such proposals was Olber's paradox. You see, in the early 20th century, it was known that the universe was bigger than the Milky Way, and most astronomers thought it was more or less static, infinite in size, infinite in age, and homogeneous at the largest scales. But if this is true, then there is no single angle that one could look into the sky from that should not intersect the surface of at least one star, barring the very rare shadow from a planet or a moon. This should mean that literally every part of the sky should be about as bright as the surface of the sun. Clearly, this is not the case, which means that something about the eternal static model of the universe had to be wrong. It turns out that what was wrong is that the universe is neither static nor eternal. Haldane's dilemma and the Olber paradox don't show that reality is wrong, they show that some part of the theory or model is wrong.
And I guess we should probably spend a little bit of time here, because if
You're going to explain that.
Yeah, if people aren't familiar with it, we had Dr. Don Batten. He's a scientist with our organization, and he did
Calvin is too young for a Mitch McConnell moment here.
Article you can check out on the website. We'll put the name up here. It's creation.com/Haldane.
Amazing. Get another bit of homework. At least this one isn't a terribly long article. Well, let's go and make sure you show your appreciation for Ferrara yet again lending his voice to the channel.
Haldane's dilemma has not been solved.
I mean, it has though, but hey, let's see what Don Batten has for us.
The famous evolutionary geneticist J.B.S. Haldane, 1892 until 1964, was one of the three founders of the field of study known as population genetics.
Along with R.A. Fisher and Sewell Wright, friend of the channel Zach B. Hancock has a video about the three of them, and you should check it out.
Haldane articulated a serious problem for evolutionary theory in a seminal paper in 1957, The Cost of Substitution.
Well, it was at one time a seriously puzzling question, but now it's seen kind of like the problem of the changes in the precession of Mercury. You see, all the planets seem to follow Newton's laws quite well, except Mercury. The orientation of its orbit, or more technically its argument of perihelion, shifted more than Newton would predict. This led to people hypothesizing that maybe there was a planet even closer to the Sun that was perturbing the orbit of Mercury. It was called Vulcan, and although various people claimed to see it, it was never confirmed to exist. And now we know that it did not exist. So what explained this problem for Newtonian mechanics? Well, we didn't really have a full picture of gravity, and while Newton's laws worked well for most things we observed, in extreme circumstances, they didn't work anymore. It took Einstein's special relativity to give us a mechanical and theoretical framework to resolve the problem. Similarly, Haldane's dilemma was resolved by further work in the theory of evolution, which brought us a better theoretical understanding of evolution and population genetics, much like Einstein's relativity work brought us a better theoretical understanding of gravity.
When a beneficial mutation occurs in a population, it has to increase in the number of copies for the population to progress evolutionarily.
Creationists just can't use English normally, can they? It has to spread in order for the mutation to fix in the population. That's the phrase he needs there. Progress isn't defined in evolution. Evolution isn't goal-oriented. It doesn't push creatures to become higher versions of themselves, whatever that means.
If the mutation remained in one individual, then evolution cannot proceed. This is fairly obvious.
Technically, that's not true, since the introduction of a new allele is itself evolution, but I will assume that he means that there will be no long-term directional change, and that's probably true. Don is really bad at writing about evolution.
In other words, it has to substitute for the non-mutated genes in the population.
There are no such things. All the alleles of all the genes and the genes themselves are the product of previous mutations. There is no sense in which one organism is more or less mutated than another, except in how many mutations it has as an individual. But that sense is not heritable. If an animal's mother had 150 mutations and its father had 100, and on top of that the offspring had 50, then that offspring is less mutated than either parent by that definition. The level of mutation doesn't compound in any meaningful sense. This idea of there being mutant and non-mutant genes and alleles assumes that there is some initial diversity that does not come from mutation and that we can uncover in organisms. No one has ever been able to do that. And so until creationists can recover a non-mutation derived ancestral genome, it makes no sense to talk about some parts of the genome being not mutated. As it is, actual scientists have discovered that all parts of the genome have a coalescence time, a time in the past where the ancestral state for all extant members of the population shared an ancestral state. That is, the common ancestral state of that part of the genome. And that time varies across different parts of the genome, which cannot be true if some large degree of diversity just poofed into existence at one time.
But the rate at which this can happen is limited. A major factor limiting the rate of substitution is the reproduction rate of the species.
That's true.
And it makes sense. Elephants clearly can't spread a new allele as quickly as, say, mayflies. What with elephants being some of the slowest reproducing animals out there, whereas mayflies sometimes hatch, live, reproduce, and die in the span of a few months, and so can spread alleles orders of magnitude faster. The question, of course, is, are there any organisms for which their reproduction is so slow that we cannot account for their evolution within the time available to us? The answer is no. Or to put it in more words, the answer is that no such organism has been discovered. One group of organisms that I have discovered, much to my delight, are my patrons on Patreon. Patreon is probably the best way to support me, as it's where I get to keep the largest percent of what is sent to me. And joining there gets you early access to videos like this, access to my Discord server for supporters only, as well as other perks at higher tiers. It starts at only $1 a month, and there's a 15% discount for pledging annually. So why not go ahead and check out the link in the description and join the team that are helping me keep this channel going.
For a human-like creature with a generation time of about 20 years and low reproduction rate per individual, the rate of growth in numbers of a mutation in a population will be exceedingly slow.
This is something you need to look out for with science writing, or in this case, pseudoscience writing. Science is about numbers and facts. Saying something like exceedingly slow doesn't mean anything in science. There's not a particular threshold for what counts as exceeding. Exceeding in this case is a weasel word. It's there so that the creationist doesn't actually have to be tied down by any real numbers that can be checked on. Is human reproduction too slow? Well, maybe, but let's see how Don makes the argument.
This is basically the cost of substitution.
What is? The rate of reproduction? Well, that's not true. The cost of substitution, as well as the strength of selection, is well defined by Haldane. The strength of selection is the natural log of the proportion of those organisms with the optimal genotype who reproduce divided by the proportion of the entire population who reproduce. So if 80% of the population with the optimal genotype reproduce, and overall 75% of the population reproduce, then the strength of selection is 0.064, and the cost of selection is the quotient of deaths without reproduction of those with the optimal genome minus the same for the overall population, or in this case, 0.05.
Imagine a population of 100,000 apes.
Done.
The putative progenitor of humans.
I was imagining humans, since they are apes. Okay, I'll imagine something that's like the common ancestor of all apes, so a population of apes that existed a bit less than 20 million years ago or so.
Suppose that a male and a female both received a mutation so beneficial that they outsurvive everyone else.
Huh? That's not how that works. Beneficial mutations don't generally work by killing those without the mutation. I guess that could work, like a mutation that causes those with it to be aggressive to the point of killing those without it, but I'm not aware of any such mutation existing in real life. As you can tell from Haldane's definitions, it's about proportionality. If the general population before this mutation already had an overall reproduction rate of 0, then they'd just be extinct. Competition in evolution is rarely so direct that the more fit literally cause the death of the less fit. So already we're off to a bad and rather insane start.
All the rest of the population died out, all 99,998 of them.
Yay, math.
And then the surviving pair had enough offspring to replenish the population in one generation.
I feel like we're not talking about apes at this point, but okay. By the way, the strength of selection in this case is 8.5, which is a bit crazy.
And this repeated every generation, every 20 years, for 10 million years.
So half the time from the first ape progenitor of humans to humans. Okay, I feel like maybe Don didn't have the first apes in mind. It would be a lot easier if he just write clearly instead of writing like he has a rotten cabbage for a brain.
Rotten cabbages? What kind of slum do you think this is? Oh, my cabbages!
More than the supposed times as the last common ancestors of humans and apes.
Nope, it's about half that time. Maybe if by apes he specifically means chimpanzees, then that might make more sense, but he didn't say that. And of course, the last common ancestor of a human and any ape is just their parents if they have siblings, since the parents of any ape are an ape, and all humans are apes. Is this just supposed to confuse people into believing that science is wrong? Is that the tactic? Why does anyone ever let Don Batten near a typewriter or a word processor? He seems more like he should be in a padded room with crayons, but hey, I'm no psychologist.
That would mean that 500,000 beneficial mutation could be added to the population.
Yeah, sure, why not?
I.e. 10 million divided by 20.
Thanks for the math.
Even with this completely unrealistic scenario, which maximizes evolutionary progress,
there is no such thing as evolutionary progress. There is evolutionary change, and this in no way maximizes it, since it eliminates several of the things that speed up molecular change, such as neutral variations and genetic drift, parallel mutations, and recombination. And you may not be surprised to learn that a combination of these things neatly solves Haldane's dilemma. So let's talk about each one in turn. First, neutral variation and genetic drift. It turns out that the vast majority of mutations have no actual fitness effect. Yet, because some parts of survival and reproduction are chance-based, neutral variation in a population will change from generation to generation, and some neutral variations will become universal in a population's genome. That is, they will reach fixation. There is no cost of selection associated with this, yet it still accounts for large amounts of the variation between populations. Second, parallel mutations, which isn't really a technical term, because really all that's happening is mutations in different individuals in the population. In Don's hypothetical, we only ever have 2 organisms reproducing, and in real life, one organism can have a beneficial mutation, while another organism can have another beneficial mutation, and they can both increase in prevalence through the population at the same time. So unlike his thought experiment, in reality, we don't have to wait for each mutation to fix before the next can occur. Finally, recombination occurs during meiosis, that is, the process by which gametes or sex cells are produced. During this event, chromosomes of the same type will swap parts, so that the resultant chromosomes are often a mixture of material from different chromosomes. So in your egg or sperm cells, you will have chromosomes that are a mix of those inherited from both your parents. So your chromosome 4 in one of your gametes might be 80% from your mother and 20% from your father. This means that those different alleles that are beneficial that are coming about in parallel could end up on the same chromosome, even if they didn't start that way, allowing beneficial mutations to accumulate. All these processes happen in parallel, and all of them are unaccounted for by Haldane. These are the parallels to Einstein's inclusion of warped spacetime and the speed limit of the speed of light, which combined to explain why Mercury was acting weird all this time. Haldane didn't know about genetic drift, recombination, and did not really account for parallel mutations. As a result, his calculations were wrong when actually applied to real organisms. That Don is repeating and in some cases exacerbating these problems is just evidence that he doesn't know what he's talking about, not that evolution is wrong.
Only about 0.02% of the human genome could be generated.
What? Why does it need to be generated? All the previous species of apes had a genome about the same size as the human genome. I honestly have no idea what he's trying to get across with this. Does he think that the ancestor of humans and chimpanzees had no genome? That seems pretty dumb even for a creationist.
Considering that the difference between the DNA of a human and a chimp, our supposed closest living relative, is greater than 5%, evolution has an obvious problem in explaining the origin of genetic information in a creature such as a human.
No, it doesn't. So the first degree of difference in the DNA isn't an actual measurement. There's no single overall number that you can use as the difference between humans and chimps in their genomes. That's because genomes are complex. For example, let's say one lineage has a section of DNA that gets reversed in a mutation, and that fixes. Let's say the reversal is 20 base pairs long. Is that 20 differences or one difference? It depends on how you count, and there is no one right answer. So the question of how many mutations would it take to get from the common ancestor of chimps and humans is that there's no easy way to know for sure. Second, Don seems to assume that all the change was on the part of the human lineage, but there's no reason to think this. It should be closer to 50% of the difference arising on the human side and 50% on the chimp side. Although in actuality, there is some reason to think that chimps might actually be more derived than humans, and so it might be more like 40% on the human side and 60% on the chimp side. Third, this whole idea is based on his ridiculous strawman of how evolution works that doesn't take into account any of the reasons that are given to resolve Haldane's dilemma. It's like complaining that relativity is false and pointing at Mercury, the very thing that was solved by relativity.
However, with more realistic rates of fitness or selection and population replenishment, the number of beneficial mutations that can be accounted for plummets.
It does the opposite, in fact, as I've already gone over.
Haldane calculated that no more than 1,667 beneficial substitutions could have occurred in the supposed 10 million years since the last common ancestor of apes and humans.
And he was wrong.
This is a mere one substitution per 300 generations on average. The origin of all that makes us uniquely human has to be explained within this limit.
No, it doesn't, because for one thing, fixation doesn't have to be selected for, it can be the result of chance. And for two, more than one mutation can be going towards fixation at a time, so that even if the average time for an allele to fix is 300 generations, the rate at which various alleles can fix in the population can be far, far higher. If just 10 alleles are going towards fixation in a population, then the average time for them to go to fixation can be 300 generations, while the rate of fixation within the population would be every 30 generations. Further, there is no guarantee that each of the differences between the genomes of humans and chimps make humans uniquely human in some sense. Some of them, probably most of them, are going to be neutral variation with no effect on phenotype.
A substitution is a single mutational event. It can be a gene duplication or a chromosomal inversion or a single nucleotide substitution.
Normally, only single nucleotide substitution is meant, but whatever. He can stipulate a definition for the purposes of this article, provided that he's consistent with it.
Biologists have found that the vast majority of substitutions are indeed single nucleotides.
I think that's true, but I'm not going to check. If you have a source that says otherwise, you should let me know in the comments, but do be warned that YouTube will hold comments with URLs in them, so just don't send a link.
So Haldane's limit puts a severe constraint on what is possible with evolution, because 1,667 single nucleotide substitution amounts to less than one average-sized gene.
He really does seem to think the genomes just grow one nucleotide at a time by inserting one base pair every once in a while. This reminds me of talking to Jungle Jargon, a creationist of no consequence, and having him tell me, in all earnestness, that batteries work by storing electrons, and to charge them was to pump electrons into them, because apparently the power going to your house is just an electron pipe that sends electrons into things that you power with it. This misunderstanding is so fundamental that it's hard to imagine anyone with an advanced degree would have it. Yet reportedly, Don Batten has a PhD from the University of Sydney, which is a rather prestigious and long-standing institution. In case it's not obvious, genes are not built one nucleotide at a time, nor are genomes. Whole large chunks are often added, and new genes normally come about through a mutation creating a new gene directly from pre-existing sections of the genome in a process called neofunctionalization. The rate of single nucleotide substitutions or additions has nothing to do with how big the genome is.
It should also be noted that Haldane made a number of simplifying assumptions that actually optimized the number of substitutions that would be possible.
Except that we know that many of his assumptions, such as no crossover and no sexual reproduction, greatly reduce the number of mutations that can fix in a population. So even if some other of his assumptions increase that number, his assumptions overall vastly underestimate it. And we can tell because if he were right, humans wouldn't be here to talk about it. If you apply Haldane's dilemma to humans, humans couldn't make it 2 or 3 generations. Yet I see humans every day, and near as I can tell from looking into genealogical information, most humans can trace their ancestry back more than 3 generations, and some of them can do it for dozens of generations.
So in reality, the number of substitutions possible is much less than Haldane calculated.
What I believe is dumb.
Furthermore, if we accept what the paleontologists say, that the vast majority of species' existence is spent in stasis.
That's morphological stasis, not molecular stasis. You can't tell that at some point in the existence of, say, Triceratops horridus, that a new, more efficient digestive protein evolved, and then swept through the population, fixing in place in only a few dozen generations. That's a hypothetical, by the way. I have no evidence of such an event. But the point is that if it had happened, we wouldn't be able to see it by looking at fossils, and such events have certainly happened, just like they happen now. Evolution in species that reach a morphological fitness maximum does not stop. And even further, neutral evolution will proceed regardless of how well fit to an environment a species is. So no, morphological stasis in the fossil record does not in any way indicate evolution not occurring.
Then the problem becomes even more acute. If 90% of the supposed 10 million years is spent in stasis,
which it's not, because we have many hominid species, and clearly they are not all the same.
Then that reduces the number to 167 substitutions.
No, it doesn't, because even if hominins evinced morphological stasis, which they absolutely do not, that wouldn't mean that they were not evolving during that time. Those things are not correlated. How have I been doing this for so long and I can still be surprised by how insufferably wrong creationists can be? And given Don's education, it's really hard for me to maintain that he could possibly be speaking or writing in good faith when he says things like this. If I came to you touting my degree in English literature and then told you that the famous book Frankenstein was written by Chaucer, you'd have to assume that I'm lying, either about who wrote Frankenstein or about my credentials. That's where I am with Don Batten. So if you're a creationist, please let me know which you think I should assume, that Don Batten is lying about genetics or that he's lying about his credentials.
Haldane's dilemma was discussed a bit in the 1960s, but since then has been largely ignored in the evolutionary journals.
Yeah, you know why things are presented as problems tend to later on not be discussed much in journals? It's because we figured out the solution to the problem. There's a reason that no one is discussing the weird orbit of Mercury much in journals. It's because that problem was solved by Einstein and it's no longer weird. You know why no one really talks about Haldane's dilemma? Because evolutionary biologists and population geneticists figured out the solution. That's how science works. If you want to revive discussion about Haldane's dilemma or the orbit of Mercury being anomalous, you're going to have to show why the solutions that everyone knows work don't work. Good luck with that if that's your plan, but the simple fact that a problem that's been solved isn't really discussed much anymore isn't really surprising. And it's certainly not evidence that the problem is, in fact, a problem.
Genetics and biology texts did not, and do not, discuss the problem. In 1992, the high-profile evolutionary geneticist George C. Williams remarked that the time has come for renewed discussion and experimental attack on Haldane's dilemma. His plea apparently had no effect on his evolutionary colleagues.
Probably because he was wrong. In 2025, astrophysicist Avi Loeb said that scientists should be talking more about the alien origin of extrasolar comets. It had no real effect on the astronomy community because it's fucking wrong. That's what tends to happen when you're wrong and a scientist. People who know you're wrong don't want to waste their time on you or your wrong ideas, because they're trying to actually learn things, which wrong ideas are bad at helping with.
However, creationist Walter Remine published a work in 1993 which treated the issue in detail.
You know, normally when someone talks about publishing something in terms of science, what they mean is that they published it in a journal after rigorous peer review. But do you think Walter did that? Pause the video and put in the comments if you think Walter's work was peer-reviewed. One group of people who would never cite non-peer-reviewed sources inappropriately in a discussion are my channel members. If you want to help the channel out and get access to videos like this months early, recognition in chat, once-a-month milestone chats, and access to my Discord for supporters, why not considering using the join button below this video? It starts at just $1.99 a month, and if you go to higher tiers, you can get more perks.
He has since worked on the matter, refining the argument and dealing with attempts at obfuscation by evolutionists, mainly anonymous persons on the internet.
The fact that Walter can't actually get any attention except from people like me, people who are not in the field, is not a sign of the quality of his work. I'm the kind of guy who responds to people like Roger from Mud Fossil University, which isn't a real university, or Raw Matt, the breatharian and ordained Essene, even though that isn't and has never been a thing. It's weird that Don Batten keeps giving me context for the work of Walter Remine that makes me think his work is really worthless.
Since the publication of Remine's book, there has been no serious dispute that Haldane's analysis, if correct, places a 1667 limit, a severe limit, on human evolution.
Sure, that's probably true. Just like if Star Trek is right, there's a severe limit on how much warp travel you can do in any part of the galaxy before the subspace in that region begins to degrade. Thing is, though, that that's not actually true. Just like Haldane's dilemma isn't actually a real problem. It was a problem with how biology was modeled, not how biology works. The model was wrong, and we fixed it. Well, I didn't. People who actually know what they're doing more than I do did that.
Remine claims that Haldane's dilemma has never been solved.
It's a mighty fine claim Don's got there. Too bad all of evolutionary biology disagrees with him. You know, you can't just say the puzzle of what fire is has never been solved just because you think phlogiston is cool and that oxygen is lame. It's still not true, even if the solution isn't the one you like. That the solution to Haldane's dilemma wasn't that humans are a recent creation by a supernatural being doesn't mean that the actual solution of recombination, natural selection, and the lack of need for mutations to fix before others arise isn't the right solution. And let's remember, Haldane's dilemma was never something that was going to turn out to be an actual insoluble problem, because just basic observation of large mammals like humans shows that it can't actually be true, since there would be no such animals if it were.
But rather has been confused, garbled, and prematurely brushed aside. The cause of the confusion largely has to do with misunderstanding or misconstruing the core concept, the cost of substitution.
Oh, I bet we're about to get a creationist redefining a well-defined term because otherwise they can't get their so-called problem to actually be a problem. You see, liars like John Sanford and idiots like Sal Cordova do this all the time with fitness, because they know that if they define fitness in the way that science does, the way that lets it be measurable, then fitness in populations can increase with no problem. So they have to make up an ill-defined wishy-washy version of it that means more like the kind of thing you go to the gym to get than actual reproductive success. That way, anytime anything gets worse at doing a thing, they can crow that fitness has actually decreased, even when it demonstrably has increased.
Some of the confusion could have arisen because Haldane was never very clear in his definition of what the cost was in physical terms, although he defined it mathematically.
No, he was extremely clear. It's genomes that do not go on to contribute to the next generation. Genomes are real physical things. Anytime a new genome forms, if it does not then go on to contribute to future genomes, it has, quote, died, in the sense that Haldane means. So if, as often happens in humans, an ovum is fertilized but fails to implant on the uterine wall, it is part of the cost of selection being paid. If it manages to implant but self-terminates as a result of some lethal mutation, it is part of the cost. If it is carried to full term but is infertile, it is part of the cost being paid. If a person fails to have children at all, that individual is part of the cost being paid. The cost is paid in genomes that do not reproduce for whatever reason. Ultimately, that means death without reproduction, but that death can be anywhere from fertilization all the way to a long, long life.
Many evolutionists have interpreted Haldane's cost of substitution in terms of death of individuals that do not have the mutation, genetic death, and superficially, this might seem reasonable.
Yes, because that's how Haldane defined it. If you think that it's not that, then you're talking about something other than Haldane's dilemma.
However, suppose competition with the mutant form causes lower birth rates for the non-mutant individuals. Then the genetic death cost becomes the number of individuals who would have been born to those who do not have the mutation, but were not born because of competition with the mutant form.
No, that's not what the cost of selection is. So we're not talking about Haldane's dilemma. Maybe this is Walter Remind's dilemma, but since apparently no one takes him seriously at all, I can see why someone would like to pretend that it's Haldane's dilemma, which is at least a thing in science historically.
In other words, it becomes a matter of virtual or imaginary deaths. It becomes an exercise in futility to try and model such a thing mathematically.
Yes, if you make up new definitions for mathematical terms that remove your ability to measure or estimate their value, that makes calculation impossible. Good thing Haldane didn't do that. How can Don think that making the problem incalculable is a good thing? If it's incalculable, then he can't really say it's a problem, since he can't even show that the problem exists mathematically.
On this basis, some evolutionists have dismissed Haldane's dilemma as imaginary, an unreal problem.
I'm pretty sure that no scientists have dismissed Haldane's dilemma as an unreal problem on the basis that creationists get it wrong. However, it was never a real problem for nature, it was only ever a problem for human modeling of nature. A problem which, again, is solved just by doing more detailed modeling and knowing that chromosomes can cross over.
In a paper in this issue, pages 113 to 125...
Yeah, this is copied from an article that appeared in print, and I'm already deep enough down the rabbit hole. I'm not going to check an article within an article. If I keep doing that, this will just become creationist inception, and I'll never actually get out of this.
Remind revisits this largely ignored problem for evolutionary theory. He significantly clarifies the nature of Haldane's dilemma as it has come to be known, by removing confusion over the meaning of the cost of substitution.
By just redefining it. Neat.
Remind defines and clarifies the cost of substitution such that no imaginary or virtual realities are involved. He puts it on a solid, physical and theoretical footing. Remain originally submitted this paper to mainstream, evolutionary, science journals.
Given how creationists normally feel about homosexuality, it's kind of surprising to see Don Batten glazing Walter Remind's knob this much. Also, why wasn't I just directed to something by Walter? This would be a lot easier if the 15 questions pamphlets just cited him instead of citing Don Batten fangirling all over Walter.
The paper has been under review for nearly 2.5 years, and it would appear that reviewers used every conceivable rationale to reject it.
First, some of the conceivable reasons for rejecting work from being published is that it's bad work. So thanks for telling on Walter, Don. Second, this has big Richard Carrier energy. Like when he was whining that by failing to pass peer review, he actually did pass peer review, which is something he did to cope with the fact that his most recent book, at the time of writing, isn't taken seriously by anyone who knows anything about the topic.
The shocking scandal. OP did undergo a full peer review at a real biblical studies press, but scandalously. The reviewers did not take it seriously, but only childishly slandered the book. When asked to identify any specific error of fact in it, they could deduce none. Instead, it was rejected solely on the grounds that it does not make a unique, original contribution to the field of biblical studies, which will help advance the discipline, and will not be interesting for readers within the discipline, and does not engage with the field/discipline. When you read it, you will be shocked at how resoundingly false all three of those accusations are, as Obsolete Paradigm does nothing but all three of those things. It is a serious work, thoroughly referenced and tightly argued. It is full of new findings and results. It thoroughly engages with all relevant arguments and publications of the field for the last 10 years. And it is essential reading to anyone within the discipline. In fact, their desire to avoid it with slander and fake charges precisely demonstrates its importance. The whole argument of the book is that the field is trying to hide from these findings and make them go away. The reviewers themselves ironically demonstrated the book's entire thesis is correct. The whole argument of the book is that the field is trying to hide from these findings and make them go away. The reviewers themselves ironically demonstrated that the book's entire thesis is correct. This means that OP actually passed peer review, because with respect to any real peer review, as in a full fact check for error or misstatement, Obsolete Paradigm passed. No specific error or misstatement in it was found, and the reviewers took 6 months to make sure. Instead, they had only blatantly Christian propagandist dismissals of the contents of the book to offer, and gave entirely bogus reasons not to publish it. And you can confirm this for yourselves by reading the book and seeing how absurd their claims are, and thus how desperate biblical historians are to avoid all of this. So now that it has survived a complete fact-checked by 2 hostile peer reviewers who could challenge no facts nor find any fallacies in it, I've published it with Pitchstone.
For example, with one journal, 2 reviewers noted that Remind's cost concept was different from the traditional view and claimed it was wrong.
Because it is. That's a good reason to reject a paper about something. If you get the thing that you're talking about wrong, you shouldn't be published on that thing. If I wanted to get published in a linguistics journal, and to do so, I decided that I was going to redefine grammar to mean the instrument which makes glyphs when writing, then I would be rightly rejected from publication. This is pretty basic.
After he showed they had misread the paper.
After he claimed they had misread the paper.
And that the paper is correct and makes correct predictions, the reviewers shifted to a different reason to reject it.
It can't make correct predictions because Haldane's dilemma never has. It's not possible that it's right. That's why it was a dilemma. Organisms don't actually produce as much as they would need to according to the dilemma. Despite this, they do in fact observably increase in fitness, and new alleles fix far faster than the dilemma would predict. These aren't questions we have to ask, they're observations. They're observations just like we use telescopes to observe stars. I don't care what theoretical objection to the existence of stars you can come up with, because when I look for stars, I can see them. I don't care what theoretical limits to the rate of fixation of new alleles you can come up with if it's lower than the actual observed fixation rates in real organisms.
With another journal, 4 reviewers, most paper are only reviewed by 2 other scientists, acknowledged Rick Mine's paper was correct, but 2 of them, high-profile evolutionary geneticists, rejected the paper on the grounds that they, and some of their associates, knew of the subject matter in the 1970s. In other words, they claimed the analysis was not original.
I'm skeptical of this claim. Who reviews a paper is usually anonymous, and so to claim that Walter knows that 2 of them were high profile means that he must know who the reviewers were. But that's unlikely. I think the story is made up. But even if it's not exactly surprising that creationists can't come up with anything new, since they rarely are much better than 50 years behind where science is, and are often more like 80 years or more behind.
However, they were unable to cite a single book or paper that contained this material. In other words, the journal had no legitimate grounds for rejecting Remine's paper.
Again, I really don't think this story is true, but failing to produce any previous work that has the exact analysis and results of Remine's isn't necessary to say that his work is unoriginal. And it's not like there's some obvious bias to suppress Haldane's dilemma. That's why, when it was still an open problem, it was in fact discussed. But no one wants to go around wasting time on a problem that was solved decades ago just because some random asshole thinks that maybe if it actually is a problem, then it proves that God exists. I'd also like to point out that many of the people who would be qualified to peer review Remine's book do in fact believe in God, even the Trinitarian God of traditional Christianity.
And, just as importantly, this response implicitly acknowledges that the evolutionary workers in the field knowingly allowed confusions and errors to thrive, overwhelmingly, even to the present day.
Does it? I don't see how it does that.
Haldane was aware his conclusions would probably need drastic revision. Remine's paper contributes to that revision. Not only does he clarify the concept of cost of substitution and show how Haldane's formulas can be rederived with the more robust concept, but he also generalizes the cost concept. He does this by eliminating the need for constant population size, small selection coefficients, etc.
Good for him. Too bad no one who actually knows what they're talking about find any value in it. It's almost like maybe, like many wackos who can't get published because they're obviously wrong, Walt is just full of himself and can't stand that people don't see his non-existent brilliance. It's almost like maybe he is acting narcissistically, something that is usually seen as negative, except apparently when someone is running to be the next elected king of the former United States.
Higher selection coefficients actually make the problem worse for evolutionists.
Okay.
Remine also deals with various attempts by confused evolutionists to circumvent Haldane's dilemma, e.g., that environmental change can alleviate the problem. The take-home message, which was not spelled out in his paper because it was written to be submitted to mainstream evolutionary journals, is the evolutionary origin of organisms with low reproduction rates and long generation times, many higher animals, is impossible, even given the usual millions of years assigned to the history of species on Earth.
Yeah, that was the conclusion that Walt selected before he began working, so it's not surprising he got there. But that's because he's not a scientist, he's a pseudoscientist. Scientists look for answers in the data, and pseudoscientists have the answers and look for data to support their conclusion. Walt, Don, and the 2 chucklefucks that are coming up next, now that we're done with this article, are all pseudoscientists at best.
And now the famous evolutionist geneticist, J.B.S. Haldane, he was one of the 3 founders in the field of population genetics.
That's true.
Now, he, as an evolutionist...
He's an evolutionist, first note.
Yes, he's passed away now, but he was an evolutionist, and he noted there was a serious problem which has become known as Haldane's dilemma. Now,
Yes, he noticed a problem with his model. Obviously, it's not a problem with reality, because whenever a model disagrees with reality, it's the model's problem, not reality. That, of course, is something that creationists and flat earthers, or wrong earthers as I call them collectively, simply cannot figure out. They're all acting like flat earthers, insisting that there is no 24-hour sun in the Antarctic summer because their model says there can't be, even after a number of prominent flat earthers went to Antarctica and saw the 24-hour sun firsthand, and after it was both live streamed and recorded in both real time and time lapse. Creationists are in the same boat. They have a model that they really like, and despite the fact that it doesn't match reality at basically any point, they insist that the model is telling them the truth and that you should ignore the evidence of your lying eyes. And hey, on the off chance that Will Duffy is watching, this is why you get people like myself saying that flat earth and creationism are basically the same thing. It's because over and over and over, creationists act just like flat earthers.
See, if a beneficial mutation were to occur,
which they observably do, go on,
in a population, and it's going to become fixed in that population, then it has to increase in numbers, obviously.
I mean, yeah, but the way we normally talk about that is that it has to increase the proportion of organisms in the population who have it. That's because we're talking about fixation. That is the process that results in all members of the population having the variation. So the raw number doesn't matter much and is dependent on the population size. So if a new allele is present in 10,000 members of a population, that doesn't tell if it's fixed. If the population is 10,000, then it is. But if the population is in the billions, like it is for humans, then it's not even close to being fixed.
For in the population for it to progress evolutionary in an evolutionary fashion, right?
Did Calvin Smith just have a mini stroke or something? That wasn't even English near as I can tell. This is like reading a tweet or truth or whatever the hell they're calling it from the King of America.
Relative to all of the other specimens in that population that don't have that beneficial mutation.
That's not how the word specimen is used. A specimen is an example of something that is currently being used for testing or investigation or observation, or has been collected for such. The word he's looking for is member, as in member of that population. Once again, it's not a huge deal, but it indicates that these two don't actually know what they're talking about when they misuse terms used in science.
That's right, because if one of the organisms gets the beneficial mutation, well, and that's a benefit, well, that's great for that organism, but that organism is going to die sooner or later. So unless that gene gets passed along and widely spread, it's not going to get fixed.
Evolution theory would suggest that beneficial mutation needs to wipe out, those individuals need to wipe out, or all the others need to somehow disappear.
That's right, over time.
And then evolution can...
No, they just reproduce less than those with the mutation. They can have perfectly normal long lives and not just disappear. In fact, they don't even have to have fewer children than they would have without the new mutation appearing. Creationists are just physically incapable of correctly or even reasonably describing evolution.
Yeah, but the problem with that, of course, is the rate at which that could happen is limited.
It is, just not enough to preclude evolution in the sense of universal common ancestry. And let's remember that the limited rate that these two are supporting here would also preclude post-flood speciation of arc kinds, which means that if this problem is real, it's as much a problem for creationists as it is for anyone else.
As a matter of fact, the biggest limiting factors, of course, is how quickly you can procreate, right? I mean, because there's two ways of doing it here. If you know, they actually talked about, you know, an imaginary scenario where, let's say you've got, you know, ape-like creatures, hominids or whatever. We're not fully human yet, and you know, let's say you've got like humans, you've got a birth rate of whatever, and it takes you, let's say, 20 years to, you know, to keep making new generations and stuff like that. So if you imagine a population of 100,000 apes, these supposed, you know, progenitors of the human race.
This guy sure likes to yap.
And you get a male and a female that somehow get this mutation that's beneficial.
You don't need to get a male and female both with the mutation. Just one will do fine.
Well, number one, in order for that to get passed on, then obviously they'd have to find each other in that group of 100,000.
No, they wouldn't. Does Calvin not know how sex works? Children inherit their genes from both parents, so your mom or your dad have a mutation, then there's a 25% chance you have it too, regardless of the fact that they don't both have it. And that's assuming it happens on one of their chromosomes and not the other, since, you know, that's how mutation works. You don't need to have two homozygous parents to get a mutation passed on. This is fucking high school Punnett Square shit. How the hell can he be this fucking dumb? This is something I'd expect from raw, eating is optional Matt Nyler, the alleged inventor of the kale chip.
People anyway, and pass it on. But you've got a couple of things you could do. If their offspring is going to become the mainstream with that mutation, 99,998 other ones, they've got to either disappear somewhere, or that particular mutant group has somehow got to get their next generation to somehow outcompete them and outpopulate them.
Yeah, that's it. They outpopulate them. And when that happens, and other parts of the population that lack the mutation mate, they'll become more likely to mate with those parts of the population that do have the mutation. And thereby, some of their offspring will have it too. And since those offspring are more likely to themselves reproduce, and reproduce more than their other offspring, the portion of the population with the new variation increases. Maybe Calvin Smith needs to go back and relearn like 9th grade biology.
An order of magnitude improvement over the remaining population.
No, it doesn't need to be an order of magnitude. It can be less than a single part in 100 improvement in spread. It just depends on population size, and the larger the population, the more likely it is for even small improvements in reproductive output to be selected for.
That they just procreate and all the others die out.
Nope, that's never been how it works, at least not in species that can exchange genetic material, which includes most bacteria, by the way.
Exactly. So I'm going to read this just so it'll make more sense to people.
Well, it couldn't make less sense.
Okay, so you've got an imaginary population. You've got 100,000 apes, and you get this male and female. They both receive a mutation so beneficial that they out-survived everybody else.
No, in addition to the dumb f***ery of thinking you need a male and female to both have the mutation spontaneously, and that they then have to mate with each other, which is something so dumb the average 15-year-old can tell you why it's wrong, they also don't have to out-survive anything. They just have to out-reproduce the rest of the population.
All the rest of the population died out, and then the surviving pair had enough offspring to replenish the population in one generation.
None of this bears any resemblance to anything in the real world.
Now, this is a supposed scenario, of course, but this is what would have to happen.
No, it's not.
If you repeated this imaginary situation every generation, every 20 years, for 10 million years, more, which of course is the supposed time since the last common ancestor, supposedly of apes and humans, that would mean that you'd have 500,000 beneficial mutations that could be added to the population. But even with this completely unrealistic scenario, which maximizes evolutionary progress,
no, it actually severely retards it because it prevents other beneficial mutations from arising and continuing to increase their frequency in the population at the same time as the one under consideration, which on its own is like half the solution to Haldane's dilemma. They need to stop treating it like you have to have a mutation fix before the next one can occur that will fix. You can have more than one mutation moving towards fixation at a time in real life, but not in this scenario.
Only about 0.02% of the human genome would be generated.
Well, since the common ancestor of humans and chimps didn't start with no genome, I don't see why this is even a thing Calvin would say, except that he's just accepting the obviously bullshit talking point of Don Batten, the known liar.
Oops. It's a totally unrealistic scenario, right?
Right, which is why it's kind of dumb to talk about.
And you keep having to, because the mutation that occurs, I mean, if you're going to have it accumulate, it keeps needing to occur in the same group. But if you've got a mutation here and a mutation here and a mutation here, somehow you've got to bring all those mutations together to keep stacking up this.
Yep, and that's where recombination comes in. This so-called problem is already solved. And Calvin Smith being unable to pass a high school biology class isn't a problem for science. It's evidence that he's either a colossal moron or he's a colossal liar. You tell me which.
So the dilemma is, even given the most favorable conditions for evolution, the results are nowhere near what they need to be.
Sure, if by the most favorable conditions you mean absurd conditions that severely retard the generation of new mutations and more or less prevents significant recombination, but most people would consider that to be among the least favorable conditions.
That's right, because that's only 0.02 of the human genome, and considering that the difference between the DNA of a human and a chimp, our supposed closest living relative, which that's another story we'll get into sometime. But anyway, even that is supposedly greater than 5%.
It depends on how you count it, and most of the differences are not the result of selection, they're the result of drift. Plus, about half of those differences would have occurred on the chimp side. Chimps aren't the common ancestor of chimps and humans. As a back-of-the-envelope way to estimate how much one lineage has changed genetically from its common ancestor with another lineage, just halve the amount of genetic difference between them, since all things being equal, they are both evolving and diverging from that ancestor. But also, in coding regions, the similarity is closer to 1%, meaning that the change from common ancestor in terms of the changes that are most likely to be beneficial is more like 0.5%. Of course, these are all very rough estimates, but then Calvin's estimates are based on just making shit up, so I'm okay with that.
Well, obviously 0.02 to 5%, that's a huge problem. So,
if it is, it's not for any of the reasons we've been given so far.
Haldane's dilemma basically has not been solved. That's what Don's article was about.
And that is incorrect, although I'd argue that to a large degree it was more about Don Batten's lust for Walter Remine. And hey, I say Don should just ask Walter to grab some coffee and see what happens. Maybe they can go retire together someplace nice, have lots of gay sex, and leave the pseudoscience to others.
There have been some evolutionists that have tried to, there wasn't much done. It was talked about a little bit in the 60s.
By there wasn't much done, what he means is that the dilemma was solved and the field moved on. But he just doesn't like that.
But then it was kind of largely ignored by most evolutionists until 1992, when an evolutionist, George C. Williams, remarked this: "The time has come for renewed discussion and experimental attack on Haldane's dilemma."
And then, since that wasn't really true and the solution to it was well known, that was mostly ignored. Just like if an astronomer today were to say that maybe we need to re-examine the precession of the orbit of Mercury since it doesn't seem to obey Newton's laws.
So basically, this is a problem that, let's not talk about it too much. All of a sudden, this evolutionist makes the statement here: we need to attack Haldane's dilemma. Why?
So they want to try and get through this dilemma for evolution.
That's right.
The why was probably that he incorrectly thought that the changes to the model that had been made that solved the dilemma were insufficient.
Yeah.
And so he wanted experimental attack. He wanted some observational proof that this wasn't true.
We already have that. We have both observational and experimental evidence that Haldane's dilemma isn't a thing in the real world. It was only ever a thing in human-made models.
And so, of course, we carry a book called The Biotic Message by Walter James Remine, and he did an extensive, restudy, you could say, on Haldane's dilemma.
Yeah, so I've heard.
And really pointed out what the problem was, because most of the criticisms that came against Haldane's dilemma, they were kind of criticizing, okay, well, you don't really need to get rid of all those other things. You just need to concentrate on, the creatures that get the mutation and stuff like that.
I have no idea what Calvin is talking about, probably because he has no idea what he's talking about. Probably because he doesn't know what Haldane's dilemma is, nor does he have even a basic understanding of genetics that would be expected of a school child.
But what Remine pointed out, and he had a great article, we have it online, you can check out too, it's called, just go to creation.com/cost.
Geez, more homework, and this won't be short either. That article is long. Oh well.
Cost Theory and the Cost of Substitution, a Clarification, by Walter J. Remine.
So the first thing to notice here is that this article was first published in the Journal of Creation, which is one of those pretend journals that young Earth creationists have. So while it seems like a peer-reviewed article, it absolutely is not. And it's no wonder this article didn't make a splash in the scientific world. It wasn't actually published there. Scientists aren't reading the Journal of Creation. They're reading real journals, with the exceptions of ones like Guts Gibbon who make busting young Earth creationism their business.
The cost of substitution has been widely misinterpreted, which has limited its utility. This paper clarifies the cost concept and re-establishes its vital role for investigating population phenomena. Many factors that traditionally caused confusion are identified and dismissed, including genetic death, genetic load, the environment, and extinction, which are not essential to the cost of substitution.
It's a bit weird that people seem to think that they are, and also that genetic death is literally how Haldane defined the term. When your argument starts with a redefinition of an already accepted term, chances are you're trying to make a straw man of the scientific claims you're fighting. But I can't say for sure that that is the case all the time. So let's let Remine cook.
Instead, the cost concept is here defined as the reproduction rate required by a scenario.
That's an incredibly weird way to define something called cost. If I told you the cost of gas for a trip was 30 cents a mile, that might make sense, but that's not normally how people think about it. Cost doesn't usually have a per unit of anything, but hey, I'm not in principle opposed to stipulated definitions, even weird unintuitive ones.
Unlike the traditional view, this cost concept is general and applies under the widest circumstances, including extreme fluctuation in population size and selective value, and for discrete or continuous generation models.
I am not convinced that the real version of the cost of selection does do this, at least as it has been updated by neutral theory and the recognition of the importance of crossover events during meiosis, but okay.
Yet, under the simplified circumstances traditionally assumed, this concept reduces exactly to the traditional formulas. For single substitutions, proofs show that the minimum total cost of substitution occurs when the cost remains constant throughout the substitution. A clarified basis for general-purpose cost theory is offered. Figure 1. A simple scenario showing the cost of substitution. 4 elephants are the initial new type. 1 generation later, 5 elephants are the new type. For that to be plausible, a reproduction rate of at least 1.25, equal to 5/4, is required in that generation.
That's true.
J.B.S. Haldane first described the cost of substitution and its limitations on the speed of evolution. That gave rise to a problem, see for example Dodson, known today as Haldane's dilemma. The problem is more severe in organisms with low reproduction rate and long generation time.
Let's be clear here. The theoretical problem is bigger for such organisms. There obviously is no real problem because such organisms exist, and in fact, beneficial alleles spread through them. One example is the tuskless trait in African elephants. It's beneficial because it prevents the elephant from being targeted by ivory poachers, which more than offsets the cost of no tusks, which is primarily making it harder to get food, since that's the main purpose of tusks on elephants. Now, this particular allele can't actually reach fixation in the population because it's sex-linked, and male embryos with the mutation die before being born, as do female embryos that are homozygous for it. But still, it has spread rather rapidly through the population, and in a population where the spread of beneficial alleles is supposed to be hardest according to Haldane's dilemma. So the dilemma is not something animals face in nature, it is something scientists face in theoretical modeling.
Such as higher vertebrates, elephants, whales, apes, and humans, etc.
All of which observably have beneficial traits spread through the population with no particular problem.
Evolutionary geneticists saw this as a compelling issue. Maynard Smith and Kimura each cited it as the main reason for their revolutionary new views of evolutionary processes. Yet the fundamental cost concept fell into long-lived confusion, which limited its deployment. Today, most commentators say the problem is solved, but exhibit little agreement as to why.
Fundamentally, it is because their models of population genetics result in realistic predictions that correlate well to observed real-world population genetics. That's what it means for a problem with modeling to be solved. If models don't produce realistic results, which is the case with Haldane's dilemma, then new models that do produce realistic results have solved the problem, even if the modelers don't know what differences have fixed the inaccuracy. The fact is that animals like whales, humans, and elephants manifestly do manage to pay the cost of selection, whatever it is. If your model says they can't, then your model is wrong. New alleles reach fixation, both in whole species as well as in subpopulations, and they don't have to wait for some unrealistic time frame to do it. So any hypothesis that says that they must wait unrealistic times for such events is wrong.
If it disagrees with experiment, it's wrong. And that simple statement is the key to science. It doesn't make a difference how beautiful your guess is. It doesn't make a difference how smart you are, who made the guess, or what his name is. If it disagrees with experiment, it's wrong. That's all there is to it.
One modern authority, George C. Williams, asserted the Haldane dilemma problem was never solved by Wallace or anyone else.
Good for him, I guess?
This paper will show the problem cannot be solved so long as confusion prevails over the fundamentals.
I mean, it is solved, but okay.
This paper clarifies the cost concept and reestablishes it as a vital tool for investigating population phenomena and our attempt to explain or describe them, here called scenarios. A cost, as here defined, is simply the reproduction rate required by a given scenario. If the given species cannot supply that reproduction rate, then the scenario is not plausible.
Under this modeling, but that's the thing. If your model says that something isn't plausible, yet it happens, then the problem is with your model.
In more concise wording, the species cannot pay the cost. At its core, a cost argument is that simple. A cost and its payment are both reproduction rates. They are defined identically, except that a cost is required by a given scenario, whereas a payment is actually produced within the given species. Historically, much confusion was generated from overemphasizing secondary matters, such as genetic death.
You mean the actual definition of cost that was defined by J.B.S. Haldane? Because let's remember, this is only related to Haldane's dilemma. Ramain is actually coming up with Ramain's dilemma, which is fine, but it's not the same thing.
The previous organisms that are eliminated, the environment, fitness, and genetics. So keep in mind, a cost is about the required reproduction rate, and all else is secondary. Indeed, the unit of cost is reproduction rate, i.e., offspring per individual per generation. Every cost is specified in terms of reproduction rate. This also means every cost is specified as a cost per generation, because per generation is the understood manner for specifying reproduction rate. As is customary, all reproduction rates are normalized by the number of producers. For example, if a male and a female produce 6 progeny, this is a reproduction rate of 3, not 6. This consistent focus on reproduction rate clarifies the cost concept. There are many types of cost, each named after what it models. The simplest cost, called the cost of continuity, CC, is the reproduction rate necessary to continue the reproducers from generation to generation. This cost is 1.0. That is, a reproduction rate of 1.0 is necessary solely to sustain their reproducers over the long term.
Except that in the real world it's higher because that assumes that all the offspring actually survive to themselves reproduce. That isn't the case, and in reality this cost is higher than 1.
All other costs require reproductive excess, a reproduction rate in excess of mere continuity. These other costs will tabulate in addition to the cost of continuity. Such costs include the cost of random loss, CR.
So I suppose that once we add all these costs together, we actually get the required reproduction rate for a species, and the 1.0 is just a theoretical thing. I suppose that's fair. Also, I've skimmed this before, and I think that later we're going to subtract out the cost of continuity, since it's always included and it makes the math easier. Even though there are actually circumstances where that's inappropriate, as we'll also get to.
The cost of eliminating harmful mutations, also known as the cost of mutation, CM, the cost of segregation, CX, and several others. This paper focuses on a specific cost, the cost of substitution, CS.
Okay, so this is the cost presumably to get an allele to fixation in a population within a given time frame under certain assumptions. Now, who wants to bet that the assumptions leave out things in the real world that would lower this cost?
In this paper, a trait is a heritable biological characteristic. The adjective old refers to the previously predominant trait that is being replaced by something new, also called the substituting trait. The new trait begins as a unique mutation and is substituted into the population. The old trait is eliminated. Individuals that have the old trait, or new trait, are called the old type, or new type. Evolution requires the substitution of traits into a population.
Technically, no. Any change in allele frequency is evolution, but it does happen and has happened, so our models of past evolution require it. But technically, evolution can and does occur without complete substitution.
The new trait goes from few in numbers to many in numbers. The new trait increases and grows. In this paper, increase and growth refer to the actual numbers of copies of the trait, not the frequency of the trait, and not growth of the trait in the embryo or phenotype.
Which is a bit weird because an allele reaches fixation when it is present in 100% of the population, not when it grows to some absolute number of individuals who carry it. It seems at this point that in order for this measure of cost to be useful, we would need detailed population estimates with high granularity for species, something we generally lack. Maybe I'm wrong though.
For example, a new trait grows from 1 copy to 1 million copies. This growth requires reproductive excess, a cost. The cost of substitution, CS, is the extra reproduction rate required to increase a specific genotype at the rate given by an evolutionary scenario.
A scenario which it still seems would require us to know the population at every step of the way, which seems unrealistic. I hope we get some explanation of such scenarios from the literature with this model applied to them so that I can see how it's supposed to actually work in real examples of population genetics.
This definition is devoid of genetic detail because the cost of substitution is not primarily about genetics. Rather, it is about the increase of a trait or traits via the increase of genotypes and the reproduction rate required for achieving it. The genetic details can be fleshed in when needed in specific case studies.
I'm not sure that does make sense though, because since more than one trait can be spreading through a population at a time, that seems like genetic detail that is important. Since, let's be honest, the whole point of this so-called paper is to say that the cost is too high and we can't reach the needed number of substitutions for, say, humans to evolve from a common ancestor with chimpanzees. Therefore, it didn't happen. But details like that mean that the rate of substitutions for a generation can be significantly in excess of the rate of a single substitution. In fact, it does not seem like there is a hard theoretical limit on it unless you limit population size to a particular maximum.
For simplicity, this paper deals only with increases achieved by excess reproduction rate. Increases or decreases due to migration and recurrent mutation are minor phenomena to be dealt with in another paper.
Yeah, I'm not sure that the founder effect, a major source of genetic differences between populations, and which can make the frequency of an allele go from very low to universal practically overnight, isn't important in figuring out the ancestry of species. I can't see calling that minor as anything but either gross incompetence or an attempt at deception.
Also, this paper focuses on a single substitution, non-overlapping in time. Multiple substitutions, overlapping in time, are an advanced topic to be covered in another paper. Until the basics are covered, we need to leave out genetic complexities like sexual reproduction or diploidy.
We wouldn't want to include the things that actually occur in large, slow reproducing organisms like the given examples of cetaceans, elephants, and apes. That might actually make evolution seem less implausible, and we can't have that now, can we?
As a simple example, assume that, averaged over the long term, the population has a uniform reproduction rate.
I mean, yeah, that's kind of what averages are for, so that seems fair.
Nonetheless, a new trait can substitute into the population. Our example scenario claims the new trait is increased by 25% in one generation, as in Figure 1. The species is therefore required to supply a reproduction rate of 1.25.
See, that's a really weird way to put it. When I hear that a trait has increased by 25%, I hear that the frequency of that trait in a population has increased by 25%. So that if it was present in 20% of a population, it is now present in 25% of the population. Because 5% is 25% of 20%. I'm not convinced that this odd way of talking about populations in absolute numbers, but still talking about fixation, which is about proportion, is intended to be confusing, but it is.
In other words, the cost of continuity, CC, of 1 plus a cost of substitution, CS, of 0.25. This example displays the cost of substitution at its simplest and most essential. If the given species cannot actually supply a reproduction rate of 1.25, which would require 2.5 births per female in species with a 1-to-1 male to female sex ratio, then the species cannot pay the cost. And the scenario is not plausible. That is how a cost argument operates. The cost amount cannot be reduced by further details concerning the environment, population size, the fate of old-type individuals, the manner of selection process, nor by fluctuations in any of those.
Yeah, I guess that's fair enough, but I've never seen people talk about such scenarios in terms that we could plug into this model. Like, pick a genetic locus in humans in which chimps, bonobos, and gorillas have the same state, but the human lineage has another. This would presumably be because of a mutation that occurred and then fixed in the human lineage since the split from the human common ancestor with chimps and bonobos, which is the same ancestor, and an ancestor that lived after that triad's common ancestor with gorillas. How do we go from that to an estimate about how many individuals would have had to be alive at a particular time in the past with that mutation? How do we get the variables to plug into this model? I don't know, and I'm not sure that Remind is going to tell us. Creating a new model is all well and good, but I am still at a loss as to how to take real-world data and check it against this model.
For example, environmental change and soft selection, no matter how those are defined, cannot reduce that cost. The cost is simple, mechanical, and unavoidable. When the scenario claimed an increase of 25% in one generation, at the moment the scenario made that simple claim, a cost of substitution of 0.25 was required.
But when are such claims ever made? It would be nice if there were a citation of such a claim. In fact, I would expect that a demand for such would be in a review of this paper if it had been actually peer-reviewed, which it has not. In that way, it's a bit like my Patreon, which has also not been peer-reviewed in any meaningful way, but is still vital to the channel. Patreon is one of the best ways to support the channel so that I don't have to ever stop making videos like this. So if you want me to be able to keep going, please consider checking the Patreon out with the link in the description. The Patreon starts at only $1 a month, and there's a 15% discount if you pledge annually. It gives you early access to videos like this, as well as access to my supporters Discord, which is one of the best ways to stay in touch with me personally. There are even more perks at higher tiers.
In the previous scenario, assigning the old trait a lower reproduction rate does not reduce the cost. Instead, it affects who must pay the cost.
I am trying to understand that sentence and having a really hard time. Traits don't have reproduction rates, organisms do. Is he saying that the trait gives the organisms that have it a different reproduction rate? If you know what he means by this, please put it in the comments. I am at a loss.
In the first scenario, the reproduction rate is uniform throughout the population, so that requires all genotypes to be produced at the reproduction rate of at least 1.25, and they all occur the same cost of substitution, equal to 0.25.
Wait, is that why we did an average and then spread it out over the population so that we can pretend that all parts of a population actually have the same reproduction rate? That's just not true. The whole point of natural selection and genetic drift is that not all segments of the population do have the same reproduction rate. So while in some cases assuming the average for all parts is fine, such as for population growth, for assessing evolutionary change, it's possibly the dumbest thing you can do.
In the second scenario, only the substituting genotype incurs that cost. The phrases genotype X incurs a cost C and the cost of genotype X is C are shorthand for the effective producers of genotype X progeny are required to produce genotype X progeny at a reproduction rate of C or greater, devoted to the task of identifying the type of cost.
So something I'm realizing here is that Remind, in focusing on absolute reproduction rate, is assuming a population that is not declining. That's why he's always willing to subtract the value of 1.0 from all of his costs, because he sees this as the cost that is always paid to maintain a population. But populations do not always go up. Suppose we have a population of sexually reproducing animals with separate male and female sexes, and each mating pair on average produces only 1.5 offspring that go on to reproduce. So the reproduction rate is 0.75. The species will be declining in population, a thing that happens in the life history of virtually all species. In this situation, a mutation that causes those carrying it to have a reproduction rate of 0.85 will still spread through the population, and could reach fixation, all while the reproduction rate of the animals in question is not just lower than Remind's alleged cost of selection, but lower than the replacement rate. This is because Remind's only contribution to this whole topic is just to misdefine the cost of selection to make it about absolute numbers and not proportionality, despite the fact that for a mutation to substitute for another allele, it has to fix in the population, or It has to reach 100% frequency across all genomes in the population.
For greatest physical understanding, focus on the genotype with the largest cost in a given generation, i.e., focus on the substituting genotype, because that sets the critical amount of the cost of substitution, and that genotype always incurs that cost.
Okay.
Moreover, that amount remains fixed regardless of whether the reproduction rate is uniform.
But it doesn't, because that depends on the unstated but obviously required assumption of constant or increasing population size. The fact that Haldane's dilemma was solved because we realized that the unspoken and often unrecognized assumptions of no genetic drift and no recombination were incorrect doesn't get reversed by adding a new false assumption. And of course, Romain is also bringing back the old false assumptions. If anything, he's making the argument that his version of the problem has even less to do with reality than Haldane's version, which already has not too much to do with it.
Call this the cost equivalence principle. Since this cost amount is unaffected by variation in individual reproduction rate, we can safely presume a uniform reproduction rate.
But what if it's below 1? That does happen. In fact, that's one of the biggest problems in conservation. If the reproduction rate of a species is low enough, then alleles that would normally be deleterious in larger populations can drift to fixation, all while the population dwindles. This is, as far as I can tell, something that Romain's model cannot account for, despite the fact that we know for a fact that it happens. In fact, fixation often happens faster while the population is declining, whether through actual decline in overall population or through the founder effect.
Haldane likewise uses that simplification in all of his tutorial example, though he did not explain it well. We can now ignore the reproducers of the old type individual and focus only on the reproducers of the substituting trait. These reproducers are required to supply a reproduction rate sufficient to cover all the costs of the scenario. These reproducers must supply replacement for themselves, which occurs cost CC.
But they don't. They only have to reproduce more than their conspecifics. If that happens, then their genotype will increase as a proportion of the population.
Plus supply for those individuals eliminated through random losses, which occurs cost CR.
Which again, they don't actually have to pay, because there's no requirement that the population be stable while an allele is moving to fixation.
Plus supply those individuals eliminated because they possess harmful mutation, which occurs cost CM.
Yeah, that is at least probably reasonably accurate if I try to turn Romain's nonsense terms into real terms that geneticists might use. If mutations occur that are so harmful that they cause death outright, then that is a hamper on reproductive output for any population in which such mutations occur. It even happens in humans. Numerous chromosomal abnormalities or mutations can cause a human to die long before reproduction, often before the mother is even aware that a fertilization has occurred.
Plus supply an increase of the substituting trait, which incurs cost CS, and so forth through the other costs. The reproducers must supply this full reproduction rate, otherwise the scenario is not plausible.
Yes, because populations only ever go up or stay the same. Except, oh, wait, that's not true at all.
In other words, the full requirement, the cost of evolution, sums all these costs. The costs all have the same units. They are all reproduction rate, so they add together conveniently. Equation 1. The cost of evolution equals CC plus CR plus CM plus CX plus CS plus any further. By focusing on the new trait, not the old trait as was traditionally done, one gets a clear physical understanding of the reproductive requirements and why they are mandatory.
No one does, for reasons that I've already gone over a lot. Let's just see if he actually has anything useful to say in the next few bits. I'll refrain from jumping in too much, or at least I'll try.
Let R be the actual, realized, reproduction rate for the new type progeny. R is called the payment, and is an observable feature of natural population. A scenario is implausible if the payment R is less than the cost of evolution. Equation 2. far less than cost of substitution. In other words, the scenario is implausible if the actual reproduction rate for new type progeny is less than the required reproduction rate for new type progeny. This plausibility criterion can be applied repeatedly to each generation in turn. It can also be applied over the long term by using long-term averages of costs and payments.
Except that it can't, because you won't capture the rapid fixation of various alleles during a bottleneck, with averages over the long term. And we know that populations suffer bottlenecks. Look at cheetahs. They have almost no genetic diversity. That is, nearly all loci in their genome are the same across individuals, meaning that a huge number of alleles have had to fix. Did this happen by cheetahs having lots of babies? No, it happened by them having almost none, to the point that they nearly went extinct. How does Walter account for cheetahs? They certainly couldn't pay the cost of selection according to his model, and yet here they are with all those fixed alleles. Literally, all you have to do to refute Riemann is just notice that cheetahs exist and are super inbred. And it's not just cheetahs. Any critically endangered species will have this problem. Cheetahs just happen to be a particularly charismatic example of it.
This framework will seem foreign to students schooled in the traditional view, which introduces confusion through a variety of other approaches. Amid the various approaches, this paper derives from the following narrow lineage. The cost concept originated with Haldane. Its math was improved by Crow, wherein he also explicitly brought in the term reproductive excess. Ney introduced a concept called accumulated fertility excess necessary for gene substitution, which he defined for a constant population size and small selection coefficient.
Assumptions which do not hold in the real world, especially the first one.
Morel best summarized the midterm comprehension. Various cost issues were clarified by Riemann.
Lied Riemann, the author of this paper, in an act of intellectual autofellatio that was deeply undeserved.
The present paper draws on those sources, further clarifies cost theory, and eliminates traditional sources of confusion, while progressively adding depth. The result is a clarified cost concept harmonious with Haldane's original intention and calculations.
Which, even if it were true, wouldn't be a good thing, since we know that the solution to Haldane's dilemma has to be that the dilemma isn't real in nature, because nature obviously doesn't act like Haldane would predict. So saying that your ideas are in harmony with his on this matter is just saying that your ideas are also wrong as evidenced by the reality of biological populations. Way to tell on yourself, Walter.
Calculating the cost of substitution. Let X be any heritable trait where the trait's cytogenetic identity is unique, indivisible, and consistent throughout the substitution. The trait is not multifactorial, where various mixes of different DNA appear to cause the same phenotypic outcome. Therefore, the trait can be identified generation by generation, and we may discuss its growth. Also, the trait is inherited, or not, as a complete unit, i.e., no partial inheritance of the trait. One may think of the trait as a block of DNA of some arbitrary length, or cytoplasmic character. We should not overfocus on the trait and its nature. The issue here is not the trait, but rather the growth of the trait and the reproduction rate necessary to achieve it. Let reproduction occur shortly after the start of each generation. In generation I, let P be the number of effective producers of type X progeny. Call this the effective starting count. As the cycle of that generation comes to a close, let P' be the ending count of type X adults who produce progeny in the succeeding generation. In other words, we are counting particular groups of reproducers at the generation's start and end. The first group is the sole producer of the second group, and that places a reproductive requirement on the first group. Note, throughout this paper, a prime, such as P', denotes a quantity as the generation comes to a close. The increase is delta P equals P' minus P. Then in generation I, the cost of substitution of type X is equation 3. Cost I equals delta P over P equals P prime over P minus 1. If greater than 0, otherwise cost I equals 0. In simple cases, such as asexual haploids, the effective starting count equals the previous generation's ending counts. In other words, P and P prime count the same group of parents one generation apart. Since the latter group is produced solely by the former group, the required reproduction rate is P prime divided by P. Subtract the cost of continuity, equal to 1, to obtain the cost of substitution. Unlike the traditional view, this cost concept does not assume a constant population size. Indeed, the population size may fluctuate wildly, and any cost associated with that will automatically be included within the above definition, with no special handling. This makes sense because population growth and substitution are virtually the same thing as far as the trait is concerned.
Wrong. Completely wrong, stupendously and almost incomprehensibly wrong and stupid. Let's do a little thought experiment. Let's say we have an island called Belisarius Island. On this island lives an endemic species of rat called the Friedish rat. The Friedish rat occurs in 3 color morphs: a sandy color or agouti, black or melanistic, and a piebald variety that occurs when agouti and melanistic rats mate. The island has no native predators that prey on the rats, and so their population is constrained primarily by food availability. As a result of this, we will assume that color is not selected for and both the agouti and the melanistic genes are present in 50% of the chromosomes that carry them, with the other 50% being the other allele. In this case, we expect 25% of the population to be agouti, 50% to be piebald, and 25% to be melanistic. Humans then settle Belisarius Island and introduce a new species, Felis sylvestris catus, the domesticated house cat. The cats go wild and start killing rats far faster than they can reproduce, causing the population to fall precipitously. But because the island is primarily sandy, the cats are worse at finding the agouti rats, whereas the piebald and melanistic rats stick out and are easy prey. In this circumstance, the agouti rats will quickly become the only rats left, all while their population declines overall, including the absolute number of agouti rats, since the cats still kill them faster than they can reproduce, just more slowly than they can kill the other rats. Suddenly, population growth and substitution are not at all the same thing for the poor Friedish rat. I struggle to imagine that such a scenario is beyond the comprehension of Walter Romein.
They both increase the trait.
But the trait can still substitute while declining in overall absolute population.
The total cost of substitution, total cost, merely sums the cost of substitution over an entire substitution cycle from beginning to end. Equation 4, total substitutions equals the sum of cost I. For given population size, the total cost often remains approximately constant over wide ranges of parameters. This consistency makes the total cost a useful figure for characterizing substitutions in a given population, and is generally used for that purpose. Equation 4 represents what the literature loosely calls the cost of substitution. But what is the physical meaning of reproduction rate summed over so many generations? It has no obvious physical interpretation and was a source of confusion.
I think the only person who is confused here is Walter. Also, I just learned that Walter is an engineer, not a scientist, because, of course, the Salem hypothesis strikes again.
Equation 4 is actually an intermediate step in finding CS, the substitutional cost per generation, which is the real focus of our concern. Therefore, total cost is calculated in equation 4, then averaged over all the substitution cycles of the scenario, and later divided by the average number of generations per substitution cycle to obtain the average cost per generation, which is used for CS. We could just as well obtain CS by averaging equation 3 over the long term. In the same way, equation 3 directly gives CS for any single generation we choose to analyze. In any case, CS is the quantity we seek, and equation 3 is what ultimately supplies knowledge of it by narrowing the term the cost of substitution to these quantities. Confusion can be reduced.
I don't think it can be, because this paper is extremely confused about extremely basic and obvious facts about nature. So if anything, it seems like we're increasing confusion here.
Cost averaging can produce simple, powerful arguments. For example, Haldane estimated the total cost of substitution is 30 on average. Therefore, if there is one substitution per 300 generations on average, then the cost of substitution, CS, is 30 divided by 300 equals 0.1 on average. Indeed, that was the kind of averaging argument that Haldane made. Figure 2. Cost versus time, showing how the average cost may not adequately represent the maximum cost. In cases like this, where the cost spikes high, the maximum cost must be used as the more stringent test of the scenario. In cases without such spiking, the average cost is often a more convenient test of the scenario.
And of course, we're going to ignore cases where the cost might drop sharply. In fact, it could go to 0, because it's possible for a trait to reach fixation without any reproduction at all. Go back to the example of the Belisarius Island Friedish rat. If that rat had a mating season and all the predation occurred between two of them, then an allele would have fixed without a single new member of the population being produced.
Equation 3 contains an if clause. It models the fact that decrements, decreases, of a substituting trait in a given generation have no physical means to reduce the total cost or the average cost.
Are you sure about that?
On the contrary, such decrements will increase total costs, and the if clause precisely incorporates that fact. Obviously, when the substituting trait decreases, it does not require extra reproduction rate, so there is zero cost, as defined herein. In this way, costs are inherently non-negative.
It actually can reduce the cost to 0 and fix an allele in place in less than a generation, as I've already explained. I understood this in my sophomore year of high school. I can see why this paper would be rejected by reviewers. It's not for the reasons that Walt's not-so-secret admirer, Don Batten, gave. It's because this is obviously written by someone who has a good understanding of math, but whose understanding of genetics rivals that of any 6-year-old.
Cost averaging is most useful over time intervals where the cost each generation, cost I, remains reasonably constant. Otherwise, the seriousness of cost problems can be underestimated or obscured. For example, take a scenario 1 million generations in length where cost I is 1 million for one generation and 0 thereafter. The average cost is 1, but that average cost figure conceals the extremely high cost in one generation. It obscures the fact that no species on Earth can pay enough in that one generation to make the scenario plausible. In other words, average cost is a helpful statistics so long as the maximum cost does not make the scenario implausible, see figure 2. Some scenarios may require partial or piecewise averaging to obtain different average cost values for different time periods. Very complex scenario may require abandoning the averaging technique altogether and instead require comparison of costs and payments generation by generation.
Yeah, that seems reasonable that cost can't be negative. That's not really a cost in that case. Except that, according to Walter's math, sometimes it is negative anyway.
Cost versus speed. A correct framing of the cost concept places unavoidable concrete limitations on scenarios. Indeed, cost theory is so unavoidable it even applies to computer simulations of evolution.
I really hope he gives us an available genetic simulation program and the parameters he used for this, but I suspect he won't, And if he doesn't, that's yet another reason to not publish this work. But how could we possibly know which to do without detailed population histories? See, that on its own makes this pretty useless. If we ignore the fact that substitution does not have to mean an absolute increase in the number of organisms carrying the allele that is moving to fixation, and just accept this moronic assumption, that still raises the problem that his plausibility test is dependent on knowing the population with some high degree of accuracy for a lineage across hundreds or even millions of generations, but we have no such knowledge. We can't even get that knowledge strictly from genetics because we can only ever cover effective population size, which is basically how big the population seems from the standpoint of population genetics, which can be larger or smaller than census population size. That is the number of individuals that physically exist in the population at any given time. So even if we accept that Ramine is right about this, it doesn't matter, because we functionally cannot test any scenario for plausibility outside the most extreme cases like minuscule or gigantic populations or substitution rates that are so high they are already implausible for other reasons. Ramine is doing nothing to advance knowledge here. He is just playing with math in ways that don't make any sense, and that even if they did, don't help us learn anything new about any existing or extinct population. That's another reason this work should be rejected by a peer reviewer.
As a simple example, set aside all the genetic complexities, diploidy, etc., and examine a simple species possessing the one essential attribute, reproduction. Suppose a single substitution requires a new trait to start in one adult and increase to 1 million adults.
Not how fixation works, but okay.
Say a given scenario allows only one generation. This would require a reproduction rate of at least 1 million progeny per adult. That equals a cost of substitution CC of 1 plus an extra reproduction rate of 999,999, which is the cost of substitution CS. It is impossible to get a lower cost for this scenario. Because of the high cost, this scenario would be impossible for all species.
Wow, so now we know that Walter Ramine can make up impossible scenarios. I don't know why we needed to have an example of that, but there it is. It's impossible for a species to be reduced to one organism that produces a generation of 1 million babies. I don't care. I don't see why Walter cares. One thing I do care about, though, is my channel members. They help me keep making content at a breakneck pace for the channel by making it a viable means to support myself. If you want to help with that, then check out the join button below the video. The memberships start at just $1.99 and they give you early access to videos like this one. They give you access to my super secret Discord server just for supporters, and at higher tiers, there are even more perks.
The only thing that can pay the cost is excess reproduction rate. The beneficialness of the trait cannot pay the cost. The trait cannot pay for itself. Only genuine reproductive excess can pay the cost of substitution.
Wrong. There doesn't even need to be reproduction for the substitution to occur.
The cost can be reduced by lowering the substitution rate. Suppose the substitution takes 3 generations. I arbitrarily assign numbers 500 and 20,000 for the intermediate generations and show the cost in Table 1.
This is basically torture right now. I think this paper has to be a violation of the Geneva Convention or the Declaration of Universal Human Rights or something. Can we report Walter to The Hague? Can we get Interpol on this?
The time in generations is 0, 1, 2, and 3. Number of individuals with trait is 1, 500, 20,000, and 1 million. The cost per generation is non-existent, 499, 39, and 49, with a total cost of substitution of 587. Table 1. A 3-generation substitution in a population of 1 million. Suppose the scenario claims the substitution happens exactly as shown in Table 1. This would require the species to pay costs of 499, 39, and 49 in the first, second, and third generations, respectively.
For many species, that actually is very easy to pay. Many invertebrates lay thousands of eggs per female. In fact, virtually all species reproduce at far below the maximum rate at which they could reproduce, provided sufficient resources. That on its own makes the cost problem Romine is going on about far less of a problem at all. In fact, even humans reproduce far more often than you'd think. You might think that looking at the birth rate would tell you about how often humans reproduce, except that, for the purposes of the cost of selection, every fertilization is a reproductive event. And we know that many fertilized zygotes do not make it to term. In fact, many of them die before even implanting on the uterine wall. So a female may have had only 2 children who were born, but had 10 or more reproductive events that did not result in a birth or even a pregnancy.
If the species cannot plausibly come up with those payments in precisely those generations, then the scenario is not plausible. In this way, the argument applies tightly, even on a generation-by-generation basis.
Yes, some made-up scenario applies to some made-up scenario. But do we have any real-world examples? Like, I could come up with scenarios that would make it impossible to land on the moon under my constraints I made up. That doesn't change that the Apollo missions happened.
My arbitrarily chosen numbers happen to give a total cost of 587. Yet no matter how you adjust the figures for the intermediate generations, the total cost will always be at least 297. It is impossible to get lower. An excess reproduction rate of 99 for each of 3 generations is just barely sufficient to satisfy the scenario. Other arrangements always total more than 297, but never less.
I'll take Walt's word for it, because I'm waiting for this to become important to, like, anything at all. So far, we're just in the creationist making things up part of the article, which isn't anything new for creationists, nor is it particularly interesting or compelling.
Thus, even if a scenario does not specify the intermediate generations, we can still set a lower bound on the total cost. So our framework still has force even when scenarios are not specific.
Only if there are good population estimates for the whole time that the scenario is supposed to occur. Like, we know where he's going with this, or where he hopes his readers will go. And it's that the number of alleles that would need to fix to go from the common ancestor of humans with chimpanzees is too high and would require reproductive output for humans that's unrealistic. But how does he know that? He doesn't, because we don't have good census population size data for hominins during that time, and we also know that at least Homo sapiens has undergone a bottleneck, an occurrence that could have led to a fixation of alleles while the total number of organisms with those alleles could even decline, something he assumes does not happen. So not only do we not have the data that we would need to use his model on human evolution, but the data we do have break his model anyway.
For a substitution of a given duration, a simple proof shows that the lowest total cost is achieved when the cost per generation remains constant. See Appendix. Table 2 summarizes this point. Table 2 is given by the following formula. Let PS and PE be the number of copies of the trait at the start, S, and end, E, of the substitution cycle. Define the substitution growth factor, K, equal to PE divided by PS. Let N be the number of generations for the substitution. The minimum total cost is given by equation 5. Total cost of substitution equals N times, open brackets, the nth root of K minus 1. Close brackets.
Hey, let's try it with our Friedish rats from Belisarius Island. Let's assume a starting population of 10,000 rats. Since it's a small island, that means that 7.5 thousand rats had the allele at the start before the cats. In one generation, all the other rats died, as did the heterozygotes. So we only have one generation, making this easy, and at the end, only 2.5 thousand rats have the allele, and it has fixed in the population. So we have the total cost of substitution equals 1 times the first root of 2.5 thousand divided by 7.5 thousand minus 1. Well, looks like the total cost of substitution is negative 2/3. So only about 1 in 3 rats would have to reproduce at all. Weird how this model can result in negative costs of selection, but Walter hasn't noticed that, or perhaps has just precluded it arbitrarily. It's almost like he doesn't know the first thing about population genetics, and I say that as someone who doesn't know much more than the first thing, but who has the sense to actually ask real geneticists, including one that specializes in animal population genetics.
These optionally low costs require the trait to have a constant growth rate throughout all generations. Since nature does not provide this constancy, real cases will always have higher costs.
Let's test that with a quick example of 4 generations. So in the first generation, we start with 500 copies of the substituting trait in a population of 2,000, giving us a frequency of 25% and a per generation cumulative cost of 0, since this is the first generation. In the second generation, population has stayed constant at 2,000, but the number of copies has increased to 1,000, meaning that now 50% of the population has the substituting trait. This is a cost of 1 for that generation according to Walter. In the next generation, the trait goes to 900 individuals, but since the population has declined to 1,500 individuals, the frequency has gone up to 60%. This means that our cost for this generation is negative 0.1, our cumulative cost is 0.9, and our average cost, according to Walt, is 1.8. Uh-oh. It already broke, didn't it? Well, let's keep going. In the fourth generation, the number of copies has gone up to 1,000, and the total population has gone down to 1,000, meaning that we have fixed the allele. The cost for this one generation is 0.1 repeating, or rounding it, 0.11. This means that the cumulative cost is 1.1. Our total average is 4. Oh, no, that can't be right. The cumulative total can't be lower than the average unless this math doesn't work. The average cost is 4, but that's higher than at any generation. If you calculate the average not with his formula, but just by using the cumulative total and dividing by 3, getting the arithmetic mean, you get 0.37, which is actually not too unreasonable an estimate, since it's right in the middle of our 3 data points. It's almost like it's trivially easy to show why this is nonsense.
Also, if the new trait decreases even momentarily, then the total cost increases, because some costs will be incurred more than once.
Weird, because when I had the trait decrease, it didn't do that in reality, only in Walter's math. In reality, the actual cost went down, even while it went up as a proportion of the population. I'm not even a peer, and I would say this does not deserve to be published.
Some theorists believe cost problems can be solved by a non-constant growth rate, such as frequency or density-dependent fitness, as employed in soft selection. But that does not reduce the problem, at least not for single substitutions, as shown above. Rather, constancy is required to minimize the problem, as it allows the lowest possible total cost for a substitution of any given duration.
It sure doesn't seem that way when I run some numbers in a scenario that Walt seems to have forgotten is possible.
The total cost is extremely high for fast substitutions and decreases for slower substitutions.
Except, if the situation is fast enough and the population is actually declining, the cost can go negative. Like really, really negative in some cases.
The absolute lowest total cost occurs under conditions never met in nature. It only occurs when the trait increases monotonically at a constant growth rate, and when that rate is infinitely slow. In that case, equation 4 becomes integral from PS to PE of dP over P equals log base E of PE over PS equals log base E of K. Note, when n goes to infinity, equation 5 simplifies exactly to equation 6 as expected. A table, on one side, substitution duration in generations, on the other side, minimal cost. 1 for 999.0. 2 for 1998.0. 3 for 297.0. 4 for 122.4911. 5 for 74.2447. 6 for 45.0.
It's 54, not 45, but hey, that's okay. Vergeware is so far doing a great job, so if you appreciate all the hard work he's done to make this happen, please let him know in the comments.
6 for 54.0.
Oh, sorry, he did fix it, and it was just my dumbass who didn't notice. Sorry.
7 for 43.3780. 8 for 36.9873. 9 for 32.7743. 10 for 28.
29, but who cares?
0.8107. 30 for 17.5468. 100 for 14.8154. 300 for 14.1386. And 1000 for 13.9114. Infinity provides 13.8155. Minimum total cost for increasing a trait by a factor of 1 million. For a single trait to increase by a given substitution growth factor k, equation 6 gives the minimum total cost under ideal conditions, which are never met in nature.
I agree that ideal conditions are never met in nature. The problem is that Walter has misdiagnosed what is possible in nature, as well as what ideal conditions would be for the substitution time and cost to be at a minimum. So while the conditions are never ideal, they can be far, far better than what Walter thinks of as ideal. Also, sorry for all the dry table reading, but I don't want any of you who are listening or who are visually impaired to miss the tables. So those of you who are watching, thanks for continuing on and suffering in the name of accessibility.
This also has been the most commonly used equation for the total cost of substitution. Traditional sources of confusion. The cost of substitution depends foremost on the growth rate claimed in a scenario. It is not primarily about the environment, the old-type organisms that are replaced, or their life histories. These had previously been suggested as ways to reduce or eliminate the cost, but they cannot possibly reduce the cost lower than the minimums above. Their predominant effect is to raise the cost and/or lower the payments, and thereby aggravate cost problems.
Notice how he doesn't mention reducing population, recombination, or genetic drift. You know, things that actually do solve this problem.
Many commentators, e.g. Van Valen, Felsenstein, Hartle, Merrill, and Grant, indicate that according to the cost of substitution, if a population undergoes too many substitutions too rapidly, the cost will be too great, and the species will become extinct. That interpretation is faulty. The cost of substitution is not a theory of extinction, and cost, by itself, does not cause extinction. Rather, the cost of substitution supplies a criterion of plausibility, which compels us to reject some scenarios as implausible.
Except that I still remain pretty confident that we can't ever actually propose those scenarios with any confidence, so it's useless for work in deep time, even if the math wasn't broken, which it is.
Then, extinction may, or may not, result for various additional reasons. For example, if a scenario assumes a certain substitution rate is necessary to fend off extinction, then this assumption itself forges the link to extinction, not cost theory. The target of cost theory is the plausibility, or otherwise, of a given scenario. Some commentators, e.g. Bruce and Merrill, suggest the cost of not evolving is greater than the cost of evolving. That reckoning uses the word cost informally, as though not evolving leads to extinction, and extinction has a high cost. But that interpretation does not correspond to any population genetic definition of cost. Used correctly, the cost of not evolving, the cost of no substitutions, is 0. Other things being equal, the cost of adaptive evolution is always greater than the cost of not evolving.
Nope, because remember, the cost of substitution can in fact be negative with Walt's math, and the cost of not evolving would still be 0 with his math. So the cost of substitution can indeed be lower than the cost of not evolving. And this is just a hypothetical. Remember, I mentioned the tuskless trait in African elephants. That's increasing while the population of elephants is declining, which means it is a candidate for a trait that, while it won't fix, is moving towards fixation and could very well have a negative cost of substitution. And further, if some other trait that does not cause fatality in some female and all male embryos were to arise that also cause tusklessness in elephants, then it actually could fix, while the Walter version of cost is negative.
The cost of substitution was traditionally taught using the concept of genetic death. The idea was that a substitution requires the genetic death or elimination of the old type individuals. That created confusion by emphasizing the wrong thing, the death of the old type, their life history, how they die, and so forth. Fundamentally, the cost of substitution is not about that. It is about the increase, the excess birth of additional copies of the new type.
See, the problem is, though, that in all scenarios, the genetic death of old type genomes is something that must occur. For Walt's version, the number of substituting type doesn't actually have to go up. So no matter how confusing Walt finds the concept of genetic death, it's a definition for cost that actually always shows up consistently as a positive number and doesn't suddenly go crazy the moment the population declines, such that the number of copies of the substituting allele could decline while still going up in proportion. Because fundamentally, fixation, which is what we're concerned about, is not about increasing in total copies, but increasing in proportion. And the original definition of cost formulated by Haldane actually takes that into account, meaning that it is not actually Haldane and the rest of population genetics that is off topic. It is Walt.
The traditional approaches assumed a constant population size where, in each generation, the actual reduction of the old type precisely equals the excess births of the new type.
Now look, I don't want to say that Walter is lying, but I'm not sure what else to say. The older definitions didn't assume a constant population. They just concerned themselves about the relative ratios, not the actual population size. This means that they could seamlessly account for changes in population size, both increases and decreases, something that Walt can't seem to do. It's like he's looking at the per capita GDP of countries and then complaining that some of them are bigger or smaller in population, just ignoring that the statistic is already adjusted for population size. And he can know that because that's what per capita means.
So, the concept had some intuitive appeal, and, in the simplest cases, yielded a correct result. This paper dismisses the concept of genetic death, along with the requirement for constant population size.
In other words, this paper dismisses the thing that actually makes the math meaningful in all scenarios, as well as another thing that was never the case. This is a disaster of an article, and the longer it goes on, the more I can see why it was rejected. I'll be honest, when I first skimmed it, it seemed unconventional, but not horrible. But then I actually started paying real attention, and I sent it to both Dr. Zach Hancock and Dan Sturden-Cardinal, and they confirmed that my problems with it were in fact real problems with the paper. In other words, I sent it to 2 more peers to review it, and they agreed with the previous peers who reviewed this, that it's not worth any paper it might hypothetically be printed on.
The above discussion did not mention whether the substituting trait is beneficial, neutral, or harmful, because it does not matter.
Now there, I 100% agree. And also, it seems to be the first acknowledgement of any genetic drift that Walt has given. Any trait that goes to fixation acts from a population genetic standpoint about the same. It has to increase in proportion until it reaches 100%. That will result from it being likely to be reproduced more than other alleles at that locus on the genome by definition. And neutral variations fix all the time. And deleterious variations fix in situations where a population is small or declining. If we go back to our rat example, it might be that just by chance, the number of agouti rats, who are the only ones left after the dread attacks of the cats, just by chance had a high proportion of some variant digestive enzyme that made them unable to digest some food that before made up a significant part of the diet of the species. And with so few rats left overall, that might fix in the population just by chance, thereby eliminating that food source from the diet of the rats. That's the kind of thing that happens in small populations because natural selection is weaker in small populations and drift is stronger in small populations.
Whenever the trait increases through reproductive means, there is a cost. This contradicts almost all the traditional literature which holds that neutral substitution have no cost, e.g. Merrill. I clarify the discrepancy by noting that neutral substitutions indeed have a high cost, because they fluctuate up and down, repeatedly occurring additional costs.
I was just agreeing with Walter, and then he goes and screws it up again. As we've already seen, his math doesn't do what he thinks it does in all scenarios, and even if it did, it's not actually useful in practical terms because it requires that we have a reasonable estimate for census population size at each generation, which is completely impossible in most cases. Like, just try to figure out how many members of the population that evolved into modern humans were alive in the year 2,400,000 BCE. You can't. So what costs of substitution are too high to make that scenario plausible? I have no idea, and neither does Walt.
But their overall substitution rate is not cost-limited. Neutral and slightly harmful mutations individually substitute very slowly.
Only in large populations. In small populations, they can substitute at very high rates. I really think Walt needs to audit some pop-gen classes.
But they have special mechanisms that allow high overall rates of substitution, unlimited by cost, but instead limited by the mutation rate. 1. Higher payments. There are a few special mechanisms for supplying larger reproductive payments. These mechanisms employ randomness in the payment, a stochastic reproductive excess at the level of the individual, plus, in sexual species, also at the level of the gene. Note, in a sexual species, in each generation, each gene locus experiences a doubling at fertilization and a halving at meiosis. From the gene's eye view, the doubling is a source of reproductive access, though of a stochastic or random nature. The doubling and halving gives an average gene-level reproduction rate of 1.0, but randomly fluctuates above and below that to provide stochastic reproductive access. Crossing over is an additional source. These sources at gene level are sufficient to pay for substitution, even if there were no reproductive excess at the level of the individual.
Excuse me, but what the f did Walt just say? So Walt is fine with crossover helping with neutral variation, but not beneficial variation. I'm sorry, Walt, sweetie, but that's not how it works. Walt himself pointed out that the requirement of paying a cost is the same for any substitution, beneficial, harmful, or neutral. So if it helps with one, it can help with any of them. Walt just gave away the ballgame. Not only does his math not work, he also just admitted to omitting from it things that he knows would help mitigate any problems he proposes. I don't know if Walt is that incompetent or if he's lying. The fact that he thought he even could pass peer review makes me lean towards incompetent, but if you're watching and you got this far, let me know in the comments by commenting either Walt is a liar or Walt is incompetent. I suppose you could also comment Walt is both.
Virtually all of a species' reproduction is random with respect to a given neutral substitution, and the stochastic component of all that reproduction can be large. The stochastic reproductive access is what pays for genetic drift and the substitution of neutral and harmful traits. These mechanisms rely on randomness and are overwhelmingly non-beneficial or harmful in outcome. With these mechanisms, beneficial evolution becomes a moot point.
Hey, fun fact. Random processes can also help beneficial traits, so this can also lower the time to substitution for beneficial traits. Of course, it can also eliminate such traits, but in that case, we won't see such traits in the genetics of the subsequent population, so they are invisible to us. The genome only shows those traits that still persist. So, of the beneficial alleles that fix, most, if not all of them, will have been given a boost by some degree of stochastic reproduction.
2. Lower costs. There are also special mechanisms for reducing the average cost of substitution when many mutations occur near each other in time, linked near each other on a chromosome, then they substitute together. In this way, many are substituted for the cost of one substitution. However, to have much effect, this mechanism requires a superabundance of the new mutation to be substituted.
Well, I hope we get a definition for what counts as a superabundance, Because it seems to me like linkage blocks always allow for more than one mutation to fix together with the same set of reproductive events, regardless of degree of superness of their abundance.
For example, with unlimited high rates of neutral or harmful mutation, unlimited high rates of these substitutions are achievable, unlimited by cost. Those same mechanisms, however, are not available to beneficial substitutions.
How convenient. It would be nice if that were explained, though, since multiple beneficial mutations can end up on the same chromosome, either by mutating on the same chromosome at the same or at a different time, or as a result of crossover. What in the world prevents them from benefiting from occurring in the same linkage block? I can't think of anything. If you can, let me know in the comments. I sure hope Walter thinks of a reason beyond his bald assertion that, near as I can tell, was made simply because it would be inconvenient for him if it were otherwise.
Long before those mechanisms can aid beneficial substitutions, the population will be overwhelmed and destroyed by harmful mutation.
Well, that's a claim that really could use a citation attached, because that just sounds like genetic entropy, which is even less of a real thing than Walt's bullshit is.
For example, the desired beneficial mutations would far more often be linked to harmful mutations, so these would travel together, usually to elimination, with no net benefit to the species.
And sometimes they won't. And when they aren't, then they'll benefit from the boost associated with being in the same linkage block as other beneficial mutations. So what prevents that from happening? Something must, otherwise it's inevitable that such occurrences will boost the substitution rate for multiple beneficial alleles. That something else happens more often doesn't change that this does happen.
In this case, the cost of eliminating harmful mutations, CM, would be high, thereby aggravating cost problems.
But the number of individuals with the crossovers that make them extra bad at reproducing is always going to be small as a proportion of the population, and even in overall terms for any reasonably sized population, meaning that the cost of eliminating them is usually going to be quite small, whether we accept Walt's nonsense or we go with the measures that real geneticists use.
Lastly, the population would likely be an error catastrophe, where harmful mutations would chronically accumulate faster than they could be eliminated.
Boy, that escalated quickly.
How would that be likely? Crossover events don't accelerate during error catastrophe. In fact, in absolute numbers, they would go down, because error catastrophe is when a population is declining so fast that natural selection can't prevent harmful mutations from fixing in the population. But Walt hasn't shown any ability to even comprehend that populations go down. And in this case, his version of cost would be negative for the harmful mutations, something he seems to not even be aware is possible. I think he might actually be conflating error catastrophe, which results from declining population and genetic diversity, with genetic entropy, which isn't real, but if it were, would be about increasing genetic diversity, basically the opposite of error catastrophe.
All this happens because harmful mutations vastly outnumber beneficial mutations in quantity and effect, long before there is the necessary superabundance of beneficial mutations,
a necessity which has not really been justified as of yet.
The scenario would suffer severely from high rates of harmful mutation. In such a situation, beneficial evolution becomes a moot point. Evolution must operate within relatively low rates of beneficial mutation, and this precludes this mechanism for reducing the average cost.
None of that is true. In fact, the rate of beneficial mutations is dependent not just on the mutation rate of the population, but also on the environment. It's time to reintroduce the fitness landscape. This is a mathematical construct that represents different phenotypes as locations on a plane, and a third vertical axis represents the fitness of those phenotypes. Higher locations are more fit and lower ones are less fit. Importantly, the shape of the landscape is determined by the environment. Each change in the environment shifts the landscape in some way. So let's suppose that a population finds itself in a valley such that the bottom of the valley also slopes down. In this case, most mutations will actually be beneficial if they change the phenotype at all, and only a minority will be harmful. And in short order, the population will climb out of the valley. Only when a population reaches or comes near to a local fitness peak, such that all or most significant phenotypic change is negative, do deleterious mutations outnumber beneficial ones. Now, that is the case for most populations most of the time because of natural selection. So that's why we can fairly say that harmful mutations outnumber beneficial ones. But it need not be the case for any given population all of the time. Once again, Walt is showing that he's not aware of some of the most basic concepts in evolutionary biology or population genetics.
In short, while all substitutions have a cost, this fact only limits the beneficial substitution rate. Examples from nature showing rapid rates of neutral or harmful substitution do not contradict cost theory or the cost of substitution, nor do they explain the biological design that evolution is called on to explain.
Imagine calling your nonsense ideas that you can't get published a theory in the very paper that you can't get published. That's a level of arrogance that is worthy of Kent Hovind, Raw Matt, or RFK Jr.
Some commentators, e.g. Felsenstein, have claimed the substitution of favorable mutants in the absence of environmental change does not impose any cost. That is mistaken. The cost of substitution embodies the unyielding fact that growth requires reproductive access.
But only in relative terms. The thing that Walt can't seem to understand. Now, I'm not going to go read Felsenstein's work to see how right or wrong it is. Because again, this series is already way longer than I ever expected it to be. But yeah, Walt isn't even thinking about the same thing as an actual evolutionary biologist. He's fundamentally wrong about what fixation even is.
The equations given in this paper represent optimal situations, independent of the environment. On average, environmental change can only increase costs and/or decrease payments, thereby intensifying cost problems.
Well, we've already disproven that by contradiction, so that's entirely wrong. We've also shown theoretically why he's also wrong about mutations in the first place. What is Walt right about? I can't really think of anything that's not trivial, like that mutations occur, or that substitution requires all members of a population to have the substituting allele.
Substitutions when population size fluctuates. Some investigators have argued as follows. Suppose the population comprises 1 million old-type individuals and 1 new-type individual. If the 1 million old-type individuals die without heirs, then fixation occurs extremely rapidly, in 1 generation. Thus, elimination of the old type, which they equated to the cost of substitution, places no limitation on the substitution rate.
I mean, yeah, that would hypothetically make it unbounded, although in reality it would actually severely limit the substitution rate in the long run by eliminating most of the alleles that could move towards fixation in any generation where this occurs.
However, that scenario is incomplete, since it does not represent a complete cycle of substitution.
It absolutely does, because populations can decline.
It does not represent how evolution happens over the long term.
That's true.
Also, how am I still not even halfway done with this?
This scenario reduced the population size down to 1 individual, where countless generations would pass before the next beneficial mutation would occur, allowing the cycle of substitution to begin anew.
Nope, that's contingent, not deterministic. Remember, there can be a situation in which mutations are more likely to be beneficial than deleterious. It's not common, but it can occur. But also, since this is an unrealistic scenario, I don't know why we're back at it. I've already talked about how stupid this is more than once. Frankly, I don't even think there's a good reason for actual people who know what they're talking about to talk about this. Never mind bozos like Walt and Calvin Smith.
For example, a population of 1 will receive beneficial mutations 1 million times slower than a population of 1 million.
Ceteris paribus, sure. But what if ceteris non paribus, eh? And yes, you should brush up on your Latin. I should too.
This mode of evolution would be exceedingly slow. To speed things up, the scenario can claim the population grows, perhaps returning to its previous size, but that requires reproductive access, a cost of substitution as defined above. A speed limit occurs because the population growth rate is limited to the species' available access reproduction rate. Once again, the focus on elimination of the old type created confusion by following the wrong thing. The issue is not elimination of the old type, but rather the growth of the new type.
Well, I won't repeat myself about Walt's misunderstandings, but I will point out that Calvin Smith used this scenario as his idea of the optimal scenario for fixation rate. which isn't what Remind thinks it is. So Calvin Smith is so stupid he can't even understand the point of Walter's article. That's a special kind of stupid, isn't it?
This paper takes a broader view of substitutions. Under the traditional view, a substitution is defined by changes in allele frequency, and a substitution ends at the moment of fixation, thereby excluding any periods where allele frequencies remain constant and ignoring population growth as irrelevant.
Yeah, because once an allele fixes, it has substituted for all previous versions of that locus or all other alleles. That's what substitution is. Fundamentally, that's what it is. And complaining that you want to change that is giving away the game that you can't actually win the argument, so you want to have a different argument, but pretend you're still talking about the same thing.
That view is inadequate for studying substitution rays over the long term.
No, it's inconvenient for Walt. And also, this reason he's given is ironic given that his model is largely impossible to use to model anything in the long term because we're missing the key parameters of population counts for basically every population over that long term.
For this purpose, a substitution cycle can be usefully defined as an interval of time beginning with the introduction of a mutation that reaches fixation, then continues after fixation until the introduction of the next mutation to reach fixation.
Well, that time is usually going to be negative because usually the next mutation to fix in a population is already spreading before the first one fixes. So that means that most of the time, by Walt's definition, the time for a substitution cycle to occur is actually less than the time to fixation for any given mutation that actually reaches fixation. That's a weird way to define things.
This interval will be at least as long, and usually longer, one entire substitution, and may involve substitutions overlapping in time.
Nope, if the substitution processes overlap, which is typical, then the substitution cycle will be smaller, and often much smaller than the substitution time as Walt has defined it. Like, all of this is literally the opposite amount of time. Like, let's say it takes 300 generations to go from mutation A occurring to mutation B fixing in the population. That means that the substitution time is 300 generations. But let's say that another mutation occurs, mutation B, and it will also reach fixation in 300 generations, but it occurs 50 generations after mutation A occurs, meaning that mutation A still has 250 generations to go. According to Walt, the substitution rate would be 1 per 300 generations, while the substitution cycle duration is only 1 every 50 generations. I have trouble imagining that he can't see that, but then he has no training in population genetics or evolutionary biology that I can find, so maybe he really is just that clueless.
Under this definition, there is a contiguous set of substitution cycles, with no overlap or omissions in time.
How the f can he even pretend that that's the case? What if the mutation that will fix after the next to fix occurs before the next to fix actually fixes? That's overlap. That's what that is. My brain is about to break.
The goal is always to tally the reproductive requirements of the scenario exactly once, with no time periods omitted or counted twice.
No, the goal is to find a reason to believe what Walt was always going to believe in order to soothe the cognitive dissonance that comes from knowing that what he believes is precluded by all the available data. That's the actual goal. This is an exercise in apologetics from first to last. Walt doesn't give two fucks about anything else and will ignore, twist, or invent whatever data and math he needs to avoid that feeling of believing something that he knows can't be true. What other possible reason could he have for even diving into this topic? No one in the field cares. Everyone who became curious about this topic and actually learned about it without a dedication to being wrong as a priority has come to the same conclusion. Haldane's dilemma is solved. It takes a perversity of the kind common to pseudoscientists to think that not only is that not the case, but that someone who doesn't know what they're talking about in the first case is going to overturn that consensus by not even understanding the basics of the field, and that they could do it by doing some math that doesn't work on its own terms.
The above definition allows calculations of the total cost even when population size fluctuates, which the traditional cost concept did not allow.
All right, guys. Sometimes you are wrong in life.
In fact, fluctuating populations kind of break Walt's math.
Such fluctuating scenarios are diverse and awkward to generalize. Their details are an advanced topic outside this paper. The assumption of constant population size is here taken solely for simplification, so further issues can be clarified.
Oh, well, that's convenient, isn't it? Since we've already shown that actually they're not just a complication, they break the whole model.
Haploids, clonal or self-fertilizing organisms, or maternally inherited cytoplasmic characters.
This ought to be good.
For the sake of clarity, the cost concept, defined above, was stated in its simplest non-genetic form. When applied to specific case studies, the definition can take on the traditional terminology of population genetics, where the substituting trait could be a point mutation, insertion, deletion, allele, inversion, duplication, the relative order of genes on a chromosome, or similar.
Since most of those things are things that produce alleles, and alleles themselves are not mutations, there's more evidence that Walt doesn't know the first thing about genetics. Well, okay, he knows the first thing, but not the second or third things.
To simplify discussions, the traditional term allele will be used as an example for all of these.
That's one of the more reasonable things in the entire article.
In a haploid, during a single substitution, let there be P individuals with an allele A. And one generation later, a scenario claims it increases by delta P. The cost is given directly by equation 3. Notice this does not depend on the old type allele, its characteristics, how it dies, or the environment. The cost simply depends on the growth of the substitution allele. Let NE be the effective breeding population size. It is typically the number of adults who breed. Its precise definition is not critical to our calculations because its impact is solely to measure the population growth or decay. It drops out and has no effect on calculations when it remains constant. Let the population growth factor G equal NE prime divided by NE. When NE remains constant, G equals 1. The concept of effective producer of progeny allows generalization of our notion about reproduction. It allows the conversion of sexual reproducers into reproductively equivalent asexual reproducers.
No, it doesn't. Not if you want to generalize this to diploid organisms, because hidden in there is the assumption that crossover events don't happen. But they do in diploids, and that's a big part of the solution to Haldane's dilemma. The dilemma is supposed to be worst for big, slowly reproducing things like apes, whales, and elephants, but those are groups that are all diploid, and cannot be effectively modeled as a population of haploid asexual reproducers, specifically because of things like recombination.
To see how this works, adopt the usual convention of population genetics. Let the new and old alleles be big A and little a, with parental frequencies little p and little q, where little p plus little q equals 1. Multiply that equation by NE to obtain NE times little p plus NE times little q equals NE. The terms of that equation identify the effective producers of the two genotypes. In effect, there are NE times little p producers of the new type progeny and NE times little q producers of the old type progeny. It is as though all the reproduction of NE times little p adults goes to producing genotype big A progeny, and all the reproduction of NE times little q adults goes to producing genotype little a progeny.
Which isn't what happens in sexually reproducing diploid species. Let's suppose we have an allele which is a recently mutated beneficial allele, and it happens to be dominant. Now let's suppose that an organism which is heterozygous for this allele and thus expresses the trait associated with it, is in a population where the prevalence of this allele is still very low. Most of its potential mates will not have the allele at all. So if it mates with a conspecific that does not have the allele, then some 50% of its offspring will lack this allele. But on the other hand, its mate, which is of the so-called old type, will have half of its offspring also having the new type. This is reasonably captured by the model, but suppose the allele spreads such that a heterozygote has a reasonable chance of mating with another heterozygote. Now they are producing offspring of the new type 75% of the time. This is not well captured by this model. Then suppose an organism which is homozygous for the new allele mates. All of its offspring will be of the new type, as will all of those offspring produced by its mate, even if that mate lacks the new allele entirely. It has converted reproducers who are of the old type into producers of the new type. This is also not well captured by this model.
Those figures are exact for asexual species. For sexual species, those figures are average value.
Look, I'm not a population geneticist, and neither is Romine. So maybe they are averages that would apply to a species like humans, but that's not clear at all, and that statement needs to be justified.
In any case, we have subdivided the population's reproductive capacity into portions responsible for producing each genotype.
Cool, it's just that that's not how reproduction works in sexually reproducing diploid species. Genomes aren't replicated as a whole. There's crossover and new alleles interacting with alleles from the other parent. All of this is significant. If sexual reproduction were functionally equivalent to asexual reproduction, except that it required the organism to find a mate, then sexual reproduction would be unlikely to even occur. The gain in evolvability is itself the driving force behind sexual reproduction.
A genotype's effective starting count is the number of effective producers of that genotype. For the substituting allele, that figure is big P equals NE times little p. By definition, the ending count is P prime equals NE prime times little p prime.
For those of you watching, I want to apologize for the horrible picture quality on the equations. That's how AIG published them, and frankly, I'm not going to take time to just adjust them and type them up for them. Just know that AIG apparently doesn't care enough about this so-called paper to make it presentable when they publish it online.
Then restate equation 3 as equation 7. Cost I equals NE prime times little p prime over NE times little p minus 1 equals G times p prime over p minus 1 if greater than 0, otherwise cost I equals 0. Equation 7 is valid under wide conditions, such as erratic fluctuation in both the population size and growth weight of genotype big A.
You know, given how badly it ruins his math as we've seen, I'm surprised that Walter even acknowledged that populations can decrease in size. So I guess he just didn't bother to actually test his previous equations with that scenario. Weird.
Next, assume a constant population size, G equals 1, and utilize the fact that genotype growth is usually specified in terms of selective values. Let genotype big A and little a have selective values 1 and 1 minus s, respectively, where s is greater than 0. We now have a table showing genotype big A compared to little a. The selective value for big A is 1. Selective value for little a is 1 minus s. We then get equation 8 for the frequency. Little p plus little q equals 1. We then get equation 9. Apply selection. Little p plus 1 minus s times little q equals 1 minus s times little q. And then equation 10, normalizing it. Little p over 1 minus s times little q plus 1 minus s times little q over 1 minus s times little q equals 1. The left-hand term is the new allele frequency, little p prime, which by equation 7 gives equation 11. Cost I equals s times little q divided by 1 minus s times little q. Equation 11 gives the minimum required excess reproduction rate. It is accurate regardless of the environment, changing or unchanging, the selection coefficient, small or large, or the type of selection, soft or hard. For example, in a population of 1 million adults, where 1 has the new allele, big A, therefore little p equals 10 to the minus 6, Suppose S equals 1, which means that the substitution occurs in one generation, then cost I equals 999,999. That reconfirms the first scenario in the above selection, cost versus speed.
Hey, we did it! We're at the halfway point of this article, and I want to note that while he says this works in all scenarios, he also said that the cost of selection cannot be negative, but again, it sure can be. Let's suppose that we have a population of 1 million, but we assume that 750,000 of the population have the substituting allele. Then we'll assume that half the population dies, and that of those, all those who lack the substituting allele will die. Suddenly, the cost of substitution is apparently minus 150,000 for the substitution to occur in one generation, according to this equation. I think it would be quite easy to pay that cost from the standpoint of population genetics.
Genetic death. Equation 11 is identical to Krauss' cost formula and, under Haldane's additional assumption of S much smaller than 1, it simplifies exactly to Haldane's formula. This is remarkable because this equation derives from different physical reasoning, though with a similar goal in mind. Haldane reasoned that the population begins with a normalized size of 1 on the right side of the equation 8. After selection, that quantity is reduced by amount seen on the right side of equation 9, S little q in this case. Haldane identified this reduction as the genetic death divided by the adult population size, so it has units of genetic deaths per adult, and defined it as the cost. Haldane used the same mathematical method, reflexively and without physical justification, in all his various case studies.
The physical justification is that there are organisms which contain genomes which do not reproduce in any given generation. Therefore, those genomes are not replicated. Therefore, they have died, whether literally or metaphorically. But in either case, with an obvious physical justification and meaning. I struggle to think that the Romain cannot understand this.
Later, Crow's formula covered the full range of S values, but based on the same physical reasoning as Haldane. Crow defined the cost as the ratio of those eliminated, S times little q, to those not eliminated, 1 minus S times little q. That definition twice uses Haldane's concept of genetic death. Their formulas give mathematically correct predictions, but based on dubious physical interpretation.
Notice that their being dubious is entirely the invention of Romain. It's trivially easy to see what is meant by genetic death. Of course, this is all pissing in the wind, since the mere existence of humans and whales and other such slowly reproducing organisms is enough to show that Haldane's dilemma is a modeling problem, not a problem faced by actual organisms.
As pointed out by Feller, Moran, Hoyle, and Wallace, Haldane's definition of genetic death does not make sense physically.
Hey, fun fact about Hoyle, he's the one who did destructive testing on an Archaeopteryx specimen in an attempt to disprove evolution, and decided that actually the feathers of the specimen had been faked by cement and chicken feathers, and that the fossil was actually of a pterosaur. All of that is absolutely insane, and while Hoyle did contribute to astrophysics, he was a kook, and none of his attempts at biology went anywhere. So citing him about evolution is like citing Andrew Wakefield about vaccine safety or Richard Carrier about early Judaism. It's completely insane to do it, yet idiots and grifters keep doing it anyway.
And they give compelling examples of its failure. I here identify the reason. Equations 9 and 10 are separate mathematical steps that represent a single, indivisible physical process. In nature, there is no step of applying selection followed by a separate step of normalizing. Rather, this all happens in one swoop. Haldane's genetic death concept exists only in the middle of these equations, at equation 9.
Imagine an equation putting different terms on different aspects of a process. I mean, next thing you'll be telling me that mass and acceleration both exist at the same time when a force is applied.
It is a mathematical phantom that does not exist in physical reality. The solution to this is to jettison his concept of genetic death and build upon firmer ground.
The solution is to redefine terms. Fun. That's not at all a sign that you can't actually win a scientific argument on its own terms, and instead you want to have a different argument and pretend it's the original one.
The physical meaning of genetic death has been a continual obstacle. Examine the simplest case, a single generation, sometime during a single substitution in haploids with constant population size and uniform reproduction rate, followed by selection through juvenile death.
Selection by juvenile death means that those juveniles don't reproduce, and so are genetic deaths per Haldane's definition. It also means that even if the previous generation had a uniform reproduction rate, the next one sure won't, since some of them don't reproduce at all.
And S is much smaller than 1. In this simplest case, and using the genetic death concept, we can construct a correct argument that limits the substitution rate. But extreme care is required because the logic is indirect and complicated. Firstly, the deaths had to be converted, somewhere, somehow, into a requirement on reproduction rate.
Only because the population is assumed to be constant. If it's not, then we don't actually have to care about the real value of reproduction rate, only the normalized value, because fixation is about proportion, not absolute numbers. At this point, I feel like Walter is trying not to understand this stuff.
Traditionally, this conversion was done implicitly, not explicitly, so it was not well understood. Secondly, in the simplest case, genetic death was interpreted as a death of old-type individuals caused by the presence of the new allele, or, equivalently, the deaths would not have occurred if the new allele had been absent.
That the genetic deaths are caused by the individuals with the substituting allele struck me as absurd on its face, but then admittedly, I'm not an expert in this field, much like our author here. Of course, the difference between him and me is that when something is claimed that seems absurd in a field that I know I'm not an expert in, I actually ask experts what's going on, and so I asked Zach Hancock and Dan Stern-Cardinal, both PhD biologists, both geneticists, both of whom have worked on evolution and population genetics, and both of whom are friends of the channel. And, yeah, they've read this whole thing, and, yeah, their take is the same as mine on basically everything.
So, identifying genetic death seemed to require that the scenario be examined under two circumstances, with and without the substituting allele. But what possible relevance could the second circumstance have? Why would the second circumstance, where substitutions are absent, have any bearing whatever on limiting the substitution rate? This mystery was cause for confusion. Some researchers interpreted the mystery as follows. If the environment is deteriorating and the new allele is beneficial relative to the deteriorating environment,
all alleles are only beneficial or deleterious relative to an environment, short of alleles that are so deleterious that they just cause a premature death regardless of environment. Those are always deleterious, but they also obviously do not persist in the population.
Then the same deaths would occur even if the new allele had been absent. Likewise, the presence of the new beneficial allele would not increase the deaths. Therefore, the deaths are not caused by the new allele, but are entirely caused by the environmental deterioration.
Okay.
I really hope there's a point to this. One that isn't just whining about Haldane's definition, but one that actually applies to real life, to real organisms, that would in some way, if true, present a problem for the consensus view in biology. I'm not expecting that to occur, though. Another thing I'm not expecting, but would absolutely love, is if you join my Patreon. My Patreon is the best way to support the channel. It starts at just $1 a month, and you can get a 15% discount if you pledge annually. You'll get access to videos like this early, as well as access to my supporters-only Discord. YouTube is very fickle, and Patreon is more predictable, and I keep more of the pledges made there than from any YouTube revenue source. And this channel only exists because of all my supporters. So if you like what I do here, please consider supporting if you can afford it.
On such basis, some researchers, e.g. Felsenstein, concluded that in a non-deteriorating environment, beneficial substitutions would have zero cost. That faulty conclusion, though still common today, was due to genetic death and the confusion it creates. In short, even in the simplest cases, many investigators struggle to understand genetic death of the old allele and how it could limit the substitution rate of the new allele. But the situation quickly gets worse. When the scenario moves away from a uniform reproduction rate and allows selective eliminations due to lowered fertility, then some, or all, of the genetic deaths are virtually or imaginary, not actual deaths. Haldane said to count these virtual deaths as equivalent to real deaths, but he did not explain why.
Because otherwise, they can't be added into the term of the cost of selection, yet these are individuals who would have reproduced but did not, so they must be included. Of course, since we're normalizing the population, it doesn't matter much either way. The substituting allele will, by definition, have a higher normalized reproduction rate and a lower normalized rate of genetic deaths, because those things end up coming out as synonymous.
Since these individuals are never even conceived, their physical interpretation is a source of confusion. For example, why would virtual, or imaginary, deaths of the old allele limit the substitution rate of the new allele? The answer can now be realized. Haldane's virtual death concept is merely an artificial tabulation device that helps us calculate the correct answer, tabulate all selective eliminations, even those due to lowered reproduction rate, as though they are actual deaths. That really means all scenarios are calculated as though they have a uniform reproduction rate. In effect, Haldane was using the cost equivalence principle. Using that artificial tabulation device, and when the selection coefficient approaches 0, the number of selective deaths of old-type individuals divided by the adult population size, which is Haldane's cost concept, happens to equal the required excess reproduction rate for the entire population. In other words, there are a certain number of selective deaths of the old type, and the population is required to produce an excess reproduction rate sufficient to supply those old type progeny.
Only if we assume a constant population size. That's why outside of Ramin's work, these values are normalized. It occurs to me that some of my audience may not know what normalization is. Data rarely comes in as raw values from 0 to 1, but such values are very useful for comparing different datasets and using statistics on. So datasets are often transformed into values from 0 to 1. This is actually not very difficult. First, the value of the smallest data point is subtracted from all data points. Then all the data points are divided by the new value of the largest data point. So for example, let's say that we had a dataset with some units that don't matter much, and values of 432, 981, 928, 10,938, and 154. 154 is the smallest of these numbers, so we subtract it from each of them. Then we get 278, 827, 774, 10,784, and 0. Now the largest remaining data point is 10,784, so we divide all the remaining numbers by that number, and we get 0.04006, 0.9097, 0.8605, and 1, and of course 0. Now the relative size of the data points are the same, but they go from 0 to 1. The data has been normalized. In the case of a population, normalization would mean dividing by the population size. So a reproduction rate of 4 in a population of 1,000 would be equal to a normalized reproduction rate of 4 divided by 1,000, or 0.004. This is done because it allows us to see the relative fitness of various phenotypes and adjust things like cost of selection to be comparable across populations. And so that we don't have to always include population size calculations, since population size ultimately does not matter that much to the question of how much reproduction above the average has to occur for a new allele to reach a certain frequency in a population. Because, as I keep saying, fixation isn't about reaching a particular absolute number of individuals with an allele, but about the frequency of the allele in the population reaching 1 or 100%.
However, under a uniform reproduction rate, this same rate is also required for producing new type progeny, and this latter requirement remains unaffected even if the reproduction rate is not uniform. This again uses the cost equivalence principle. In this way, we can translate Haldane's cost concept, genetic death, into my cost concept, and provide legitimate physical rationale for Haldane's cost argument.
So let's agree there. Now what? That doesn't help any, since his cost calculations break the moment population declines, and they in no way present a problem for fixation of alleles in slowly reproducing organisms, since they also studiously ignore things like crossover and sexual reproduction. I'm getting tired of Walt trying to justify changing the definition for cost. I would like him to just make an actual argument against evolution, since that's the whole point of this.
That rationale, however, is also awkward and convoluted, which has kept Haldane's argument under a cloud of confusion. Larger selection coefficients add another layer of complication and confusion. Moreover, when a scenario includes multiple substitutions, overlapping in time, or diploids with non-dominant substitutions, a correct physical interpretation of Haldane's argument appears intractable. Genetic death quickly becomes a mere mathematical equation, the correct physical interpretation of which becomes obscure at best. Nonetheless, many investigators advanced the genetic death concept, and the accompanying confusion fostered various mistaken solutions to Haldane's dilemma. They failed to realize that Haldane was not focused on death, but on something else. Haldane's paper repeatedly speaks of reproductive capacity. It was clearly a key focus on his thinking. His paper did not explicitly use the term reproductive access, though later commentators, such as Morel, understandably attributed that essential concept to him. Crow and Morel advanced this clearer wording, though still heavily intermingled with the concept of genetic death. Their usage of the term reproductive access further suggests that the cost concept they were grasping for is clarified in this paper. Unfortunately, their proper focus on reproductive access was largely brushed aside by the rise of the new concept, genetic load.
Well, that whole section was mostly a waste of time. Thanks, Walt.
Genetic load.
Okay, we have 3 sections left.
The substitutional load is defined as the percentage decrease of average fitness within a given generation caused by the substitution of beneficial mutation.
Oh my god, that's a dumb and confusing way to define it. It's actually the difference between the average fitness in a population and that of some genotype under consideration, whether it be some actual or hypothetical genotype. I don't know why Walter feels the need to be so damn obtuse.
Let WO be the fitness of the optimal genotype, and let WA be the average fitness of the population. The substitutional load is WO minus WA, all divided by WA.
Well, he does know the real definition, at least there's that. I'm not sure why he can't explain it well, but whatever.
In the haploid case, WO equals 1, and WA equals 1 minus S little q, so the load is S times little q divided by 1 minus S times little q. In other words, the load and cost happen to be equal.
Okay, maybe it's me, but I do not follow why the optimal fitness must equal 1. He's stating that like it's an obvious conclusion, but the fitness optimum of a haploid individual could be any arbitrarily large number. For example, a haploid fern could send out 100 million spores, all of which, at least hypothetically, could form a new diploid fern. By the way, ferns actually have a split life cycle where they grow as diploid plants that you think of as ferns, but those reproduce asexually to form gametophytes pictured here, which look nothing like a fern. Ferns are weird and neat. But anyway, the gametophyte fern could have a fitness of 1 million. Only if we actually normalize the population could we ever say that the optimal fitness value is just 1. But that would mean that that individual would be the sole progenitor of the next generation. What's actually happening, without Walter telling us, is that it's actually the fitness values that are actually normalized. Why he didn't bother to point this out, I don't know. He could have just said that the optimal fitness value is 1 by definition. Literally 2 words and it would have made sense. Romine really sucks at this.
Note, some researchers define load as WO minus WA divided by WO equals WO minus WA equals S times little q, which makes load equivalent to cost so long as S is much smaller than 1. A similar equivalence is also found for diploid cases. Kimura popularized this evidence, and load soon became the predominant way of discussing the cost of substitution. That is unfortunate because the load concept caused much confusion.
I get the sneaking suspicion that when he says that, what he means is that it has not lent itself to anti-evolutionist interpretations to the degree that he would like.
First, cost and load became viewed as identical concepts when they are different physically. They merely happen to have a weak mathematical equivalence, and then only under special circumstances, such as constant population size, s much smaller than 1, etc. Load obscures the physical processes, especially in diploids, and defocuses the concept of reproduction rate, which is rightly the central focus of cost theory.
Except that it's obviously not. Walter wants it to be, but that's not the same thing. This whole screed is his attempt to make his wish a reality, and it crashed hard because his work is trash, and he should feel bad as a person about that.
Second, substantial load has a strong counterintuitive flavor.
Guess how much being intuitive matters in science? It's not at all. Lots of things about science are counterintuitive, and are true nonetheless. You think it's intuitive that light travels as both a wave and a particle, and that its speed is always constant for all observers? It's not. You think it's intuitive that objects of different mass experience the same gravitational acceleration toward the ground? It's not.
Kimura notes that one popular criticism is that the substitution load of more advantageous alleles for less advantageous one cannot be considered a load, since the fitness of the species is hereby increased. The load argument claims the substitution of a new beneficial mutations into the population temporarily decreases the average fitness of the population, so a beneficial mutation causes a fitness depression.
No, because the way we measure fitness is by reproductive output. So if fitness went down, that means reproduction went down, and that means the allele is deleterious and fixed anyway. The only way for an allele to fix because of natural selection is for the reproductive rate of those with it to increase relative to the overall fitness of the population. Now that means a relative decline in fitness for the old type genome. That is not a depression of the fitness of the population as a whole. Why can't Walter figure out the difference between relative and absolute numbers?
While that made some sense to mathematicians, its physical interpretation has been a major source of confusion. Third, load strongly emphasizes the concept of fitness, which creates confusion.
This guy must be a fan of John Sanfraud, who also likes to pretend that fitness is some mysterious concept that's hard to define.
Fitness values are re-relativized frequently as various mutations enter, exit, or reach fixation, and the timing of which is generally unknown.
You just normalize the data to get relative fitness values, and you can and should do that every time there's a new period of time under study. But you can also use absolute fitness values if the concept of normalization confuses you. But just like there's no particular time you should renormalize data to figure out, say, the per capita crime rate of various cities, there's no special time to renormalize fitness data. That doesn't make fitness hard to define, it just means that, like with all things in science, your numbers are going to have a margin of error.
The continual conveyor belt of re-revitalized fitness values creates confusion, much like identifying the 10th of man on an escalator.
Weird, because I'm not confused, and the population geneticists I know aren't confused, although this paper does confuse them with how bad it is.
The load calculations depend on WO, the fitness of the theoretically optimal individual, whose identity and fitness is never changing.
It is changing in absolute terms. It's just that for modeling purposes, it's fixed to 1 because we're normalizing fitness values. And the highest data point in a normalized set of data is always 1 because that's what it means to normalize a data set. If I gave you 500 data sets, each of which was the measured weight of $1,001 coins from Canada, the loonie, each dataset would have the heaviest coin as 1 and the lightest coin as 0 if you normalized them. Of course, that does not in any way mean that all the heaviest coins have the same weight or that all the lightest coins do. The fact that all the datasets go from 0 to 1 is part of the definition of normalized data, not something to do with the uniformity of the coin mass of Canadian currency.
Moreover, in scenarios involving substitutions at many loci, a theoretically optimal individual typically never exists within the population.
That's true.
Load calculations have confusion surrounding the optimal fitness, WO, and how to handle it. And the current definition of load does not clarify it.
Even if that's true, so what? I mean, it's great that we can identify problems with modeling, and that would mean we have ways to improve models, but on the other hand, problems with modeling are not problems for real organisms. Basically, this whole paper is just an exercise in the reification fallacy, where one mistakes a model for the reality. It's like complaining that the real hills and valleys of the terrain don't have the relief lines that are printed on your relief map. Or that in reality, the state of Kansas isn't actually blue, even though it was on your map of states that you had in your classroom. Even if everything Walter has said about modeling population genetics were completely right, which it's not, it would mean that we have a problem with both his and the standard models of PopGen. But getting a model wrong doesn't mean that the problems with the model, that of inability to evolve, are problems for populations. The evidence that evolution has occurred is overwhelmingly from genetics, morphology, and paleontology. A model that shows it couldn't have happened is a model that does not match reality, and so it's a bad model. All Walter is doing is proposing more bad models in response to a model that started out bad and was made better, if perhaps more confusing to Walter.
A given fitness value can be realized with dramatically different trade-offs between reproduction rate and viability rate. These differences were often viewed as irrelevant because load focuses so intensely on fitness. This led to indiscriminate use of, and allocation between, these fitness components, and confusion was the result.
Whose confusion? It seems like it's Walter's, not anyone else's. The thing is that fitness is what matters in evolutionary biology. Evolutionary biology is about what the state of the genome is, was, and will be, and hence what the phenotypes that result from it will be at different points in time, and how those changes got to be that way. What matters to that is total reproductive output that then goes on to itself reproduce. It doesn't matter how granular you get. When you combine all those factors, they result in reproductive output. Think about all the factors in, say, the reproductive output of a male mammal. First, our mammal father-to-be must survive childhood diseases, predators, potential famines. Then he must strike out and find his own food independent of parental care. He must find and woo a mate. His testes must produce enough sperm to have a good chance of fertilization. He must produce enough seminal fluid. The muscles that propel the semen must be strong enough to actually propel the semen into the female reproductive tract. A high enough proportion of his spermatozoa must actually be motile enough to reach the ovum or ova of his mate, depending on species. The resulting zygotes must themselves be viable, and then his children will have to go through all of that childhood stuff again in order to find their own mates. We could account for all of this granularly, but we could also just count up his grandchildren and get the same basic result of fitness without needing to examine the muscles attached to his penis. I know which one I'm going to do if I'm trying to figure out population genetics.
Cost theory focuses on reproduction rate, and thus keeps a clear eye on this distinction.
And no one cares, because reproduction rate doesn't mean anything if all your kids are sterile, for example. That's why ideally fitness is measured over more than one generation.
This confusion increases manyfold in situations involving numerous substitutions overlapping in time.
Well, yeah, more complex situations result in more complex math. One example of math that isn't complicated is joining the channel as a member. It starts at just $1.99 a month, and you get early access to videos like this, access to my Discord for supporters, a cool badge and chat for live streams and videos, as well as access to channel-exclusive emojis.
Fourth, Load furthered the mistaken idea that neutral substitutions have no cost. This occurred because nearly neutral substitutions have very low load, and neutral substitutions have a load, or fitness suppression, of 0, by definition, and load was casually equated with cost. Moreover, neutral mutations and their substitutions cause 0 genetic deaths, and this seemed confirmed by the cost equation. Since S equals 0 for neutrals, their cost, approximately S times little q, seems to be 0. However, that is a faulty interpretation, because these cost equations are derived using S-values to specify the growth of the allele.
Here, Walter just complains that his definition of cost isn't used by other people, and so they don't agree with him. My message to Walt is, either write a paper that's worth a damn, or cope and seethe, maybe do some molding too.
When S equals 0, there is no growth, and hence no cost, but no substitution either.
Yeah, under Walt's version of cost, sure, but why do we care? Actually, why do we care is probably the biggest question anyone, even the now barely remembered hosts of the Answers in Genesis Canada show this series was supposed to be about, should ask.
In effect, it calculates the cost of not substituting a neutral mutation, 0. This again shows a real difference between the load and cost concepts.
I don't think anyone is trying to claim that Walter's personal meaning for cost isn't the same as genetic load. But the problem is that he's trying to convince us that his idea is better and more useful, and in service to that, he's now just pointing out how it's novel, but that was never a contentious point.
The erroneous notion that neutral substitutions have no cost caused confusion and encouraged various claims that beneficial substitutions likewise have no cost.
I noticed that there's no citation here. There probably should be one. Generally, if you want to claim that people are making a claim, you need to actually cite some people making that claim. That's just how you write papers. I guess we have another reason to reject this paper if we were peer reviewers: insufficient citations. Although in this case, I probably wouldn't recommend rejecting the paper on that ground. I just recommend revisions to add the citations. I'm not that mean.
Fifth, load is a single value for the entire population. Calculate using the entire population's average fitness, WA, and the optimal fitness, WO.
I feel like Walter has forgotten that we can and do make load calculations based on particular real phenotypes, not just hypothetical ideal ones. I'm not sure that would change his argument much, but I just feel like it's important to point out that load is not always some hypothetical calculation based on the accuracy of our estimate of a potential maximally fit organism.
There is no generally acknowledged method for calculating the load for each genotype, and, even if there were, there is no if clause to rectify negative loads and provide consistent physical meaning suitable for testing scenarios. By contrast, the cost of substitution has a distinct, physically meaningful value for each genotype.
Yeah, but one where just having a population decline breaks Walter's math, so it's useless, regardless of how bad a model genetic load is.
Though usually only the largest costs are calculated for testing a scenario. And these give the reproductive requirements for each genotype. In other words, load theory is a blunt instrument, while cost theory provides finer detail and a more transparent connection to the underlying physical processes.
Me! Too bad it so easily breaks and can't account for things like bottlenecks or the founder effect. You know, things that we know for sure happen in the real world. Things that are part of many species' history, including Homo sapiens.
Cost and load are different lines of physical reasoning. These terms ought to be used more carefully and no longer interchangeably.
I don't care. I'm skipping this last paragraph. I've been at this too long already, and all he does is continue to whine about how his model isn't the same as load, and he likes his idea better.
Continuous generation models.
When I first saw that heading, I thought of spontaneous generation. Luckily, that's not where we're going with this. This is also somehow only our antepenultimate section of this so-called paper. This might actually kill me. If it does, then all your Patreon and membership money will go towards funeral expenses.
The discrete generation model from equations 3 and 4 can be generalized into a continuous generation model. At time t, let p of t and dp of t dt be the effective number of individuals who produce type big A progeny, and its time rate of change, respectively.
Are we really just going to be turning the already useless model into a useless model that uses calculus? Why do I do this to myself? Oh, right. Self-loathing.
Of course!
Let big T be the effective generation length. This is approximately the parents' ages when they give each birth, averaged over all births that reach mid-parenthood.
What the hell is mid-parenthood? If you think you know, let me know in the comments, even if it's just a wild guess.
Big T is also equivalent to the time unit from the corresponding discrete generation model. This is what links the two models together. The instantaneous cost per unit time is equation 12, cost density of t equals 1 over p of t minus big T times dp of t dt, if greater than 0, otherwise cost density of t equals 0.
I just want to say thanks again to Verwache for reading all these equations. I know it wasn't easy, and it helps with accessibility big time.
This equation includes the fact that, at any instant, the increase in p is due to the value of p from a time 1 generation previous. That is, the t-parameter models the time delay between when progeny are born and when, on average, the next generation of progeny is produced. With this in mind, the analogy with equation 3 is exact. The difference is that equation 3 gives a cost per whole generation, whereas equation 12 gives a cost per infinitesimal time slice, not per whole generation. Other than that, they both represent the same thing, the required excess reproduction rate.
Then they'll both have the glaring problem that they don't actually work, since sometimes, contrary to what Walter will allow, we get negative costs in his framework, and not under crazy exotic circumstances, but anytime deaths outnumber births.
As before, the total cost merely sums, i.e. integrates, over a substitution cycle. Equation 13, total cost of substitution equals the integral over cost density of t dt. If the substitution is very slow compared to the time delay t, then the total cost will asymptotically approach the classical formula, equals log base e of k, my equation 6. However, the total cost increases rapidly for faster substitution speed, such as shown in Table 2. This continuous model is directly applicable to haploids, and can be extended to diploids using methods given in the next section. We can contrast with Flake and Grant's continuous generation haploid model. They define the cost density as the ratio of instantaneous rate of loss of the old trait to the instantaneous population size. Their derivation then arrives at equations identical in form to equations 12 and 13, absent the big T parameter. Their final result then gives a total cost identical to the classical formula, equals log base e of k, equation 6, independently of the substitution speed. This mistake arose because of a focus on genetic death and loss of the old trait, where a time delay would not suggest itself. Once again, genetic death was a source of confusion.
Okay, well, it's good to know that we can use some pre-calc stuff to turn a discrete model that's useless into a continuous model that's useless. I mean, I already knew that, but hey, it's nice that Walt knows it too.
Diploids.
Hey, I'm a diploid. I bet you are too. Also, this is the last section before a very short conclusion.
Since we are interested in required reproduction rates, we must refer to some identifiable reproductive individuals. This paper views the individual in the ordinary sense as a body. Therefore, we focus on genotypes because genotypes correspond to individuals, bodies capable of giving progeny. For example, genotype big A corresponds to a well-defined reproductive body, but allele big A does not, as it ambiguously refers to big A or big A little a.
No, it doesn't. It refers to the variation at a particular locus on the genome, not to individuals at all. This is pretty basic, and the fact that Walt gets it wrong shows you why he's so off base with this whole so-called paper. He doesn't know what he's talking about at the most basic level. He is probably worse at genetics than a high school student who got an A in biology.
Alleles substitute, but genotypes are the vehicle for the increases.
That's fair.
Therefore, we always calculate the costs of specific genotypes, not alleles. This distinction is invisible for single substitutions in haploids, where the allele and genotype have a one-to-one correspondence, but this distinction is essential in all other cases.
I would actually agree, although I suspect that he's going to ignore that genotypes of the non-substituting type can result in production of the substituting type. I don't know. Let's find out together, shall we?
In diploidy, individuals correspond to the three genotypes: big A, little a, and little a.
Correspond is a weird word here. I would rather say that individuals have those genotypes, but whatever.
At the start of generation I, let the starting count of breeding adults be denoted as P big A plus P big A plus P little a equals NE. The ending count of one generation becomes the starting count of the next. Dividing by NE gives the starting genotype frequency denoted by F big A plus F big A plus F little a equals 1. The parental allele frequencies are then easily calculated. Small p equals F little a plus 1/2 F, and little q equals 1 minus little p. Mendelian segregation, in combination with various mating schemes, such as randomly mating and inbreeding, alter genotype frequencies between parents and progeny, while tending to leave allele frequencies unaffected.
No. Inbreeding is defined by a reduction in genetic diversity, which means alleles dropping to 0 frequency, with other alleles increasing in frequency. If allele frequencies aren't changing, then there's no inbreeding going on. Once again, Walter is showing that he doesn't have the knowledge of this topic to even have an opinion on it.
By this means, some genotypes can decrease while others increase. This predictable change is due solely to the passive remixing of alleles at the gene level and does not require reproductive excess of whole bodies. Therefore, we do not tally it into costs that whole bodies must pay. So, we next assess its effect and disallow it from our tally costs.
It's not clear what the antecedent for it is there. Hopefully that gets cleared up.
That is accomplished with the concept of effective producers. I will use random mating to exemplify how all cases are handled.
That's a pretty common simplification in PopGen, so I have no problem with that.
Random mating produces genotype frequencies given by small p squared plus 2 small p times small q plus small q squared equals 1. For other mating schemes, that equation will be different, but the following steps will usually remain the same. Multiply by the effective breeding size of the population to obtain.
Put in the comments what you think the chance is that he could actually define effective population size and tell you what kinds of population structure raise or lower it.
NE times little p squared plus NE times 2 times little p times little q plus NE times little q squared equals NE. That equation gives the effective producers of the three genotypes. An effective producer can be modeled as an abstract asexual adult that, in effect, produces progeny of a given genotype at the same reproduction rate required of its real sexual counterparts. This parcels the breeding population into 3 portions, each portion going toward the production of a specific genotype. See Figure 3, for example. It is as though all the reproduction of NE times little p squared, asexual, adults go solely towards producing big A genotypes, while all the reproduction of NE times 2 times little p times little q, asexual, adults go solely towards producing big A genotypes, and so forth. That equation shows how the population's reproductive capacity is redistributed. A genotype's effective starting count is the number of effective producers of that genotype and is given by the terms of the previous equation, here labeled as big P, big A, asterisk, plus big P, big A, asterisk, plus big P little a asterisk equals NE.
I feel like it's going to be a while until he gets to a point, and I'm pretty sure that the point will just be that for a genotype to spread, it has to have lots of kids, and he'll again ignore that fixation is about proportion, not absolute numbers.
As the cycle of one generation completes, call the number of breeding adults of the ending count, labeled with a prime, as big P big A prime plus big P big A prime plus big P little a prime equals NE prime equals NE times, in brackets, F big A prime plus F big A prime plus F little a prime. In generation I, each genotype has a cost given by equation 3. Equation 14, cost big A I equals big P big A prime over big P big A asterisk minus 1 equals G times F big A prime divided by little p squared minus 1 if greater than 0, otherwise cost big A I equals 0.
Sorry, I'm just making sure that you're still with me. If you got to this point, put Rosebud was actually the sled the whole time in the comments, so I know who the real ones are. Also, if you actually get this whole reference, then good job.
Equation 15, cost big A I equals P big A prime over P big A asterisk minus 1 equals G F big A prime over 2 little p little q minus 1 if greater than 0, otherwise cost big A I equals 0. Equation 16, cost little a I equals big P little a prime over big P little a asterisk minus 1 equals G F little a prime divided by little q square minus 1 if greater than 0, otherwise cost little a I equals 0.
He really hates negative costs, even though that's just what happens when populations shrink, or at least can happen. So far, he's only acknowledged that that's even a thing once, but of course we know from things like the founder effect that shrinking populations are a significant part of the fixation of alleles in a new species. His model just can't account for this. Or rather, it says that in such cases, cost is 0, which rather removes the problem of the cost for replacement of new alleles precluding evolution at large scale. While it's true that aspects of punctuated equilibrium, like group selection, have largely not held up to scrutiny, this aspect of it has indeed held up quite well, and here's Walter just pretending that it's not important.
Those equations have a straightforward interpretation. Take a case where the population size is constant.
He sure does love those cases, since they're usually the only ones that work out well. But also, if the population is constant, then a new beneficial allele starts spreading. While keeping the population constant, it means something has made the other alleles less fit than they were before, leaving the overall fitness of the population the same. That's not normally how it works. Populations as a whole can become more fit, and then their whole population goes up. That's what adaptation is in the first place.
G equals 1, and focus on the substituting genotype big A. Its effective producers have a frequency of little p squared, and this genotype ends the generation at a greater frequency, F big A prime. That requires a reproduction rate F big A prime divided by little p squared, which is a cost of continuity of 1 plus a cost of substitution of F big A prime divided by little p squared minus 1. Within a given generation, the three costs big AI, cost big AI, and cost little ai, do not stack onto each other. They are three separate pipelines, incurred and paid in parallel. The payments that go towards one cannot go towards paying the others. Those three costs are the minimum conceivable cost of substitution incurred by each genotype.
Assuming a constant population, which we know isn't a realistic assumption, as I've already shown with Walter's own math, once you relax that assumption, suddenly there's no minimum cost. Although in practical terms, there may be, since some starting population numbers needed to get to particularly low costs might be unrealistically high for an actual population.
Each of those is a mechanical limit that cannot go lower regardless of further details about the selection process.
Wrong.
For decreasing genotypes, such as big A little a during the second half of the substitution and little a, this limit is 0.
Yeah, but only because Walter arbitrarily capped it at 0. If you ignore his arbitrary cap of 0, you'll find it's actually negative. And what was his reason for capping it at 0? He didn't give one. He just likes it that way, I guess.
But that cost can, and usually does, go substantially higher. For pure viability selection, all subgroupings of the population have the same reproduction rate.
Viability selection has not been defined, so I suppose it falls to me. Viability selection is selection from fertilization to reaching the age of sexual maturity. So for instance, if a genetic condition causes one to die as an infant, then that individual is being selected against in viability selection. I don't know if he has any reason to say that viability selection is the same across all variations of a genome. It's not actually true, since some organisms with the same genome will in fact die before reaching sexual maturity, while others do not, and that rate could very well be something affected by genomic differences that result in phenotypic differences. Is this just another assumption he's making? I'm not sure.
Then, some unfavored genotypes are eliminated by premature death.
Oh, I misunderstood. Okay, so he's just saying that in this case, they all reproduce equally, but some phenotypes are actually more likely to die before sexual maturity. I mean, yeah, that'll happen. Oh well, at least I got an excuse to tell all of you what viability selection is. It's not a term that I hear much, so I figured it was a good time to explain it.
Therefore, all genotypes incur the same cost of substitution, cost big A I.
I guess with Walter's definitions, where cost is literally about the absolute number of babies you pump out, that makes sense. But really, does he want to have a substituting allele that spreads through a population but has the exact same cost as all the other alleles in the population? That seems like it runs counter to his whole thing. And also, like, maybe this is an example of why genetic deaths are used by real geneticists, since those dead juveniles count toward that cost.
On the other hand, for pure reproductive selection, genotypes differ in the rates at which they are produced, therefore their costs may approach these mechanical minimums.
I prefer the term minima, but that's because I'm a snobby dickhead. So I don't know, maybe don't listen to me. It's not like I'm a linguist. But also remember, these minima only exist under his arbitrary conditions of the minimum being capped at 0, and only for populations that have a steady population size. The first is arbitrary. The second is known to be violated in nature all the time. So this whole model just falls apart. And that's to be expected, since his model is being made to suggest that evolution more or less is impossible. Yet we see it happen in the real world, so obviously his model is wrong. Remind is in the same position as a flat earther telling you that you can't just sail around Antarctica, because if you look at his map, you'll see that it's a huge ice ring with a circumference of hundreds of thousands of kilometers. That just means that his map is wrong, because there are boat races around Antarctica that get completed in sailing ships. When your model conflicts with reality, reality wins. It's true for flat earthers, just like it's true for Walter.
In any case, the cost of substituting genotype, cost big A I, is unaffected by those circumstances and remains the critical focus for testing the plausibility of a scenario.
Except that it's only unaffected by how selection works and the environment and such if we assume a particular required time to fixation, which he tends not to mention, and we assume a constant population, so in reality, this doesn't apply.
It is usually sufficient to focus on the greatest of the three costs, as this cost, and its payment, almost always forms the most stringent test of the scenario. Thus, equation 17, cost I equals maximum cost big A cost big A little cost little a. For a well-behaved substitution,
that implies the existence of naughty substitution. I want to know about that.
Cost big A always dominates, therefore, by equation 4, equation 18 total cost of substitution equals the sum of cost big A, I. The above method applies under the widest circumstances. Under the same model assumptions used by Crow and Haldane, the above equations reduce to theirs, including Haldane's 1957 equations for all his substitution case studies.
Congratulations to Walter on making a bad model that also reduces to another bad model. I'm not sure why that's something you'd want to do, assuming you're both honest and know what you're doing. But of course, we all know that he's not both of those, and he might be neither.
That includes 1. Haploids, 2. Diploids with varying degrees of dominance, 3. Inbreeding, and 4. Sex-linked loci.
Walter doesn't seem to know what inbreeding is, since he seemed to think you could inbreed while increasing genetic diversity, which is like saying you can descend by climbing up a ladder.
For each and every generation, the cost of substitution identified in this paper reduces to the traditional value,
making this paper pointless, once again giving us a reason it wasn't published.
Figure 3. Reproductive capacity is redistributed by Mendelian segregation in diploids. This example shows the percentage of the population's conceptions that produce progeny of each genotype. In this example, where little p equals 0.4, only 16% of the conceptions go towards reproducing progeny of genotype big A.
Cool, I guess? Not sure why that's a figure, but I guess I can't say that there's anything wrong about it. And there's nothing wrong with a bit of a call to action either. So I'll take a minute to say to you that if you're enjoying this video, please hit the like button, leave a comment, and share the video outside of YouTube. All of those things really help with the algorithm. And with a video like this, I really need that help.
Conclusion.
Finally.
Darwin noted that evolution requires excess reproduction. Cost theory quantifies how much excess is required, and Haldane made a tantalizing initial contribution to the establishment of cost theory.
Haldane more or less invented it, noticed that it was wrong, and then other people fixed that.
Haldane was aware his conclusions would probably need drastic revision,
because they didn't match reality.
This paper contributes to that revision by 1. Rebuilding the cost concept on foundations more direct and less confusing than genetic death.
Except that, as we just saw, it doesn't really know how to deal with viability, selection, or declining populations, meaning that it's actually less useful than the already not very useful model of Haldane.
2. Generalizing the cost concept, thereby eliminating any requirement for constant population size.
Imagine saying that you eliminated the need for constant population size, when in the previous section you relied on that very assumption to get your math to work out. This is just a clown show of an article.
Small selection coefficients, discrete generation models, etc. 3. Identifying the compatibility, mathematical, terminological, and conceptual, between this clarified cost concept and the traditional thinking.
And in every case where he claimed that it just came down to, see, I got the same numbers and equations as Haldane when you think about it. Okay, cool. I guess that means there's no point in having written this then.
4. Showing the difference between cost and load, and why cost is inherently clearer.
No, it was shown why Walter prefers his own bespoke definitions of cost, but since his definitions break all the time in the real-world scenarios, who cares?
5. Eliminating various matters of confusion, such as notions that beneficial substitution incur no cost, or pay for themselves, or cause extinction.
I'm going to disagree that he did any of that.
6. Showing that the critical cost of substitution is set by the substituting genotype's growth rate and is not reduced by environmental change or the type of selection processes, soft or hard.
Well, he tried to do that, but it was trivially easy to show that he did not.
The treatment given in this paper is fundamental and will eventually need to be expanded to achieve broadest applicability.
Or really even narrow applicability, since right now I'd argue it applies to more or less no actual populations in any kind of actual diachronic investigation of genetics. You see, I too can use big fancy words. I'm on at least the same level of qualifications as Walter or mine, Of course, I also have the advantage of bothering to check with real experts, so I think that gives me a leg up.
A. Scenarios fall into various further categories, depending on the detailed nature of the selection process, each one affecting the cost analysis in distinct ways. B. Multiple substitutions, overlapping in time, introduce additional subtleties into cost calculations. C. The various other costs, such as the cost of mutation, cost of segregation, and the cost of random loss, must be incorporated for a complete picture of cost theory and its applications, and these will be addressed in a separate paper.
I wonder if that paper ever got written. I assume not, since this paper never got published, at least not anywhere that matters.
I share with Haldane and Kimura and Crow the belief that quantitative cost arguments should play a part in all future discussions of evolution. I also agree with George C. Williams that the time has come for renewed discussion and experimental attack on Haldane's dilemma.
Great for him, but that is finally the end of that interminable pile of trash. Now onto the shorter pile of trash that is Answers in Genesis Canada telling us how impossible evolution is, and to check out some paper if we don't believe them. Of course, I've checked out all their citations so far, and found them all to be embarrassingly bad. So let's see what else they have for us.
The article title was Cost Theory and the Cost of Substitution, a Clarification.
Yep, it sure was.
What Ramin really pointed out was this cost of substitution. You're really talking about how quickly the mutants, the new creatures with new abilities, how quickly they can procreate.
Yes, that was what he talked about, ignoring the fact that they can become the whole of the population without even procreating once. Walter Ramain doesn't understand population genetics any more than Austin Whitsitt understands the globe or how not to spit on his romantic partners, or any more than the average megachurch pastor understands that no means no, or that children can't consent.
It's not so much to do with how you wipe out the other 98,000 ones. It's how you actually get those. Okay, so you finally get a new mutation. You finally get a couple that's got these genes, and they can start passing them on. Or if you've only got one, and only one can pass on the gene, how quickly could that overtake the population?
Overnight.
Meanwhile, all these other mutations are happening. A lot of them bad.
Usually, yeah, more mutations are bad than good. Although, as already discussed, that's neither always a problem nor always true.
That's what we observe in things. So anyway, the bottom line is Haldane's dilemma has not been solved. It's actually gotten worse.
On the basis of an unpublished paper that did not make its case and that, by its own admission, didn't actually add anything to Haldane's dilemma, all while everyone else can just look around the world and notice that Haldane's dilemma is false, regardless of if it has been solved.
And you can check out those two articles to get more information.
Yeah, that's a quick summary. There's of course a lot more details that we're not gonna do.
Oh, I did them, and it only lowered my opinion of Answers in Genesis Canada. That's what turned what was supposed to be a quick 1 or 2 hour video into the 6-part monstrosity that this series is.
Okay, we're still on answer number one, supposed answer number one.
Right, if those 8 mutations were passed on for 3 billion years, it gives you 3 gigabytes of information.
So we've got a third response to that one answer. Third, it assumes that the mutations that will be fixed are the sort that create new structures, new things like wings and feathers and horns and beak or whatever.
But those aren't new structures, they're modifications of what came before. Wings are modified arms, beaks are modified mouths, feathers are modified placodes. Evolution always just tinkers with what was there before. From one step to another, you'd never really identify anything new, and yet when you step back, everything is different and it seems like there's a lot of new stuff. It's similar to how language changes. At no point did one generation realize that they spoke a different language than the generations before them. In fact, most of my audience has met their parents and grandparents, and some perhaps even their great-grandparents. In all those cases, they probably spoke the same language as those ancestors, unless there was some immigration involved. Yet we know that over time, languages change to the point that if you speak contemporary modern English, you'd need to learn the earliest forms of English like a foreign language. Here's a quick example. The first sentence in the original text of Beowulf is Hwat weigardene in jardagom theon kuninga thrumjefrunon hutha athelingas elen fremadon. My guess is that you didn't understand that unless you studied Anglo-Saxon. In modern English, it would be, Lo, the spear Danes in days gone by, and by the kings who ruled them, had courage and greatness. We have heard of those princes' heroic campaigns. Similarly, while you might see a huge difference between an animal like Lagasuchus and a songbird, if you watch the evolutionary transition, nothing dramatic would really happen in terms of morphology the whole time. And at no point could you point to some aspect of the animal and say that there had been no antecedent to that aspect.
But it's becoming the consensus, even among evolutionists, that mutations are like spelling mistakes in an instruction manual, where you're going in and randomly mutating things.
So it's becoming the consensus that mutations are random. That's what he just said after removing the well-poisoning stuff about spelling mistakes in instruction manuals. And yeah, that's long been the consensus. That's not new.
These changes can be adaptive, they can be helpful to survival, or
they can be
beneficial.
Yeah,
we can use that term. That's not a no-no for us to use that term.
Right. Yeah.
I mean, it was a no-no for you for decades, but whatever.
There are beneficial mutations in certain circumstances, but they're still heading in the wrong direction.
That's a phrase that doesn't mean anything. Evolution isn't directional.
Even the beneficial mutations that have been published in the scientific literature, they're still information-deleting mutations.
Right.
Wow. And if only creationists would tell us what their definition of information is, and how they know that one genome has more or less than another, and what the effect of a given mutation is based on that definition, then that might be a meaningful sentence. Since they have not done so and never will do so, it's just a meaningless thing to say. But it makes people who don't know anything about biology impressed, and that's the important thing for these two.
Sometimes these mutations produce, even though they delete information, they still produce some really interesting features.
Right.
Oh, you mean features that weren't there before, so new features. Get the fuck out of here, AIG, you bag of malodorous dicks.
But it's going the wrong direction to try to answer this question of how do you get the DNA code? How do you get that information?
Mutations are new DNA code. If you change the DNA, then you have new genetic code. There's no further question to answer. These two might not like it, but that's because they're idiots who probably are also horrible people.
So if you've got a small island,
like Belisarius Island, go on.
And it's quite windy, and you've got a group of beetles on the island, and some of them, they're winged beetles, and sometimes that's bad because they fly around and then the wind blows and they blow them out to sea and they die.
Yeah.
You get a mutation in the population, their wings don't get expressed, or let's say they're deleted from the genome.
That's not really how that works. I'm not aware of any beetles that simply do not have wings, although I'm sure in the gigantic diversity of beetles there probably are some. Usually what happens with flightless beetles is that the front wings, which form a shield over the animal, fuse along the center line, but there are still wings underneath.
Well, that's a benefit to the beetle in that situation. So all the wingless beetles...
It's a survival advantage.
That's right.
We could call it a benefit.
It's a
because
benefit
it's a survival advantage.
in that particular situation.
Benefit from any genetic trait is always situational.
So survival of the fittest, natural selection is going to occur because of this mutation.
Right.
Right. You select all the winged beetles out of the population. The wingless beetles, they're the ones that live. But if the water level drops in that area and all those wingless beetles get to the mainland where there's all sorts of snakes and bugs and lizards that are going to eat them, I'd rather be a winged beetle.
And here's Calvin with the amazing observation that adaptation for one environment is the same as becoming less adapted to other environments, as if that's not the most basic baby level shit.
So if you go from a winged beetle to a wingless beetle, you're not really evolving.
Did allele frequencies in the population of those beetles change? Yes. So they evolved. Calvin's opinion doesn't matter. Also, it's nice that they chose a specific example that seems like a bad result as a result of Dollo's law. Dollo's law isn't really a law, it's more of a principle, but it states that you cannot recover ancestral states in a lineage. So snakes can't go back to having 4 limbs, and birds can't go back to having teeth, and cetaceans can't go back to walking on land. In some cases, that puts organisms in dead ends, where they adapt in irreversible ways to conditions that will not persist, and then when they do not persist, that organism goes extinct. But what about lactase persistence in humans? That's a benefit that could also be lost, and that is not restricting further evolution in humans. And even in cases where the loss is completely unrecoverable, like teeth in birds, it doesn't always seem like it's a big hindrance. Birds get along just fine without teeth, and their beaks are quite effective. In fact, birds have managed to get just about every type of diet out there without teeth, including folivory, frugivory, omnivory, carnivory, etc. Cherry-picking a particular adaptation of a beetle doesn't make the rest of evolution wrong. It just shows that Calvin has a bad imagination, or is just dishonest. Given that he can imagine a boat carrying all the land animals through a global flood, I suspect that the problem is not with his imagination.
It's beneficial in the sense that it produces a survival advantage.
Right.
Yeah, that's what beneficial is. It's like he's complaining that seatbelts keep you safe in the sense of reducing the chances of injury or death in a car crash, and by lessening the severity of injuries that do occur. Yeah, that's what keeping you safe is.
That's great. No one's disputing that. But it's not beneficial in the sense of generating information. The beetles have lost the ability to produce wings.
Yeah.
Well, again, we have no idea if that means more or less information. No one has defined information in a way that creationists accept. But further, every morphological change has involved the loss of the ability to produce the ancestral morphology. You have arms and legs because your ancestors lost the ability to produce fins. You have a jaw and ear bones because your ancestors lost the ability to make gill arches. You have lungs because your ancestors lost the ability to make a digestive system that doesn't cause choking. You have a hair because your ancestors lost the ability to make protective hard bumps on the skin. If you want to discuss morphological change in terms of the loss of ancestral states, you can. It just doesn't mean anything in terms of the validity of common ancestry and evolution.
And so for this, like we mentioned, if we were observing these things all the time, I mean, there are a lot of scientists observing genetics today.
And watching evolution happen in real time, including the real-time observation of the evolution of brand new irreducibly complex features in organisms.
And so, even with all the random, information and mutations that are happening, even if you could show one or two beneficial mutations that actually, did actually increase something or cause something new to happen.
Like lactase persistence, the brand new muscles in dog faces that aren't in wolf faces, the evolution of a brand new metabolic pathway in E. coli, and the evolution of human tetherin antagonism in HIV, etc. Yeah, if only we could show that.
Even if you could show one or two, which they don't have any hands-down cases of that.
Yeah, except for the 4 I already had at the top of my head, and there are, of course, more.
In order for evolution to have occurred, you would have needed billions and billions and billions of these things to occur.
Yes.
I'm not sure where he's getting that number. The human genome itself is only billions of base pairs long, and it includes at least one whole genome duplication in its history. I'm not sure how you get to needing billions and billions of mutations to get to that point when you can get half of it as a result of a single mutation. Seems like he's just making up a big number, something creationists love to do.
Yeah. And so, and that's not what we've seen.
Yeah, we don't see billions and billions of mutations. Too bad that was just something Calvin Smith made up based on vibes.
So there's more details on some of these things. We've written up some of that example with the winged beetles that we just talked about. That was an actual observed example. We didn't just make that up.
Yep.
It might have been based on an example, but unless they produce a citation for an actual wingless beetle rather than one with a fused carapace, call them bullshit.
And you can there's links to these things and things like antibiotic resistance, blind fish in caves, and so on. Examples of where mutations have produced an advantage. And if you go to creation.com/QE or question evolution, creation.com/QE,
we're not reading through that. This is already far too long.
That'll take you to the page that has all 15 of the questions for evolutionists. And if you scroll down to the bottom of the page, it has our rebuttals, the content that we're talking about on the show.
By which he means just links to this video and those like it.
So we got other supposed answers?
Yes, answer number 2. Let's move right along.
I can't believe how long it took us to get through number 1.
Computer models have shown how mutations can lead to large-scale change. That's one of the responses that we got to this. Computer models have demonstrated this beyond a shadow of a doubt.
For example, Richard Dawkins, right? He has the weasel program.
The weasel
he thinks
program.
it is a weasel. I think that's the phrase he's looking for.
You're trying to get to a particular phrase and so on.
Yeah.
I do not think that computer models are great evidence of actual evolution. What they are is a way to check a model and see if it gets results that are similar to what we observe in reality. So it's a check on a model, not on reality. So I would not use this as my answer to the question, although it is very interesting.
But all of those computer simulations, every single one, they're artificial situations. They don't duplicate what's actually going on in nature.
Right.
Sure they do. I mean, they at least can. We know because they often give realistic results. And now I miss the Creatures franchise.
You might as well argue that the computer game Spore is a good example of is support for evolution.
Which was programmed by intelligent designers, of course.
Yes.
Spore is not a simulation of evolution. But the aforementioned creatures games were, and they did in fact result in evolution. But also scientists aren't using video games to test their models. They're using things like forward time genetics simulations. It's just like aircraft designers aren't using Microsoft Flight Simulator, but they absolutely are testing designs with fluid simulators that are more advanced than that and are designed to accurately simulate airflow over physical objects, and that do it well enough that the results match real-world observations. The same is true for the simulations of genetics that population geneticists run.
Spore, if you've never heard of that game, it's where you start off as a microbe and so on. You have a microbe, and then you can direct the evolution to try and get into some creature or something.
So it's exactly how evolution doesn't work. Got it. In fact, it's a simulation of constrained intelligent design, if anything. Also, maybe I should just switch to being a gaming channel. Then at least I wouldn't have to listen to these assholes and their ilk. And yeah, that was a jab at the now thankfully dead murderer, Gunter Beckley. Rest in piss, bozo.
But that's all of those things, and Dawkins' Weasel program, if you want more information on that.
Which is also not a great example of evolution, although it's better than Spore. And don't get me wrong, I love Spore. I spend hours and hours playing Spore. So did my frequent co-host Benthoven, no relation.
Just go to creation.com/selection. That'll take you to the Q&A page that has many articles on natural selection.
Off. I already went to 4 links like these assholes suggested. None of them helped their case. I'm done doing their homework assignments.
How natural selection is a fact. We have observed that. You observe how
it fits the creationist model perfectly.
It fits the creation model perfectly.
Tell Randy Galuza, who has made the baffling decision to just drop natural selection and pretend it doesn't exist.
If you start with all the information there in the beginning.
Sorry, the numbers on that have already been run. They don't work. There is in fact too much genetic variation in modern organisms for them all to have been descended from ark pairs after Noah's flood. Further, the different coalescence times for various parts of the genome are not consistent with a single origin for genetic diversity, nor are they compatible with the timeline of creationism. So we know from 2 lines of evidence that the created diversity model doesn't work as a model for genetic diversity in extant organisms.
And then you select out for various traits and so on, or climate selects out in this case.
Yeah.
Now, if you want to get a computer program that simulates nature and simulates these mutations to try to build up the human genome or the genome of something else.
That simulates what science is observing today, which is what our foundation
and then
science
extrapolates back in history, given you can put in mutation rates, you can download this from the internet, actually.
Yep.
So he wants a forward and backward simulation? I'm not sure why that would be a thing, but there are indeed simulations of genetics that are used all the time. And yes, you can download them. And they do in fact produce evolution. Although you're not going to get one that produces humans somehow, because humans aren't a goal of evolution, because evolution doesn't have a predetermined goal.
It's called Mendel's accountant.
Jesus Christ. Mendel's accountant can't even simulate known populations. It is designed to drive populations to extinction, even in scenarios that mirror real-life populations that do not go extinct in the time frame demanded by Mendel's accountant. The program borders on academic fraud. I would suggest that if you want to know more, you look at the paper Back to the Fundamentals, a reply to Basner and Sanford 2018 by friends of the channel Zachary B. Hancock and Daniel Stern Cardinal, published in the Journal of Mathematical Biology on April 3, 2024. It proves mathematically that not only is Mendel's accountant useless, but that genetic entropy is impossible.
And if you go to creation.com/Sanford, John Sanford is one of the developers of this software. And
the inventor of the gene gun as well.
The gene gun, yeah.
Wow! A guy who invented a thing later went on to be a liar for Jesus. Amazing. Also, that article is just biographical things about Sanford. If you want to know more about the story of some hack who decided that because, according to him, Jesus saved his marriage, he'd start lying for the guy, feel free to read it. I don't plan to, nor do I plan to subject you to it. This series is already too long, as I've said.
And he was a former evolutionist and is no more.
Mm-hmm.
Because his wife didn't like him, but she started going to church. I don't actually know, but I suspect that she still doesn't like him because he's pretty despicable, but now she feels guilty about divorce. Maybe I'm wrong. I don't really care. I'm just taking an opportunity to shit on the guy who said with a straight face that long sections about the origin of species was written in German and the book had no numbers or math. All of which is untrue and can be verified for free since the whole text of every edition is available online.
And we'll do an interview with him at some point. There's another show we need to do.
Yep.
So many ideas.
All of them bad.
Creation.com/Sanford, and you can find a link there to Mendel's account and download the program for yourself. Experiment with how to try to build up the human genome.
Mendel's accountant can't do that. It doesn't simulate genomes down to the nucleotide. Even if it weren't basically a program designed by a liar to tell you lies, it still couldn't do that. These guys just don't actually know what the hell it is.
The most favorable conditions that you put in there
That's right.
all lead to massive destruction of the genome.
I mean,
well, that's true. That is what happens no matter what you put in there. Yet that's not what we see in nature. It's almost like Mendel's accountant doesn't accurately reflect the actual behavior of populations in real life, and so is a useless simulator. Like if you were designing an aircraft and someone sent you a fluid dynamics simulator that resulted in all planes crashing whenever it was used, should you conclude that planes are impossible and aviation is fake, or that you were sent a useless simulation program? I'd conclude the latter.
You watch, evolutionists try to justify their position by creating, using computer software and computers to come up with these ideas. I mean, computers themselves are sophisticated. But anyway, so you're simulating, okay, we're looking for this target sentence. He thinks it is a weasel.
Which isn't what Mendel's accountant is doing. It's just making mutations that are abstracted to just fitness effects, and then it's running a simulation based on weighted fitness effects that predicts the future state and fitness of the next generation. Then it does it again. Of course, its weighting means that all populations will decline to 0, because it's designed to be wrong, but it doesn't simulate genetic sequences at all.
And so you randomly, you flip these letters around, but as soon as you get an M at the first one, it goes, oh, and it stops and it fixes it there.
Which isn't realistic, and it's why I don't much like Dawkins' computer program. I mean, I don't like much about Dawkins, to be fair, although this complaint is fairly minor compared to his bigotry, so whatever.
Even before you've got the entire sequence.
So the software has a goal in mind already.
Yes, that one does, but actual forward-time genetic simulations do not. That's why actual population geneticists and evolutionary biologists aren't running Dawkins' little program.
That's right, but with mutations should be completely random. So even if you got the M in place, and you got the E, and you got all the way, oh, what if that one's switched around again?
Yeah.
Then in the analogy, it is selected out of the population, something these two have already said is possible. The real problem with Dawkins' program is that it has a specified end state, unlike evolution. Not that it can eliminate low fitness changes. That's something it does correctly, and these two already agree with that. They're just too stupid to realize it.
See, you're actually programming the computer falsely to save what you want, but the problem is, with genes, you need all these letters of information to all be complete and then get expressed, and then they're a benefit to the organism.
And I feel like I did when a flat earther asked me to explain the half moon, and I didn't even know what he thought needed an explanation.
Getting the right letter inside before it's been expressed, that's no help.
Like getting it in a zygote? Well, mutations end up in zygotes and then they're expressed, so that's just how mutations normally work? Is that not what he means? It seems dumb if that's what he means, but given that we're talking about Calvin Smith, that something is incredibly stupid isn't a reason to think that that's not what he means.
So these are false simulations of what evolution would need to
That's right. Another thing we can mention is it's not the amount of change that we're discussing when we talk about beneficial mutations and so on, which we admit there are beneficial mutations.
Of course.
And it's not the amount of change. Within dogs, there's a huge range of different types of dogs.
Yep.
Or cabbages in the plant world. You have broccoli, cauliflower,
Brussels sprouts.
the little ones, Brussels sprouts,
Yeah.
and then you have cabbage, the big cabbage.
Right.
All the same group.
Yep.
The same created kind, baramine, we would say from the Hebrew.
Only because they don't know any Hebrew. But I've covered why that's a stupid etymology and neologism that could only be made by someone who doesn't know what grammar is, or that different languages have different grammar. But this is already too long, so I'll skip it. I would love for them to get to a point.
But the change between a broccoli and a head of cabbage, that's a huge change.
Right.
They don't look anything like each other.
They taste different too.
And they taste different. Most people...
I'm cutting until they get to a point.
It's not the amount of change, it's the type of change.
Right.
So I guess that's a point we can make as well.
Yep.
What are these types? The only thing they've said that I can think of are gain versus loss of information, but since they still refuse to let us actually have a way to test that, it's meaningless and just comes down to they accept the change that they can't deny and reject the change that they can't accept.
There's a third answer. You want to do that one?
Sure. Here was an objection that said, using words such as accidental and mistakes is misleading and misses the point entirely.
I don't know. I think that it can be misleading, but I also think that such terms can be used in good faith. Now, these two wouldn't know good faith if it bit their noses off, but whatever.
Well, this is a kind of, again, it's one of those, supposed answers that you're really missing the point.
Aw, does Calvin not like having his rhetoric criticized?
Number one, scientists, evolutionary scientists themselves use words like that.
They use
mistakes,
those terms.
accidental,
Yes.
because it's a random process.
And I wish they wouldn't, because those terms have negative connotations that are inappropriate. But then most biologists aren't concerned with how they'll be misrepresented by creationists any more than most astronomers are concerned with how they'll be misrepresented by flat earthers.
Even if Richard Dawkins or evolutionists want to argue that natural selection is actually a guided process because it selects the most favorite and all that.
They don't want to argue that. It's not guided, it's also not random.
The mutation process itself is an accidental copying mistake. That's what it is.
No, because accident implies a desired outcome, but that isn't part of DNA replication. So it's not a mistake, it's a change that happens stochastically. It's only an accident by analogy.
I mean, you can use all sorts of different terms, but when you get down to brass tacks, that's what it is. And evolutionary scientists are using these phrases themselves. So,
yes, and like I said, they shouldn't, in my opinion. But what they're not doing is using them dishonestly, just unwisely. That's a big difference.
Carl Sagan, right?
Carl Sagan, yeah, we can read that quote. That's in the rebuttal. You can go to creation.com/QE and go to the rebuttals there.
He said, mutations occur at random and are most uniformly harmful.
Which is wrong. They're most uniformly neutral. But then Sagan wasn't a biologist, so I'll forgive him for getting some details wrong.
It is rare that a precision machine is improved by a random change in the instructions for making it.
This is from the book Dragons of Eden. Let's read more of the context. Accidentally useful mutations provide the working material for biological evolution, as for example a mutation for melanin in certain moths, which changes their color from white to black. Such moths commonly rest on English birch trees where their white coloration provides protective camouflage. Under these conditions, the melanin mutation is not an advantage. The dark moths are starkly visible and are eaten by birds. The mutation is selected against, but when the Industrial Revolution began to cover the birch bark with soot, the situation was reversed and only moths with the melanin mutation survived. Then the mutation is selected for, and in time almost all the moths are dark, passing this inheritable change on to future generations. There are still occasional reverse mutations eliminating the melanin adaptation, which would be useful for the moths were English industrial pollution to be controlled. Note that in all this interaction between mutation and natural selection, no moth is making a conscious effort to adapt to a changed environment. The process is random and statistical. Large organisms such as human beings average about 1 mutation per 10 gametes. That is, there is a 10% chance that any given sperm or egg cell produced will have a new and inheritable change in the genetic instructions that determine the makeup of the next generation. These mutations occur at random and are almost uniformly harmful. It is rare that a precision machine is improved by a random change in the instructions for making it. Most of these mutations are also recessive. They do not manifest themselves immediately. Nevertheless, there is already such a high mutation rate that, as several biologists have suggested, a larger complement of genetic DNA would bring about unacceptably high mutation rates. Too much would go wrong too often if we had more genes. If this is true, there must be a practical upper limit to the amount of genetic information that the DNA of larger organisms can accommodate. Thus, large and complex organisms, by the mere fact of their existence, have to have substantial resources of extragenetic information. That information is contained in all higher animals except man, almost exclusively in the brain. This is about how the pace of human cultural evolution is so much faster than biological evolution. The point of the quote is not that beneficial mutations are too rare for evolution, it's that they're too rare to account for the changes in human society over the past few thousand years. In fact, earlier Sagan uses beneficial mutations to explain biological information, so he clearly thinks that they can do that. Can you believe that once again we have a dishonest quotation?
Well, he's an evolutionist. He believes evolution occurred through that process, but he's admitting, okay, why is he admitting
He's using
that?
those same terms.
Right, and he's admitting it because that's what we're observing.
No, you're a fucking liar, Calvin. He's saying it because of the fact that biological evolution cannot explain how humans went from hunter-gatherers to putting people on the moon in the span of fewer than 10,000 years. He's contrasting the relative slowness of biological evolution and his alleged information maximum to the rapidity of cultural evolution. He is in no way saying that beneficial mutations are too slow to account for biological evolution.
How can random changes be anything but accidental?
Right.
And
try it on your car motor while it's running and
random.
see how that works for you.
Yeah, because car motors work just like biological organisms.
Anyway, so far our
show has been a bunch of lies and reading assignments that did nothing to help our case? Is that what he's going to say?
Our evolutionist friends have not really come up with any really good answers for the questions that we're posing.
Yes, they have, but it doesn't matter, because these two and those like them aren't interested in answers and wouldn't accept any answer no matter how good, because evidence, logic, and reason aren't important to them. What's important to them is being right about God, which they are also not, but whatever.
Yeah, the 15 questions that are in the Question Evolution flyer, the responses that we're getting back don't really address the legitimate problems that are listed in this flyer.
Well, there don't seem to be any legitimate problems in that flyer, so that's not surprising, but that's it. We're finally done. I might go collapse into a heap now. This was far worse than I was expecting. Thanks again to Phil Rivara for all his work on this. He might have done more work than I did. If you liked this video, please hit like and let me know what you liked in the comments. If you didn't, then hit dislike and let me know what the problem was. Either way, I hope you subscribe and check out the channel memberships and my Patreon. I'm the Dapper Dinosaur. Hey, before you leave, I just want to take a second to thank my patrons and channel members, especially those pledging $20 or above. Ben Tovand, Tapioca Weasel, Sphincter of Doom, Regios, Landon Knoll, Ian Chen, Elleron Teller, Phil Rivera, Kelvin Rostick Van Manen, David Hercules, Joshua Geese, Vollus, Dan Sheedy, Pega Jace, The Plastic Mouse, Arrow Dodger, Gordon Love Science, Amber Athelian, Prixie Purple, Agricola Siqua, and John Ackerman. It's because of my channel members and patrons whom you're seeing on screen that this channel can stay afloat. Without you, it would all shut down. If you want to join the team, there's a link to join the channel below this video, and there's a link to join the Patreon in the description. On the Patreon, you can get a 15% discount for pledging annually, and either way, you get early access to virtually all of my scripted videos months before they come out for the general public. Thanks for watching.